Somewhere within the spiral of a single molecule, a hydrogen atom waits in two places at once, a quantum ghost, suspended between positions, and in that suspension lies the power to rewrite the instructions of life itself. This is the strange and quiet territory where physics meets biology, where the smallest uncertainties ripple outward into the vast story of evolution, of illness, of adaptation across millions of years. The double helix, so often imagined as a stable ladder, is in truth a place of constant quantum negotiation, protons tunneling through barriers that should be impassable, electrons flickering between states, and in those flickers, the possibility of mutation or repair. If these unhurried explorations help carry you toward rest, subscribing means more quiet nights like this one. Now, settling into stillness, the story can unfold from here. In the early decades of the 20th century, as physicists were still coming to terms with the strange new world that quantum mechanics had revealed to them, a few curious minds began to wonder whether the peculiar laws governing atoms and subatomic particles might also play some role in the processes of life itself. This was, at the time, a rather speculative notion, as biology and physics were considered quite separate disciplines, each with its own methods and its own concerns, and the idea that the mathematics describing electrons and photons might have something meaningful to say about the workings of living cells struck many as fanciful at best. Yet there were those who sensed that the boundary between the living and the non-living might not be so firmly drawn as conventional wisdom suggested. And among the first to voice this suspicion was a German theoretical physicist named Pascual Jordan, who in 1932 published a series of papers proposing that quantum mechanical effects might be essential to understanding how genes function and how mutations arise. Jordan was a complicated figure in the history of physics, brilliant and productive in his scientific work yet troubled in other aspects of his life and career, and his contributions to quantum biology have sometimes been overlooked or approached with caution for reasons that had little to do with the science itself. Nevertheless, his early speculation that the amplification of quantum events at the molecular level might be responsible for genetic change was remarkably prescient, even if his specific proposals did not survive scrutiny in their original form. He imagined that the indeterminacy inherent in quantum mechanics, the fundamental uncertainty about the position and momentum of particles at the smallest scales, might somehow be magnified through biological processes until it produced observable effects at the level of whole organisms. A single quantum event, he suggested, might trigger a cascade of consequences that would ultimately manifest as a heritable change in the characteristics of a living creature. And in this way the randomness at the heart of quantum theory might be connected to the randomness that seemed to characterize mutation and, by extension, evolution itself. Jordan's ideas were met with interest in some quarters but remained largely undeveloped, in part because the tools to investigate them simply did not exist, and in part because the structure of genetic material was still entirely unknown. It would take another decade, and the peculiar circumstances of a physicist in exile, before these speculations would be taken up again and presented to a wider audience in a form that would prove enormously influential. Erwin Schrödinger, the Austrian physicist who had given his name to the famous equation describing the behavior of quantum systems, found himself in Dublin during the Second World War, having fled the turmoil of continental Europe. There, at the Institute for Advanced Studies, he delivered a series of public lectures in 1943 that were published the following year as a slim volume entitled What is Life?, with a question mark that invited readers to consider how physics might approach the deepest mysteries of biology. The book was written in an accessible style, intended for a general educated audience rather than specialists, and yet its impact on the scientific community, particularly on young researchers seeking new problems to solve, was profound and lasting. In What is Life?, Schrödinger asked how the gene, whatever it might turn out to be at the molecular level, could possibly be stable enough to carry hereditary information across generations while also being capable of occasional sudden changes that would be faithfully copied in all subsequent descendants. He noted that the gene must be quite small, perhaps containing only a few hundred or a few thousand atoms, and yet it seemed to behave with a regularity and permanence that one would not normally expect from such a tiny collection of particles. At ordinary temperatures, small groups of atoms are subject to constant thermal fluctuation, jostled about by the random motion of their neighbors, and one might think that any information stored in such a fragile arrangement would quickly be scrambled beyond recognition. Yet genes persisted, sometimes for millions of years, passing from parent to offspring with remarkable fidelity, and this stability demanded explanation. Schrödinger proposed that the gene must be a kind of crystal, but not the regular repeating crystal familiar from minerals and salts, with the same unit repeated over and over in a monotonous pattern. Instead, he suggested that the genetic material might be what he called an aperiodic crystal, a structure in which the atoms are arranged in a definite and stable configuration, held in place by strong chemical bonds, but in which the precise sequence of elements varies along the length of the molecule in a way that encodes information. Just as a simple code might convey complex messages through different arrangements of a small number of symbols, so an aperiodic crystal might carry the instructions for building and maintaining a living organism through the specific order of its constituent parts. This was a remarkably accurate intuition, considering that the structure of DNA would not be determined for another 9 years. And when James Watson and Francis Crick finally worked out the double helix in 1953, they acknowledged the influence of Schrödinger's book on their thinking, as did many others who entered the new field of molecular biology in the decades that followed. What is Life also touched on the quantum mechanical aspects of mutation, though in a somewhat tentative way, suggesting that the transition from one stable configuration of atoms to another might involve the kind of discrete jumps that characterize quantum systems. Schrödinger invoked the idea of quantum mechanical stability to explain why the gene did not gradually drift from one state to another under the influence of thermal noise, but instead remained fixed in one configuration until a sudden and relatively rare event caused it to switch to a different arrangement. This switching, this mutation, was imagined as a quantum transition, a jump over an energy barrier that separated one stable state from another, and once the jump had occurred, the new configuration would be just as stable as the old, faithfully replicated in all future copies. The details of how such transitions might occur were left vague, as indeed they had to be given the state of knowledge at the time. But the suggestion that quantum mechanics might be directly involved in the most fundamental processes of life was planted firmly in the scientific imagination. For nearly 2 decades after the publication of What is Life?, the question of whether quantum effects played any significant role in biological processes remained largely speculative, a topic for occasional discussion but not for rigorous investigation. The structure of DNA had been determined, the genetic code had been cracked, and the machinery of protein synthesis was being mapped out in ever greater detail. Yet all of this progress was achieved using the concepts and methods of classical chemistry and biochemistry, without any obvious need to invoke the peculiar features of quantum mechanics. The atoms in biological molecules were certainly governed by quantum laws at the most fundamental level, as all atoms are, but it was widely assumed that these quantum effects averaged out in the warm, wet environment of the living cell, lost in the thermal noise and the constant collisions with surrounding water molecules, so that for practical purposes the behavior of biological systems could be described in purely classical terms. It was in this context that a Swedish theoretical chemist named Per-Olof Lodin, working at Uppsala University in Sweden, published a paper in 1963 that would eventually be recognized as a turning point in the history of quantum biology, though at the time it received relatively little attention and was greeted with considerable skepticism by many of his colleagues. Lodin was interested in the fundamental chemistry of DNA, and in particular in the hydrogen bonds that hold the two strands of the double helix together, linking the complementary base pairs, adenine with thymine and guanine with cytosine, through a pattern of shared hydrogen atoms. These hydrogen bonds are weaker than the covalent bonds that form the backbone of each strand, which is why the two strands can be separated during replication and transcription, but they are strong enough to maintain the integrity of the double helix under normal conditions, and the specificity of the base pairing, with adenine always facing thymine and guanine always facing cytosine, is what ensures that the genetic information is accurately copied. Lodin noticed that the hydrogen atoms in these bonds occupy particular positions, closer to one base or the other, and that these positions correspond to one of two possible arrangements, which he called the normal form and the Tautomeric form. In the normal form, the hydrogen is attached to the expected nitrogen or oxygen atom, and the base pairs in the standard way. In the Tautomeric form, the hydrogen has shifted to a different position, and the base can now pair with a different partner, leading to a mismatch that, if not corrected, will result in a mutation when the DNA is copied. The existence of tautomeric forms had been known for some time, and it had been suggested that rare tautomers might be responsible for some spontaneous mutations, but the mechanism by which a base might switch from one form to another was not well understood. Lodin's insight was that the transition from the normal form to the tautomeric form might occur through quantum tunneling, a process in which a particle passes through an energy barrier that it would not have enough energy to surmount according to the classical laws of physics. In quantum mechanics, particles such as the proton that constitutes the nucleus of a hydrogen atom do not have definite positions and momenta, but are instead described by a wave function that gives the probability of finding the particle at any given location. This wave function does not stop abruptly at the edge of an energy barrier, but instead extends into and through the barrier, decaying exponentially with distance, so that there is always some small but non-zero probability of the particle being found on the other side. If the barrier is thin enough and the particle light enough, this probability can become significant, and the particle may effectively tunnel from one side to the other without ever having occupied the intermediate positions that a classical particle would have to traverse. Lodin calculated that the protons in the hydrogen bonds of DNA base pairs might be capable of tunneling from their normal positions to the tautomeric positions, and that the timescale for such tunneling events might be extraordinarily short, on the order of 10 to the power of -13 seconds, or about 1/10 of a picosecond. This is far faster than the timescales of most biochemical processes, and it suggested that tunneling could occur frequently enough to produce a small but steady supply of tautomeric bases, which would then be available to cause mutations during DNA replication. The probability of any given base being in the tautomeric form at any given moment would be very small, since the normal form is energetically favored, but over the course of a cell's lifetime, with billions of base pairs and countless opportunities for replication, Even a tiny probability could translate into a significant mutation rate. The scientific community did not immediately embrace Lodin's proposal, and for good reason, as the theory made predictions that were difficult to test with the experimental techniques available at the time, and there were many other potential sources of mutation that seemed more straightforward to investigate. Radiation, chemical mutagens, errors by the enzymes responsible for copying and repairing DNA. All of these could account for observed mutation rates without invoking quantum effects, and many biologists were understandably reluctant to add the complexities of quantum mechanics to an already challenging field. Moreover, there was a widespread intuition, sometimes made explicit and sometimes simply assumed, that the warm, wet environment of the living cell was fundamentally hostile to quantum effects, which were thought to require the cold, isolated conditions of a physics laboratory to manifest themselves. Coherent quantum states, the reasoning went, would be destroyed almost instantaneously by interactions with the surrounding molecules, a process known as decoherence, and so any quantum tunneling that might occur in DNA would be washed out long before it could have any biological consequences. Lodin himself remained convinced that quantum effects were important in biology, and he continued to develop his ideas over the following decades, both in his own research and in his encouragement of others to pursue this line of inquiry. He wrote and spoke about DNA as being a quantum mechanical document, a phrase that captured his belief that the genetic information was not merely stored in the positions of atoms, but was fundamentally governed by the same probabilistic laws that described electrons in atoms and photons in light beams. The double helix, in his view, was not a static structure but a dynamic one, with protons constantly flickering between different positions, their behavior determined not by classical mechanics but by the strange rules of quantum theory, and the mutations that drove evolution were not simply accidents caused by external damage but were in some sense built into the very fabric of the molecule. For many years, Lodin's ideas remained at the margins of mainstream biology, cited occasionally but rarely pursued in detail, a tantalizing possibility that lacked the experimental support needed to compel widespread acceptance. The gap between theoretical speculation and experimental verification is often long in science, sometimes spanning decades, and the quantum biology of DNA was no exception. The techniques needed to observe proton tunneling in biological molecules, to measure the populations of tautomeric forms, to trace the quantum mechanical origins of individual mutations, simply did not exist in the 1960s or 70s or even 80s. Researchers could calculate what might be happening at the quantum level, could build models and run simulations, could argue about the plausibility of various mechanisms, but they could not directly observe the tunneling events themselves, and without such observations the debate remained somewhat abstract. It was not until the late 20th and early 21st centuries that advances in experimental physics and chemistry began to provide the tools needed to test Lodin's proposals. Ultrafast spectroscopy, which uses pulses of light lasting only femtoseconds to probe the dynamics of molecules, made it possible to track the movements of protons on timescales that had previously been inaccessible. Isotope substitution experiments, in which hydrogen atoms are replaced by heavier deuterium atoms, provided a way to test for the signature of tunneling, since the heavier isotope should tunnel more slowly and produce measurably different results. Computational methods grew powerful enough to simulate the quantum behavior of DNA base pairs with increasing accuracy, allowing theoretical predictions to be compared with experimental observations. And gradually, piece by piece, Evidence began to accumulate that Lodin had been onto something important all those years ago. That proton tunneling does indeed occur in DNA, and that it may indeed contribute to the spontaneous mutations that provide the raw material for evolution. The story of how physicists first suspected that quantum effects might be at work in the molecules of life is, in many ways, a story of patient waiting and persistent curiosity. From Pascual Jordan's early speculations in the 1930s, through Schrödinger's influential musings in the 1940s, to Lodin's detailed proposals in the 1960s and the slow accumulation of evidence in the decades that followed, the idea that quantum mechanics might be essential to understanding heredity and mutation has been a thread running through the history of science, sometimes prominent and sometimes nearly invisible, but never entirely abandoned. The ghost in the molecule, the quantum uncertainty at the heart of the gene, has proven to be an elusive quarry, difficult to observe and even more difficult to characterize. But the hunt continues, and each new experiment brings us a little closer to understanding how the strange laws of the very small might shape the processes of life itself. The story of how we came to understand the structure of deoxyribonucleic acid, which we commonly call DNA, begins in earnest in the middle of the 20th century, during a period when several brilliant minds were converging upon the same fundamental question, each approaching it from slightly different angles and with slightly different tools at their disposal. In the early months of 1953, two young scientists working at the Cavendish Laboratory in Cambridge, England, a physicist named Francis Crick and a biologist named James Watson, constructed a physical model of the DNA molecule that would prove to be essentially correct in its major features. And this achievement, which built upon the work of many others, would eventually earn them a share of the Nobel Prize in Physiology or Medicine. Their model proposed that DNA consists of two long strands wound around each other in a gentle spiral, forming what we now recognize as the iconic double helix, a structure that has become perhaps the most famous molecular shape in all of biology, reproduced countless times in textbooks and museums and works of popular science. Yet the Watson and Crick model did not emerge from nothing, and it is important to remember, as we settle into this quiet exploration, that scientific discoveries rarely belong entirely to any single individual or pair of individuals, but rather arise from a broader community of researchers, each contributing pieces to a larger puzzle. One of the most crucial pieces of evidence that guided Watson and Crick toward their correct structure came from the laboratory of physicist and X-ray crystallographer Rosalind Franklin, who was working at King's College London along with her colleague Maurice Wilkins. Franklin was exceptionally skilled at the demanding technique of X-ray crystallography, which involves directing beams of X-rays at carefully prepared samples and then analyzing the patterns that emerge when those X-rays are diffracted by the atoms within the sample. In May of 1952, Franklin and her graduate student Raymond Gosling obtained a particularly clear X-ray diffraction image of DNA, an image that became known as Photograph 51. And this single image contained within its pattern of dark spots and streaks the essential information needed to determine that DNA had a helical structure. The significance of Photo 51, as it is often called, cannot be overstated, for it displayed a distinctive X-shaped pattern that is characteristic of helical molecules, and from the spacing and intensity of the spots within that pattern, one could deduce important measurements about the dimensions of the helix itself. Franklin herself was cautious in her interpretations, as a careful scientist ought to be, but the image nonetheless made its way to Watson, shown to him by Wilkins without Franklin's knowledge or consent, and it provided critical confirmation that the helical model they had been developing was on the right track. Franklin's contribution has not always received the recognition it deserves, for she passed away from ovarian cancer in 1958 at the age of only 37, several years before the Nobel Prize was awarded to Watson, Crick, and Wilkins in 1962, and since the Nobel is not awarded posthumously, her name was absent from that honor, though her work was absolutely essential to the discovery. Now, as we begin to consider the structure of DNA through a quantum mechanical lens, which is to say through the lens of physics at its most fundamental and subtle level, we must first establish what that structure actually consists of, for it is in the fine details of molecular architecture that quantum effects become relevant. The two strands of the double helix are composed of long chains of smaller units called nucleotides, and each nucleotide contains three components, which are sugar molecule, a phosphate group, and one of four possible nitrogenous bases. The sugar and phosphate groups alternate to form the backbone of each strand, creating a kind of molecular ribbon that winds around the outside of the helix, while the bases project inward toward the center, where they meet and pair with the bases extending from the opposite strand. It is through these base pairs that the two strands are connected, and the connections are made by means of hydrogen bonds, which are a particular type of weak chemical bond that will prove to be of great interest to us as we explore quantum effects in DNA. The four bases found in DNA are adenine, thymine, guanine, and cytosine, and they pair together in a very specific way that was part of what made the Watson and Crick model so elegant and so immediately suggestive of how genetic information might be copied and transmitted. Adenine pairs with thymine, and guanine pairs with cytosine, so that wherever one strand has an adenine, the opposite strand will have a thymine, and wherever one strand has a guanine, the opposite strand will have a cytosine. This complementary base pairing is enforced by the geometry of hydrogen bonding, for an adenine-thymine pair is held together by two hydrogen bonds, while a guanine-cytosine pair is held together by three hydrogen bonds, and these numbers are fixed by the positions of hydrogen atoms and electronegative atoms on each base. The hydrogen bonds span distances of roughly 1.8 to 2.0 angstroms, where an angstrom is a unit of length equal to 1/10,000,000th of a meter, or 10 to the power of -10 meters, which is to say an almost inconceivably small distance, smaller than the wavelength of visible light by a factor of thousands. To understand why hydrogen bonds are of such interest from a quantum mechanical perspective, we must consider what a hydrogen bond actually is and what particles are involved in its formation. A hydrogen bond occurs when a hydrogen atom, which is covalently bonded to an electronegative atom such as nitrogen or oxygen, experiences an attractive force toward another electronegative atom nearby. The hydrogen atom consists of a single proton in its nucleus and a single electron in its orbital, making it the lightest and simplest of all atoms. And this lightness is significant because quantum mechanical effects become more pronounced for lighter particles. The proton at the heart of each hydrogen atom has a mass roughly 1,800 times smaller than that of the electrons in heavier atoms combined with their nuclei, but it is still a particle with mass, and according to quantum mechanics, all particles with mass exhibit wave-like properties described by a mathematical function called the wave function, which gives the probability of finding the particle at any particular location. For heavier particles, this wave-like behavior is negligible for all practical purposes, and we can treat atoms and molecules as classical objects with definite positions and momenta. But for very light particles such as protons, especially when they are confined to the small spaces characteristic of chemical bonds, the quantum wave nature becomes significant, and the proton cannot be thought of as simply sitting at one fixed point in space. Instead, the proton must be described by its wave function, which spreads out over some small region, and there is some probability of finding the proton at any point within that region. This delocalization of the proton, as it is called, becomes particularly interesting in the context of hydrogen bonds, where the proton sits between two electronegative atoms and might, under certain circumstances, shift its position from being closer to one atom to being closer to the other. This process, known as proton tunneling or proton transfer, is a purely quantum mechanical phenomenon with no classical analog, and as we shall explore in later sections, it may have profound implications for the stability and mutability of genetic information. Before Watson and Crick assembled their model, and indeed before Franklin captured her famous photograph, a biochemist named Erwin Chargaff had made careful measurements of the chemical composition of DNA from various organisms, and he discovered a pattern that came to be known as Chargaff's rules. What Chargaff found was that in any sample of DNA, the amount of adenine was always approximately equal to the amount of thymine, and the amount of guanine was always approximately equal to the amount of cytosine, regardless of which organism the DNA came from. This one-to-one relationship between adenine and thymine, and between guanine and cytosine, was a crucial clue pointing toward the base pairing rules that Watson and Crick would later incorporate into their model. For if adenine always pairs with thymine, then of course the two must be present in equal amounts, and similarly for guanine and cytosine. From a quantum mechanical perspective, Chargaff's rules reflect the fact that the hydrogen bonding patterns between base pairs are determined by the electronic structures of the bases themselves, which is to say by the quantum mechanical arrangement of electrons within each molecule. The reason adenine pairs with thymine rather than with guanine is not arbitrary but follows from the positions of hydrogen bond donors and acceptors on each base, which are in turn determined by the distribution of electron density within the molecules. And this electron density distribution is a quantum mechanical property arising from the solutions to the Schrödinger equation for the electrons in each base. The specificity of base pairing, which is so essential to the storage and transmission of genetic information, is thus ultimately a consequence of quantum mechanics, even though it is often discussed in terms of classical chemistry. The bases in DNA are flat, planar molecules, and when they stack on top of one another along the length of the double helix, their ring structures overlap in a way that creates additional stabilizing interactions beyond the hydrogen bonds between base pairs. These interactions arise from the pi electrons in the aromatic rings of the bases, which are electrons that occupy molecular orbitals extending above and below the plane of each base. And when two bases are stacked with their planes parallel and separated by a distance of about 3.4 angstroms, these pi electron clouds interact in a way that lowers the overall energy of the system. This phenomenon is known as pi-pi stacking or base stacking, and it contributes significantly to the overall stability of the DNA double helix, helping to keep the structure intact even as it flexes and bends and unwinds during the various processes that occur within a living cell. The pi electrons involved in base stacking are described by quantum mechanical wave functions that extend over the entire ring system of each base, and there has been considerable interest in recent years in whether these electrons might exhibit quantum coherence across multiple bases, which is to say whether the quantum states of electrons in one base might be correlated with the quantum states of electrons in neighboring bases in a way that persists over time. Quantum coherence is a property that allows quantum systems to exist in superpositions of states and to exhibit interference effects, and it was long thought to be too fragile to survive in the warm, wet environment of living cells, where thermal fluctuations and interactions with surrounding molecules would be expected to destroy any coherence almost instantaneously. However, experiments in other biological systems, particularly in photosynthetic complexes, have suggested that quantum coherence might persist for longer than expected, and some researchers have begun to explore whether similar effects might be relevant in DNA, though this remains an active area of investigation with many open questions. The 3.4 angstrom spacing between adjacent base pairs, which we mentioned in the context of pi-pi stacking, is one of the key dimensions that define the geometry of the DNA double helix, and it was one of the measurements that could be extracted from Franklin's X-ray diffraction data. This spacing represents the distance along the helical axis from one base pair to the next, and it is determined by the balance between the attractive pi stacking interactions and the repulsive forces that arise when electron clouds get too close together. The helix makes a complete turn, returning to the same rotational position, after approximately 10.5 base pairs, which gives what is called the helical pitch, and this means that the helix rises by about 34 angstroms, or 3.4 nanometers, for each complete turn. The double helix also has two grooves that wind along its length, one wider and one narrower, which are called the major groove and the minor groove respectively, and these grooves are important because they provide surfaces where proteins and other molecules can interact with the DNA and read the sequence of bases without needing to unwind the helix. The major and minor grooves arise because the two strands of the helix are not directly opposite each other but are slightly offset, and the patterns of electron density exposed in each groove are different, with the major groove generally providing more information about the identity of the base pairs than the minor groove. Proteins that need to recognize specific DNA sequences often do so by inserting portions of themselves into the major groove, where they can make contact with the edges of the base pairs and detect the unique pattern of hydrogen bond donors and acceptors that characterizes each type of base pair. The electron density distributions that determine what a protein sees when it examines the major groove are quantum mechanical properties of the bases, arising from the shapes and occupancies of the molecular orbitals. And so even this aspect of DNA structure, which is crucial for gene regulation and many other biological processes, has its roots in quantum physics. It is worth noting that the correct structure of DNA was not immediately obvious to everyone working on the problem, and indeed one of the most accomplished scientists of the 20th century, the chemist and physicist Linus Pauling, proposed an incorrect model shortly before Watson and Crick announced their double helix. Pauling had already made enormous contributions to the understanding of chemical bonds and molecular structure, including the discovery of the alpha helix, which is a key structural element in proteins, and he was widely regarded as the leading authority on the application of quantum mechanics to chemistry. His model for DNA proposed a triple helix, with 3 strands wound around each other and the phosphate groups facing inward toward the center of the structure, which would have placed the negatively charged phosphates in close proximity and created severe electrostatic repulsion. The error was uncharacteristic of Pauling and may have resulted partly from working with outdated or incomplete data, and Watson and Crick were aware of his efforts and were motivated to move quickly in part because they knew that such a formidable competitor was working on the same problem. The failure of Pauling's triple helix model, despite his mastery of quantum chemistry and molecular structure, serves as a gentle reminder that scientific progress is rarely straightforward and that even the most brilliant researchers can be led astray by incomplete information or by the natural human tendency to see patterns that fit our expectations. The correct double helix model emerged not from a single flash of insight but from the careful accumulation of evidence from multiple sources, including the X-ray crystallography of Franklin and Wilkins, the base composition rules of Chargaff, and the chemical knowledge of how hydrogen bonds form and what distances and angles they prefer. And this double helix, with its two strands of complementary bases connected by hydrogen bonds spanning approximately 2 angstroms, with its pi electron stacking interactions separated by 3.4 angstroms, with its major and minor grooves exposing patterns of electron density, with its graceful helical turn completed every 10.5 base pairs. This structure is where the quantum mechanical nature of matter meets the biological phenomenon of heredity, and it is this meeting that we will continue to explore as we proceed through the quiet hours ahead. In the year 1953, 2 young scientists sat in Cambridge, England, surrounded by cardboard cutouts and wire models, trying to work out how the molecule of heredity might copy itself. James Watson and Francis Crick had just proposed their famous double helix structure for deoxyribonucleic acid, and in their landmark paper, tucked away almost as an afterthought, they made a quiet speculation that would take decades to fully appreciate. They suggested that the bases of DNA might occasionally exist in alternative chemical forms, rare structural variants that could cause the wrong bases to pair together during replication, and that these mispairings might be the molecular origin of mutations. It was a gentle aside in a paper that changed biology forever, and yet this small observation opened a door into one of the most fascinating intersections of quantum physics and molecular genetics that we are still exploring today, here in these quiet hours as we settle into rest. The 4 bases of DNA, the adenine and guanine, the cytosine and thymine, are the letters of the genetic alphabet, and in their normal everyday forms they pair together in the familiar Watson-Crick manner. Adenine reaches across the helix to form 2 hydrogen bonds with thymine, and guanine extends 3 hydrogen bonds to clasp cytosine, and this complementary pairing is what allows DNA to be copied so faithfully, one strand serving as the template for its partner. But these bases are not frozen sculptures of atoms. They are dynamic molecules whose hydrogen atoms can, under certain circumstances, shift their positions ever so slightly, moving from one location to another within the same molecule. And when this happens, the base takes on a different shape, a different arrangement of its bonds, which we call a tautomeric form. The word tautomer comes from the Greek for same part, referring to how these alternative forms contain exactly the same atoms but arranged in a different bonding pattern, and you might picture it like a gentle rearrangement of furniture in a familiar room, where everything is still present but the chairs have moved to different corners. The normal forms of the DNA bases, the ones that predominate overwhelmingly in the double helix, are called the canonical forms, and they exist in what chemists describe as particular electronic states. Guanine and thymine, in their canonical forms, contain a chemical arrangement called a chaeto group, where an oxygen atom is double bonded to a carbon, and this chaeto form is the shape these bases almost always take. Adenine and cytosine, meanwhile, contain amino groups in their canonical forms, where a nitrogen atom carries 2 hydrogen atoms and connects to the ring structure of the base in a particular way. But here is where the shape-shifting begins, and you can let your breathing slow as we explore this gentle molecular transformation. The hydrogen atoms in these bases can sometimes migrate, hopping from one position to another, and when this happens to guanine or thymine, the chato group transforms into what is called an enol group, where the hydrogen has moved to sit on the oxygen atom, changing the double bond into a single bond and creating a hydroxyl arrangement instead. This chato-enol tautomerism, as chemists call it, is a subtle shift, a quiet rearrangement that changes the shape of the base just enough to alter which partner it prefers to bind with. Similarly, adenine and cytosine can undergo amino-amino tautomerism, where one of the hydrogen atoms on the amino group migrates to a different nitrogen atom in the ring, converting the amino group into an amino group. And again, this gentle shift changes the hydrogen bonding face of the molecule, making it suddenly compatible with a different partner than usual. The frequencies of these rare tautomeric forms are very low. Perhaps only one base in 10,000 or 1 in 100,000 might exist in the alternative form at any given moment. And yet when we consider that a human genome contains some 3 billion base pairs, and that DNA replication involves copying every single one of these letters, even such a small probability begins to matter across the vast landscape of genetic information. The bases spend the overwhelming majority of their time in their canonical forms, contentedly pairing in the correct Watson-Crick manner. But every so often, briefly, fleetingly, one will slip into its tautomeric state. And if this happens to occur at just the moment when the replication machinery is reading that particular position, a mispairing can result, and a mutation may be born. Now we come to a remarkable idea that was proposed in 1963 by a Swedish physicist named Per-Olof Lodin, and his suggestion brought the strange world of quantum mechanics directly into the heart of the double helix. Lodin was interested in the hydrogen bonds that hold the two strands of DNA together, those gentle bridges where a hydrogen atom sits between two electronegative atoms, linking adenine to thymine or guanine to cytosine. He proposed that the hydrogen atoms in these bonds might not behave like classical particles, sitting in one fixed position, but might instead exhibit quantum behavior, specifically the phenomenon known as quantum tunneling. In quantum tunneling, a particle can pass through an energy barrier that classical physics would say is impassable, appearing on the other side without ever having had enough energy to climb over the top. And Lodin suggested that protons, which are the nuclei of hydrogen atoms, might tunnel along the hydrogen bonds between paired bases, shifting from one side to the other and thereby creating tautomeric forms. Picture a small hill between two valleys, and imagine a ball resting in one valley. In the classical world of everyday experience, the ball would need a push strong enough to lift it over the crest of the hill before it could roll down into the other valley. But in the quantum realm, there is a small probability that the ball might simply appear in the other valley, having tunneled through the hill as if it were partly transparent. This is not magic but simply the mathematics of quantum mechanics, where particles are described by wave functions that can extend through barriers, and there is always some probability, however small, of finding the particle on the other side. For protons in DNA, the energy barrier they would need to tunnel through has been estimated at roughly 0.4 electron volts, which is not a large barrier by quantum standards. And Lodin calculated that such tunneling events could occur at biologically meaningful rates. If a proton tunnels from its normal position on one base to a new position on its partner, or shifts within a single base, the result can be the creation of a tautomeric form. And if this happens at the moment of replication, the base may pair with the wrong partner. The replication machinery of the cell reads the template strand and selects complementary bases to build the new strand, and it does this largely by checking the hydrogen bonding pattern, the shape of the base faces that come together across the helix. A tautomeric base presents a different face, a different pattern of hydrogen bond donors and acceptors, and the replication enzymes may be fooled into inserting a base that would normally be considered a mismatch. In 1976, researchers named Topol and Fresco published a careful analysis of how tautomeric forms could give rise to specific mutation patterns, examining which bases in their rare forms would preferentially pair with which normal partners. Their work showed that the mutations caused by tautomeric mispairings would tend to follow particular patterns, especially the class of mutations known as transitions, where a purine base is replaced by another purine, or a pyrimidine by another pyrimidine. One particularly important pathway they identified involves the enol form of cytosine, which rather than pairing with its normal partner guanine, can instead form hydrogen bonds with ADE9, and when this mispairing is propagated through subsequent rounds of replication, the result is a transition from a GC base pair to an AT base pair. This GC to AT transition is in fact one of the most common types of point mutation observed in nature, and the tautomeric mechanism provides an elegant explanation for why this particular change occurs so frequently. The enol form of cytosine, UC, has shifted its hydrogen arrangement so that it presents a face resembling thymine, the base that normally pairs with ADE9, and so when the replication machinery encounters an enol cytosine in the template strand, it may place an ADE9 in the new strand, thinking it has found a thymine. In the next round of replication, that ADE9 will pair normally with a thymine, and the original GC pair will have become an AT pair, a permanent change in the genetic sequence. This is how the fleeting existence of a tautomeric form, lasting perhaps only microseconds or less, can become immortalized in the genome, passed down through all subsequent generations of cells and potentially affecting the organism in ways large or small. The challenge for scientists has long been that these tautomeric forms are so rare and so short-lived that detecting them directly in DNA has been extraordinarily difficult. The equilibrium between the canonical and tautomeric forms lies so heavily on the side of the canonical that finding the rare forms is like searching for a single particular grain of sand on a very long beach. And moreover, the tautomeric forms may convert back to their canonical shapes in mere picoseconds or nanoseconds, making them difficult to catch in the act. For many decades, the tautomeric theory of mutation remained a compelling hypothesis, supported by the logic of chemistry and the patterns of observed mutations, but without direct experimental confirmation of tautomers actually forming within DNA base pairs. In more recent years, however, advancing technology has begun to provide glimpses of these elusive shapeshifters. Between 2015 and 2022, researchers including Zoe Waller and colleagues at several universities have used sophisticated techniques to detect and characterize short-lived tautomeric forms in DNA structures. Using methods such as nuclear magnetic resonance spectroscopy, which can observe the positions and environments of individual atoms in molecules, and computational approaches that can model the quantum behavior of protons, these scientists have found evidence that tautomeric forms do indeed exist within DNA, albeit briefly and rarely. The experimental work has confirmed that the energy barriers and tunneling rates are in the range that Lodin predicted, and that the tautomeric forms have the mispairing properties that Topol and Fresco described, providing validation for ideas that were first proposed more than half a century ago. These studies have also revealed additional complexity and subtlety in the tautomeric system, showing that the rates of tautomer formation may depend on the surrounding sequence context, on the presence of water molecules, on the particular base pair involved, and on the dynamics of the DNA double helix as it breathes and flexes. The double helix is not a rigid structure but a gently undulating molecule that opens and closes its base pairs in a process called breathing, and these dynamics may influence when and how proton tunneling can occur, creating moments of opportunity when the energy barriers are lowered and tautomeric forms can more readily appear. What emerges from all of this research is a picture of mutation as a phenomenon that touches on the deepest levels of physical reality, where the quantum behavior of individual protons can have consequences that propagate up through the scales of biology to affect the traits of organisms and the course of evolution. The mutations that drive natural selection, that cause genetic diseases, that create the diversity of life, may trace their origins in part to events happening at the scale of single atoms tunneling through energy barriers in the hearts of molecules. It is a quietly astonishing connection, linking the equations of quantum mechanics to the pageant of life on Earth, and yet it unfolds so gently, so continuously, in every cell of every organism, that we might never notice it happening at all. The canonical forms of the bases remain overwhelmingly dominant, and the faithful replication of DNA proceeds with remarkable accuracy, with error rates of perhaps one mistake in a billion base pairs copied once the proofreading and repair systems have done their work. But the rare tautomeric forms provide a gentle, persistent source of variation, a quantum uncertainty built into the very chemistry of heredity. And this variation is the raw material upon which evolution acts. Without mutations, life would be frozen, unable to adapt to changing environments or explore new possibilities. And so these brief tautomeric excursions, these fleeting quantum events, play an essential role in the grand story of biology. As you rest now, you might find it soothing to think of the bases of DNA as patient molecules, spending almost all of their time in their familiar canonical forms, doing their job of encoding information and pairing faithfully with their partners, but occasionally, briefly, trying on an alternative shape, like a person stretching in a different direction for just a moment before settling back into their usual posture. These moments are so rare and so brief that the vast majority pass without consequence, the replication machinery moving on before any mispair can occur, or the proofreading enzymes catching and correcting any mistakes that do arise. But every now and then, in just the right circumstances, a tautomeric form persists long enough to cause a change, and a new mutation enters the genome, carrying with it the potential for new traits, new adaptations, or sometimes, new challenges for the organism to face. The science of tautomeric mutations continues to develop, with researchers using ever more sophisticated tools to study the quantum dynamics of protons in DNA, building computational models that can simulate these events at the level of individual atoms, and designing experiments that can detect the presence and behavior of rare tautomeric forms. Each new study adds detail to our understanding, refining the estimates of tunneling rates and tautomer lifetimes, mapping out the landscape of energy barriers, and clarifying how the molecular environment of the double helix influences the probability of these quantum events. It is slow, careful work, conducted in laboratories around the world by patient scientists who find beauty in the details of molecular behavior and satisfaction in the gradual accumulation of knowledge. And so we have traced the thread from Watson and Crick's original speculation in 1953, through Lodin's quantum mechanical proposal in 1963, to Topol and Fresco's analysis of mutation patterns in 1976, and onward to the recent experimental work that has at last provided direct evidence for the existence of tautomeric forms in DNA. It is a story that spans decades and disciplines, connecting the physics of the very small to the biology of the very complex, and it is a story that continues to unfold even now, as researchers probe ever deeper into the quantum nature of the molecules of life. As this section draws gently to a close, you might simply rest with the thought that the 4 letters of the genetic alphabet, those familiar bases adenine and guanine and cytosine and thymine, are not quite as fixed and immutable as they might first appear. They carry within them the possibility of transformation, the capacity to shift briefly into alternative forms that can give rise to new variations in the code of life. These tautomeric shapeshifters are rare and fleeting, but their effects accumulate over the vast timescales of evolution, contributing to the endless diversity of living things. The quantum world, with its tunneling protons and energy barriers, reaches gently into the double helix, and in this quiet meeting of physics and biology, we find one of the sources of genetic change that has shaped all life on Earth. And with this knowledge settling softly in your mind, you can let your thoughts drift now, breathing slowly, peacefully, as the science of DNA and its quantum intricacies fades into the gentle quiet of rest. And so we come now to a quieter part of our story, the part where scientists, patient and methodical, set about the careful work of catching something that does not wish to be caught, something that happens so quickly and so subtly that for many decades it was easier to assume it simply did not occur at all. We are speaking of the experimental hunt for proton tunneling, the search for evidence that the smallest particles in our bodies do not always follow the rules we learned in school, that they sometimes pass through barriers rather than over them, and that this ghostly behavior might be woven into the very fabric of how our cells work, how enzymes catalyze reactions, and perhaps even how the genetic code itself changes over time. The story of this hunt begins, as many good scientific stories do, with a researcher who noticed something peculiar, something that did not quite fit with the expected explanations, and who had the patience and insight to follow that peculiarity wherever it might lead. At the University of California, Berkeley, beginning in the 1980s and continuing for decades afterward, a biochemist named Judith Klinman began to ask careful questions about how enzymes manage to accelerate chemical reactions to the extraordinary speeds that living systems require. Enzymes, you will recall, are the molecular machines that make biology possible, proteins folded into precise shapes that bring reactive molecules together and coax them to transform in ways that would otherwise take far too long to sustain life. The question of how enzymes achieve their remarkable rate enhancements is one of the central puzzles of biochemistry, and Klinman approached it with a particular tool, a technique that would prove to be one of the most powerful methods for detecting the quantum behavior of atoms in biological systems, something called the kinetic isotope effect. To understand what the kinetic isotope effect tells us, we must first consider what isotopes are, and the concept is gentle enough to hold in mind as you settle deeper into your pillow. Isotopes are versions of the same element that differ only in the number of neutrons in their nucleus, the dense central part of the atom. Hydrogen, the lightest element, normally consists of just one proton and one electron, with no neutrons at all. But there exists a heavier form called deuterium, which has one proton and one neutron in its nucleus, making it roughly twice as heavy as ordinary hydrogen while remaining chemically almost identical. This difference in mass might seem negligible, but when it comes to the quantum behavior of particles, mass matters enormously, And this is where the kinetic isotope effect becomes such a revealing tool. When a chemical reaction involves breaking a bond to hydrogen, as many enzyme-catalyzed reactions do, replacing that hydrogen with deuterium will slow the reaction down, because the heavier deuterium moves more sluggishly than its lighter cousin. This slowing is expected from classical chemistry, the chemistry of particles moving over energy barriers like balls rolling over hills, and for purely classical reactions the ratio of reaction rates, hydrogen to deuterium, typically falls somewhere around 2 to 7, with 7 being considered something of an upper limit based on the different vibrational frequencies of the two isotopes. This ratio is what chemists call the kinetic isotope effect, often abbreviated as KIE, and for many years it was assumed that measured values should fall within this classical range. But Judith Klinman and her colleagues began to find reactions where the kinetic isotope effect was much larger than 7, values of 10, or 15, or even higher, numbers that could not be explained by classical mechanics alone. These anomalously large isotope effects suggested that something else was happening, that the hydrogen atoms were not simply climbing over energy barriers but were instead tunneling through them, passing through the quantum mechanical route that heavier deuterium atoms, with their shorter wavelengths, found much more difficult to traverse. The difference in tunneling probability between hydrogen and deuterium is far more pronounced than the difference in their classical behavior, because tunneling depends exponentially on both the mass of the particle and the width of the barrier, and even a doubling of mass can dramatically reduce the likelihood of quantum passage. This insight, that kinetic isotope effects exceeding the classical limit could serve as fingerprints of quantum tunneling, opened an entirely new avenue of investigation, a way of detecting quantum behavior in the warm, wet, seemingly inhospitable environment of living cells. Klinman's work on alcohol dehydrogenase and other enzymes revealed that these biological catalysts seem to be not merely tolerating quantum effects but actively exploiting them. Their protein structures apparently evolved to position atoms in just the right configurations to facilitate tunneling, conformations that Klinman would later describe as tunneling-ready, molecular arrangements that minimize the barrier width and maximize the probability of quantum passage. Across the Atlantic, at the University of Manchester, a group of researchers led by Nigel Scruton took up this thread and pushed it further, examining enzyme systems that displayed even more dramatic signatures of quantum behavior. Scruton and his colleagues, working through the late 1990s and into the 2000s, studied a class of enzymes that process amine compounds, and what they found was remarkable: kinetic isotope effects that reached values of 50 or more, numbers so far beyond the classical limit that there was essentially no way to explain them without invoking quantum mechanical tunneling as a major component of the reaction mechanism. These findings were not merely curiosities, not simply strange numbers to puzzle over, but fundamental challenges to the way biochemists understood enzyme catalysis. If quantum tunneling could contribute so significantly to reaction rates, then the entire field would need to reconsider how it thought about biological chemistry, how it modeled enzyme mechanisms, and how it designed drugs and industrial catalysts. The Manchester work, alongside Klinman's continuing studies at Berkeley, established beyond reasonable doubt that quantum effects were not marginal phenomena in biology but could be central players in the chemistry of life. One particularly elegant demonstration of enzyme tunneling came in 2006, when Laura Masgrau and colleagues published a paper in the journal Nature examining an enzyme called aromatic amine dehydrogenase. This enzyme, found in certain bacteria, catalyzes the oxidation of aromatic amines, and the researchers were able to show that the hydrogen transfer step in this reaction proceeded primarily through quantum tunneling rather than classical barrier crossing. The evidence was multifaceted, including large kinetic isotope effects, but also something else, something that provides perhaps the clearest signature of tunneling behavior: the temperature independence of the reaction rate. In classical chemistry, reaction rates typically increase with temperature, because higher temperatures mean more energetic molecules with a greater probability of having enough energy to surmount the activation barrier. This relationship, described mathematically by the Arrhenius equation, predicts that plotting the logarithm of reaction rate against inverse temperature should yield a straight line with a negative slope. The steeper the slope, the higher the activation energy. But quantum tunneling does not require thermal energy to overcome barriers. The particle simply passes through them, and so tunneling-dominated reactions can show reaction rates that are remarkably insensitive to temperature changes. There are Arrhenius plots displaying unusually shallow slopes or, in extreme cases, being nearly flat. The aromatic amine dehydrogenase studied by Masgrau showed exactly this behavior, its hydrogen transfer rate remaining stubbornly constant across a range of temperatures that would have dramatically affected a classical reaction. The combination of these experimental signatures, kinetic isotope effects exceeding classical limits and temperature-independent reaction rates, provided a robust toolkit for identifying quantum tunneling in enzyme systems, and researchers around the world began applying these methods to a widening array of biological catalysts. What emerged was a picture of enzymes as exquisitely tuned quantum devices, their protein structures not merely bringing reactive groups together but arranging them with the precision necessary to exploit the wave-like nature of matter, to allow protons and hydrogen atoms to tunnel through barriers that would otherwise be insurmountable on biological timescales. But enzymes, as important as they are, represent only one arena where quantum tunneling might operate in biological systems, and researchers interested in DNA and genetic mutation faced a more difficult challenge. Detecting tunneling in enzymes is relatively straightforward because enzymes perform the same reaction millions of times, allowing researchers to measure average rates and isotope effects with good statistical precision. DNA mutation, by contrast, is a rare event, a single proton shift in a single base pair, occurring perhaps once in every billion replications. And the event itself lasts only femtoseconds, millionths of billionths of seconds, far too brief to observe directly with any conventional technique. This temporal challenge, the problem of observing events that flash into and out of existence faster than any camera can capture, represents one of the central difficulties in the experimental study of quantum biology. A femtosecond is to 1 second as 1 second is to roughly 32 million years, a span of time so compressed that light itself travels only about 300 nanometers, roughly the width of the smallest bacterium. Chemical bonds vibrate on femtosecond timescales, electron transfers occur within femtoseconds, and proton tunneling, the quantum leap from one potential energy well to another, is similarly instantaneous, leaving no lasting trace that conventional spectroscopy can detect. One approach to this problem involves nuclear magnetic resonance spectroscopy, the same technology that underlies medical MRI scanning but applied at the molecular level to detect the positions and movements of atomic nuclei. In 2011, work by Olga Semikina and colleagues explored the use of NMR techniques to detect proton dynamics in molecular systems, attempting to capture some signature of the rapid motions and transfers that occur when protons shift between bonding partners. NMR is particularly sensitive to hydrogen nuclei, which are simply protons, and sophisticated pulse sequences can tease out information about how quickly protons are exchanging between different chemical environments, providing at least indirect evidence about the mechanisms of proton transfer, even if the tunneling event itself remains too fast to observe directly. The challenge with studying DNA specifically is that the tautomeric forms created by proton tunneling are thought to be extremely short-lived, existing for perhaps only picoseconds or nanoseconds before the proton tunnels back to its original position. Only occasionally, through some combination of circumstances that is not yet fully understood, does a tautomeric base persist long enough to encounter a replication enzyme and be incorporated as a mutation. This rarity makes experimental detection enormously difficult, because even the most sensitive spectroscopic techniques struggle to detect species that exist only transiently and in vanishingly small concentrations. Computational studies have therefore played an increasingly important role in understanding proton tunneling in DNA, allowing researchers to explore theoretically what experiments cannot yet directly observe. Judith Klinman, in addition to her experimental work on enzymes, contributed to this computational understanding through studies examining how protein and molecular conformations influence tunneling probability. Her concept of tunneling-ready conformations, molecular geometries in which the donor and acceptor atoms are brought into particularly close proximity and favorable alignment, has proven influential across the field, suggesting that biological systems may have evolved structural features that specifically facilitate quantum tunneling at key moments in their catalytic or replicative cycles. When applied to DNA, computational methods have shown that the double helix is not a rigid, static structure but a dynamic one, constantly breathing and flexing, its base pairs opening and closing, stretching and compressing. These thermal fluctuations occasionally bring the proton donor and acceptor atoms in a Watson-Crick base pair into particularly close proximity, creating transient windows of opportunity during which the barrier to proton tunneling becomes narrow enough for quantum passage to occur with significant probability. The challenge for experimentalists is to find ways of detecting these rare events, either by observing the tautomeric products before they revert or by measuring the mutagenic consequences of tunneling-induced mispairing. Some researchers have approached this problem indirectly, examining whether the mutation rates in living organisms show the kinds of signatures one would expect if quantum tunneling were contributing to genetic change. Temperature effects, isotope effects, and the specific patterns of mutations might all, in principle, reveal the quantum fingerprint, but the complexity of living systems, with their many interacting variables and evolved error correction mechanisms, makes such analyses challenging. A mutation rate that appears temperature-independent might reflect tunneling, or it might reflect cellular processes that compensate for temperature changes, maintaining constant fidelity through active quality control. The hunt for quantum ghosts in DNA thus continues, a patient endeavor conducted in laboratories around the world, using techniques that range from ultrafast spectroscopy to precision enzymology to sophisticated computer simulations. What the work of Klinman, Scruton, Masgrau, Semikina, and many others has established is that quantum tunneling is not merely a theoretical possibility in biological systems but a demonstrated reality in enzyme catalysis, occurring under physiological conditions, at body temperature, in the aqueous environment of the cell. Whether this quantum behavior extends to the spontaneous mutations that drive evolution, whether the double helix of DNA is itself a stage on which quantum dramas play out, remains an open question, tantalizing and profound. The experimental tools continue to improve. The simulations grow more sophisticated, and each year brings new insights into the subtle quantum mechanics that may underlie the chemistry of life. For now, we can say with confidence that the boundary between the quantum and classical worlds is not where physicists once assumed it to be, not confined to the realm of isolated atoms in vacuum chambers, but extending into the warm complexity of living cells, into the intricate machinery of enzymes, and perhaps into the helical strands that carry the instructions for life itself. As you rest there, breathing slowly, you might imagine the experiments proceeding in their quiet way, the spectrometers humming, the computers calculating, the researchers pipetting and measuring and thinking, all in pursuit of something that cannot be seen directly, only inferred from its consequences. A quantum ghost that leaves no footprint but changes everything it touches. A tunneling proton that might, just might, have something to do with why you are different from your parents, and why your children, should you have them, will be different from you. The story is incomplete, as all scientific stories are, but the pieces are falling into place, one careful experiment at a time. One isotope effect measured, one temperature series completed, one simulation converged, and the outline of something true beginning, slowly, to emerge from the uncertainty. For many years, the study of quantum effects in living systems remained a quiet corner of scientific inquiry, a place where a small number of researchers worked patiently on questions that the broader community regarded with some skepticism. The prevailing wisdom held that quantum mechanical phenomena, while absolutely fundamental to chemistry and therefore to the molecules of life, could not persist long enough in warm, wet biological environments to play any meaningful functional role. Coherence, that delicate quantum property in which particles exist in superpositions of states and maintain phase relationships with one another, was thought to be far too fragile to survive the constant thermal jostling of a living cell. And then, in the year 2007, a paper appeared in the journal Nature that would quietly begin to change this perception, not through dramatic claims or revolutionary rhetoric, but through careful experimental observation of something rather unexpected happening inside the light-harvesting machinery of photosynthetic bacteria. The laboratory at the University of California, Berkeley, where this work took place was led by Graham Fleming, a physical chemist who had spent decades developing increasingly sophisticated techniques for watching molecular events unfold on timescales almost impossibly brief. Fleming had been interested in photosynthesis for many years, drawn to the remarkable efficiency with which plants and bacteria capture sunlight and convert it into chemical energy. When a photon of light strikes a photosynthetic organism, it is absorbed by one of many pigment molecules arranged in what are called antenna complexes, and the energy of that photon must then travel through a network of these pigments until it reaches a reaction center where the actual chemistry of energy conversion takes place. This energy transfer happens with an efficiency approaching unity, meaning that almost no energy is lost along the way, and Fleming wanted to understand exactly how biological systems achieved such extraordinary performance. The particular system that Fleming and his colleagues chose to study was something called the Fehn-Matthews-Olsen complex, a protein structure found in green sulfur bacteria, which are ancient organisms that live in conditions of extremely low light and have therefore evolved to be exceptionally good at capturing whatever photons they can find. The Fehn-Matthews-Olsen complex, often abbreviated simply as FMO, had been structurally characterized decades earlier, and its relative simplicity compared to the photosynthetic machinery of plants made it an attractive target for detailed physical study. It consists of just 7 bacteriochlorophyll molecules held in precise positions by the surrounding protein scaffold, and energy absorbed by pigments in the bacterial antenna system passes through this complex on its way to the reaction center, where it will drive chemical reactions. What Fleming's group did, using a technique called two-dimensional electronic spectroscopy, was to watch the energy move through the FMO complex with a time resolution measured in femtoseconds, which are millionths of a billionth of a second. Two-dimensional electronic spectroscopy works by hitting a sample with a carefully timed sequence of ultra-short laser pulses and then measuring how the sample responds, and by varying the timing between these pulses and analyzing the resulting signals, researchers can construct a kind of map showing how energy flows from one part of a molecular system to another. Gregory Engel, who was a graduate student in Fleming's laboratory at the time and would later become a professor in his own right, played a central role in conducting these experiments and interpreting the complex data they produced. What Engel and the team observed in their two-dimensional spectra was something that theory had not clearly predicted and that challenged comfortable assumptions about the behavior of biological systems. They saw what came to be called quantum beating, an oscillatory signal that indicated the presence of quantum coherence in the energy transfer process. Rather than energy hopping randomly from one chlorophyll molecule to another in a classical, incoherent fashion, the data suggested that the excitation was delocalized across multiple pigments simultaneously, exploring many pathways at once in a quantum superposition before settling on its final destination. The coherent oscillations they detected lasted for approximately 660 femtoseconds, which is still an extremely short time by everyday standards, but is remarkably long for a quantum effect in a complex molecular environment. And crucially, these oscillations were observed at temperatures relevant to living organisms, not just in the extreme cold of laboratory conditions designed to minimize thermal noise. The finding that quantum coherence could persist for hundreds of femtoseconds in the warm, wet, noisy interior of a protein at physiological temperatures was genuinely surprising. The traditional expectation had been that the thermal fluctuations of a biological environment would destroy quantum coherence almost instantaneously, causing any superposition states to collapse into classical behavior before they could serve any functional purpose. The FMO results suggested that this expectation might be wrong, or at least incomplete, and that perhaps evolution had found ways to protect or even exploit quantum effects in service of biological function. The idea that began to emerge was that the protein environment might not simply be a source of destructive noise but could potentially be structured in ways that actually support coherent dynamics, perhaps by having vibrations at frequencies that resonate with the energy gaps between pigments, creating a kind of constructive interplay between quantum coherence and environmental motion. The publication of the Berkeley paper sparked a wave of activity in laboratories around the world, as researchers sought to confirm, extend, and understand these findings. Some groups worked to replicate the observations in FMO and other photosynthetic systems, while others began to debate the interpretation of the data and the extent to which coherence actually contributed to the efficiency of energy transfer. The field of quantum biology, which had existed in a somewhat dormant state for years, suddenly found itself reinvigorated by concrete experimental evidence that quantum effects might indeed play functional roles in living systems. And among the many questions that arose in the wake of the photosynthesis discoveries was whether similar phenomena might be at work in other biological processes, including the behavior of that most central of biological molecules, deoxyribonucleic acid. The question of whether quantum coherence might play a role in DNA was not entirely new, but the photosynthesis results gave it fresh urgency and credibility. DNA, after all, is an extended molecular structure containing bases that have conjugated pi-electron systems capable of supporting electronic excitations, and charge transfer through the DNA double helix had been studied for years with results that were sometimes puzzling and difficult to explain using purely classical models. If the protein scaffolds of photosynthetic complexes could support coherent energy transfer, perhaps the regular, stacked structure of DNA bases could support coherent electron or hole transfer in ways that might be relevant to mutation, repair, or damage recognition. The parallel was not exact, since energy transfer in photosynthesis and charge transfer in DNA are somewhat different physical processes, but the conceptual door had been opened to taking quantum effects in biological systems more seriously. Among those who began to explore these connections was Alexandra Olaya-Castro, a theoretical physicist working at University College London who had already been thinking about quantum effects in biological systems before the Fleming paper appeared. Olaya-Castro brought rigorous methods from quantum information theory and open quantum systems to bear on biological questions, asking not just whether quantum effects existed in biological contexts but whether they provided any actual advantage, whether there were tasks that quantum-assisted processes could perform better than purely classical ones. Her work emphasized the importance of moving beyond simple demonstrations of coherence to quantitative analysis of how quantum effects might contribute to biological function, and she developed theoretical frameworks for understanding how the interplay between coherence and environmental noise could be optimized in molecular systems. The concept that emerged from these investigations was that of quantum-assisted processes in biology, a more nuanced idea than simply asking whether biological systems are quantum or classical. The recognition was that biological systems exist in a regime where quantum and classical effects are both present and intertwined, where coherence may be partial and transient but still functionally significant, where the environment is not merely destructive but can be an active participant in dynamics. In this view, evolution may have tuned biological structures to operate at a kind of boundary between quantum and classical behavior, taking advantage of quantum effects where they are helpful while also benefiting from the robustness that comes from classical, thermally averaged behavior. The question for DNA specifically became whether similar tuning might exist in the molecular machinery that reads, copies, and repairs genetic information. The relevance to DNA research became clearer as investigators began to consider the various processes in which quantum effects might make a difference. In the case of photosynthesis, the putative advantage of coherence was in allowing energy to find the most efficient path through a complex network of pigments, sampling many routes simultaneously rather than wandering randomly. For DNA, the analogous questions might involve how electrons or holes created by oxidative damage travel along the helix, how enzymes that repair DNA locate damaged sites among the vast excess of undamaged bases, or how the process of base pairing during replication maintains its remarkable fidelity. Each of these processes involves molecular events occurring on very short timescales and very small length scales, exactly the regime where quantum mechanical effects are most likely to be significant. The photosynthesis discoveries also prompted methodological advances that would eventually find application in DNA research. The two-dimensional spectroscopy techniques developed by Fleming, Engel, and others represented a powerful new way of watching ultra-fast molecular dynamics, and similar approaches began to be applied to DNA and related systems. Understanding how to interpret coherent signals in complex molecular environments, how to distinguish genuine quantum effects from classical artifacts, and how to design experiments that could probe relevant timescales all became more sophisticated in the years following the FMO work. The theoretical tools developed by researchers like Olaya Castro for modeling open quantum systems in biological contexts similarly found broader application, providing frameworks for thinking about quantum effects in molecular biology beyond the specific case of photosynthesis. It is worth pausing to note that the interpretation of the original FMO experiments has continued to evolve and has been the subject of considerable scientific debate. Some subsequent studies suggested that the long-lived coherences observed in the initial experiments might have been partly electronic and partly vibrational in character, arising from coupling between electronic excitations and molecular vibrations rather than from purely electronic superpositions. Others questioned whether the coherences, even if present, actually contributed to the efficiency of energy transfer or were simply epiphenomena without functional significance. These debates, while sometimes contentious, are a normal and healthy part of how science refines its understanding of complex phenomena, and they did not diminish the fundamental impact of the original discoveries in opening up new ways of thinking about quantum effects in biology. What the photosynthesis work established, regardless of the ongoing refinements to its interpretation, was that it was no longer acceptable to dismiss the possibility of functional quantum effects in biological systems simply because those systems are warm and wet. The burden of proof shifted, and researchers studying processes like DNA damage and repair could no longer assume that quantum coherence was irrelevant without actually checking. This change in perspective was perhaps the most important legacy of the FMO experiments for the field of quantum biology as a whole and for the study of quantum effects in DNA specifically. It moved the conversation from whether biology could possibly be quantum to asking which biological processes might be quantum, how quantum they might be, and what difference it might make. The years following the publication of the Fleming paper saw a steady growth in research exploring quantum effects in DNA. Experimental techniques improved, theoretical frameworks became more sophisticated, and a community of researchers dedicated to these questions gradually coalesced. The connection between photosynthesis and DNA research was not one of direct analogy, since the two systems are chemically and physically quite different, but rather one of conceptual inspiration and methodological cross-fertilization. The demonstration that quantum coherence could survive in one biological context made it reasonable to look for it in others, and the tools developed for studying photosynthesis could be adapted and applied to the very different molecular architecture of the genetic material. As the field matured, it became clear that the relationship between quantum mechanics and DNA was likely to be subtle and multifaceted, not a simple matter of DNA being either quantum or classical, but rather a complex interplay of quantum effects operating at different levels and on different timescales. The quantum nature of chemical bonding, which determines the basic structure and stability of DNA, had never been in doubt. What the post-FMO era added was a serious consideration of whether higher-level quantum effects, involving coherence, tunneling, and entanglement, might play roles in the biological function of DNA that go beyond simple molecular structure. The question was no longer whether to think quantum mechanically about DNA, but how, and in what contexts, and with what functional consequences. The story of how photosynthesis research influenced thinking about DNA is in some ways a story about the interconnectedness of science, about how discoveries in one area can open doors in another, and about how the boundaries between fields are often more permeable than they appear. Graham Fleming, studying light-harvesting bacteria in Berkeley, Gregory Engel analyzing femtosecond spectroscopy data, Alexandra Olaya Castro building theoretical models in London, and countless other researchers contributing pieces to the puzzle were all part of a larger conversation about the role of quantum mechanics in living systems. That conversation continues today, and its insights inform our understanding of the molecular processes by which genetic information is stored, copied, and sometimes changed. The quantum mechanics of DNA, which might once have seemed an exotic or speculative topic, has become a legitimate and active area of research, and the lessons learned from leaves and bacteria have played no small part in making it so. Let us turn now to the matter of sunlight, that most familiar presence in our daily lives, and consider how it interacts with the molecules of inheritance at the very smallest scales of time and space. The sun, which warms our skin and illuminates our world, also sends forth invisible rays that carry just enough energy to disturb the delicate arrangement of atoms within our DNA, and this disturbance occurs through processes that unfold according to the strange and wonderful rules of quantum mechanics. We shall examine these processes slowly and carefully, following the path of discovery that scientists have traced over many decades, beginning with observations made in a laboratory in Tennessee in the early 1960s. In the year 1960, at the Oak Ridge National Laboratory, a researcher named Richard Setlow was investigating why ultraviolet light proved so harmful to living cells. This laboratory, nestled in the hills of eastern Tennessee, had been established during the Second World War for purposes related to nuclear research, but in the years that followed, its scientists turned their attention to the broader questions of how radiation of various kinds affected biological systems. Setlow, working with patience and methodical care, exposed samples of DNA to ultraviolet light and then analyzed what had become of the molecules. What he discovered was that certain bases in the DNA, specifically the pyrimidines known as thymine and cytosine, had become chemically linked to one another in an abnormal fashion. Two adjacent thymine bases, which ought to remain separate and properly bonded only to their complementary adenine partners on the opposite strand, had instead formed direct chemical bonds between themselves, creating a fused structure that came to be called a thymine dimer. This discovery of thymine dimers marked a turning point in our understanding of how sunlight damages genetic material. The finding was significant not merely as a chemical curiosity, but because it revealed a specific molecular lesion that could explain the biological effects of ultraviolet exposure, including the death of cells, the induction of mutations, and eventually, as would become clear in later years, the development of skin cancers. Setlow and his colleagues demonstrated that these dimers interfered with the normal processes of DNA replication and transcription, creating obstacles that the cellular machinery could not easily overcome. The discovery opened an entire field of research into photobiology and DNA repair that continues to yield insights to this day. To understand how these dimers form, we must consider the quantum mechanical behavior of light and matter at extraordinarily brief intervals of time. Ultraviolet radiation, unlike the visible light that our eyes perceive, carries photons of sufficient energy to excite the electrons within certain molecular structures. The DNA bases absorb ultraviolet light most strongly at wavelengths between 260 and 280 nanometers, with a peak absorption occurring at approximately 260 nanometers. This corresponds to the energy required to promote electrons from their ground state orbitals to higher energy excited states. When a photon of the appropriate wavelength encounters a pyrimidine base such as thymine or cytosine, There exists a probability that the photon will be absorbed and its energy transferred to the electronic structure of the molecule. The absorption of a single ultraviolet photon by a thymine base initiates a cascade of events that unfolds with almost unimaginable rapidity. Within the span of approximately 1 picosecond, which is 1/1,000,000th of a second, the excited thymine may react with an adjacent thymine on the same strand of DNA to form what is called a cyclobutane pyrimidine dimer. In this structure, the two thymine bases become covalently linked by a four-membered carbon ring, the cyclobutane ring, which bridges between the molecules and holds them in an abnormal configuration. The formation of this ring requires that the electrons in both thymine bases rearrange themselves in a coordinated fashion, breaking certain bonds and forming new ones in a process that occurs faster than most chemical reactions we encounter in ordinary experience. The quantum yield of this reaction, which describes the efficiency with which absorbed photons lead to dimer formation, is relatively low, falling in the range of approximately 1 to 3%. This means that for every 100 ultraviolet photons absorbed by thymine bases, only 1 to 3 will actually result in the formation of a cyclobutane pyrimidine dimer. The remaining 97 to 99% of absorbed photons have their energy dissipated through other pathways that do not produce this particular type of damage. This low quantum yield might seem like a small number, but given the enormous quantity of photons in sunlight and the vast number of pyrimidine bases in a human genome, even this modest efficiency results in the formation of thousands of dimers in sun-exposed skin cells over the course of a day spent outdoors. The cyclobutane pyrimidine dimers are not the only form of damage that ultraviolet light inflicts upon DNA. A second type of lesion, known as the 6,4 photoproduct, also forms when adjacent pyrimidines absorb ultraviolet radiation. In this case, a covalent bond forms between the carbon at position 6 of one pyrimidine and the carbon at position 4 of the adjacent pyrimidine, creating a different geometric distortion of the DNA helix. The 6,4 photoproducts form with somewhat lower frequency than the cyclobutane dimers, typically accounting for perhaps 20 to 30% of the total ultraviolet-induced pyrimidine damage, but they create more severe distortions in the DNA structure and may be more mutagenic in certain contexts. The quantum mechanical processes underlying these photochemical reactions were explored in exquisite detail by Bern Kohler and his research group at Ohio State University beginning in the early 2000s. Kohler, employing a technique called femtosecond spectroscopy, was able to observe the behavior of DNA molecules on timescales of femtoseconds, which are quadrillionths of a second, roughly 1,000 times shorter than the picosecond timescales on which dimers form. By using extremely brief pulses of laser light and sophisticated detection equipment, Kohler could follow the fate of electronic excitation energy in DNA bases from the moment of photon absorption through the subsequent relaxation processes. The findings published by Kohler and his colleagues in 2004 and the years that followed revealed a remarkable picture of what happens in the immediate aftermath of ultraviolet absorption. When a DNA base absorbs a photon and enters an excited electronic state, it does not remain in that excited state for very long. Instead, the energy is dissipated back to the ground state through a process called internal conversion, and this occurs on a timescale of less than 1 picosecond, often within just a few hundred femtoseconds. The extreme rapidity of this relaxation is crucial because it means that the excited base has very little time to undergo photochemical reactions such as dimer formation. In a sense, the DNA molecule is racing to get rid of the dangerous excitation energy before it can cause damage. The mechanism by which this rapid relaxation occurs involves features of the molecular energy landscape known as conical intersections. These are points where two different electronic states of a molecule have the same energy, creating what one might visualize as a funnel or a cone in the multidimensional space that describes the molecular configuration. When an excited molecule reaches a conical intersection, it can efficiently transfer from the excited electronic state to the ground state without emitting a photon, the energy instead being converted into vibrational motion of the atoms. The DNA bases, through billions of years of evolution, have molecular structures that place conical intersections in readily accessible locations on their potential energy surfaces, allowing them to shed excited state energy with remarkable efficiency. This intrinsic photostability of the DNA bases represents a form of quantum mechanical self-protection that was likely essential for the emergence and persistence of life on Earth. In the early history of our planet, long before the ozone layer had formed to filter out much of the ultraviolet radiation reaching the surface, the ancestors of modern organisms were exposed to far more intense ultraviolet bombardment than we experience today. The fact that the particular bases found in DNA, the adenine, guanine, cytosine, and thymine that spell out the genetic code, all share this property of ultra-fast excited state relaxation suggests that their photostability may have been a key factor in their selection as the informational molecules of life. Bases that absorbed ultraviolet light and remained excited for longer periods would have been far more prone to photochemical damage and thus less suitable as reliable carriers of genetic information. The sub-picosecond relaxation that occurs through conical intersections dissipates the majority of absorbed ultraviolet energy safely, converting electronic excitation into heat that is quickly distributed throughout the molecule and its aqueous surroundings. However, this protection is not absolute, and the small fraction of excitation events that do lead to photochemical reactions are responsible for the ultraviolet damage that accumulates in our DNA over time. The competition between safe energy dissipation and harmful photochemistry occurs on the same ultrafast timescales, with the outcome of each individual photon absorption event determined by the precise quantum mechanical state of the molecule at the moment of excitation and the subsequent trajectory through the potential energy landscape. Beyond the cyclobutane pyrimidine dimers and the 6,4 photoproducts, there exists a third category of ultraviolet-induced DNA damage known as dual isomers, or dual valence isomers. These structures form not directly from the initial ultraviolet absorption, but rather through a secondary photochemical rearrangement of the 6,4 photoproducts. When a 6,4 photoproduct absorbs a second ultraviolet photon, particularly at the slightly longer wavelengths found in UVB radiation, it can undergo an isomerization reaction that converts it into the dual form. This involves a rearrangement of the bonding pattern within the damaged site, producing a structure with different geometry and different biological consequences. The dual isomers are generally less efficiently repaired by cellular enzymes than the other forms of damage, and they may persist in DNA for longer periods, contributing to the mutagenic burden of ultraviolet exposure. The biological significance of these various forms of ultraviolet damage is perhaps most dramatically illustrated by the condition known as xeroderma pigmentosum, a rare inherited disorder that affects approximately 1 in 1 million individuals in Europe and North America, with somewhat higher prevalence in certain other populations such as Japan and North Africa. Patients with xeroderma pigmentosum carry mutations in genes that encode the proteins responsible for repairing ultraviolet damage in DNA. Without functional repair pathways, the thymine dimers, 6,4 photoproducts, and dewar isomers that form upon sun exposure accumulate in the cells of the skin, leading to severe consequences that become apparent early in life. Children with xeroderma pigmentosum develop extreme sensitivity to sunlight, experiencing severe sunburns after only brief exposure. Their skin ages prematurely and becomes covered with freckles and irregular pigmentation. Most significantly, they develop skin cancers at rates roughly 1,000 times higher than the general population, with many patients experiencing their first malignancies before the age of 10. Without rigorous protection from sunlight, including remaining indoors during daylight hours, wearing protective clothing, and using sunscreens, patients with xeroderma pigmentosum face a dramatically shortened lifespan due to metastatic skin cancer. The identification of the genetic defects underlying xeroderma pigmentosum has revealed that there are at least 7 different genes whose mutation can cause the disorder, each encoding a different protein component of the repair machinery. This genetic heterogeneity reflects the complexity of the repair pathways that have evolved to cope with ultraviolet damage, involving multiple proteins that must work together in a coordinated fashion to locate damaged sites, excise the abnormal bases, and restore the original sequence. The study of xeroderma pigmentosum patients has thus provided invaluable insights into the mechanisms of DNA repair, insights that have illuminated not only this particular condition but the broader processes by which all cells maintain the integrity of their genetic information. The quantum mechanical processes we have been discussing, the absorption of photons, the excitation of electrons, the traversal of conical intersections, and the formation of covalent bonds between adjacent bases, all occur within the first few picoseconds after an ultraviolet photon encounters a DNA molecule. These events are governed by the wave-like behavior of electrons, by the quantization of energy levels, and by the probabilistic nature of quantum transitions. And yet their consequences extend far beyond the molecular scale, influencing the health and survival of organisms, the prevalence of disease in populations, and perhaps even the direction of evolution over geological time. The gentle rays of sunlight that warm our faces on a pleasant afternoon carry within them this capacity for quantum mechanical interaction with our most essential molecules. The ultraviolet component of that light, invisible to our eyes but present nonetheless, sends forth photons that occasionally find their way through our skin to the nuclei of our cells, where they may excite the electrons of our DNA bases and, in a small fraction of cases, cause the formation of lesions that our repair machinery must then address. This process occurs continuously in all sun-exposed tissues, representing an ongoing dialogue between the external environment and the informational molecules that encode our biological inheritance. The low quantum yield of dimer formation, that 1 to 3% efficiency we mentioned earlier, represents a compromise of sorts between the inevitability of some photochemical damage and the remarkable protective mechanisms built into the very structure of the DNA bases. Were the quantum yield significantly higher, life as we know it might not have been possible under the sun's illumination. Were it 0, we would be entirely immune to the mutagenic effects of sunlight, though this scenario appears to be chemically impossible given the fundamental properties of ultraviolet photons and organic molecules. The actual value reflects the outcome of quantum mechanical processes that have been operating since the first appearance of nucleic acids on Earth, and it has shaped the evolutionary pressures that have led to the development of our sophisticated repair mechanisms. As we consider these matters of quantum photochemistry and molecular damage, we might find a certain comfort in the knowledge that our cells possess elaborate systems for correcting the lesions that inevitably form. The discovery of thymine dimers by Setlow and others was followed by decades of research into the repair pathways that reverse this damage. Research that has revealed an intricate network of enzymes capable of recognizing, removing, and replacing damaged bases with impressive fidelity. These repair systems will be our subject in due course, but for now, let us simply note that the quantum mechanical assault of ultraviolet radiation upon our DNA is met by an equally remarkable molecular response that has been refined by billions of years of evolution. The work of Kohler and other physical chemists continues to reveal new details of the ultrafast processes that occur when light meets DNA. Their femtosecond spectroscopy experiments have shown that the base stacking interactions within the double helix influence the pathways of energy dissipation, that the sequence context of a particular base affects its photochemical behavior, and that the aqueous environment surrounding DNA plays a role in stabilizing certain excited states and facilitating others. Each new finding adds another piece to our understanding of how quantum mechanics governs the interactions between sunlight and the molecules of life. And so we have traced the path of an ultraviolet photon from the sun, through the atmosphere, to the surface of our skin, through the layers of epidermis, and finally to an encounter with a thymine base in the DNA of a keratinocyte. We have followed the excitation of electrons, the brief residence in an excited state, the rapid race toward either safe relaxation or photochemical damage, and the occasional formation of a dimer that will require repair. All of this occurs in time shorter than the blink of an eye, shorter than the beat of a heart, shorter than any interval we can directly perceive, and yet these fleeting quantum events have shaped the course of evolution and continue to influence our health with every moment spent in the sun. And now we come to one of the most elegant repair systems that nature has ever devised, a mechanism so beautifully efficient that it uses light itself to heal the damage that light has caused. And in understanding this system, we will encounter some of the most precise measurements of quantum mechanical processes ever observed in biological molecules, all unfolding in timescales so brief that they challenge our ordinary sense of what it means for something to happen at all. The story begins in the late 1950s, in a laboratory at Johns Hopkins University, where a researcher named Claude Rupert was studying the effects of ultraviolet radiation on bacteria, and observing something that seemed, at first, to be a simple puzzle in need of a straightforward explanation. Rupert had noticed that bacteria exposed to damaging doses of ultraviolet light could sometimes recover from that damage, but only under certain conditions. And those conditions turned out to be surprisingly specific. When the damaged bacteria were kept in darkness, they remained damaged and often died. But when they were exposed to visible light, particularly light in the blue and violet range of the spectrum, they recovered at rates that seemed almost miraculous. This phenomenon had been observed by others before Rupert, but it was Rupert who, in 1958, began to systematically characterize the enzymatic basis of this recovery, demonstrating that the cells contained a specific enzyme that could use light energy to directly reverse ultraviolet damage, and he gave this process the name photoreactivation, a term that captures the essential feature of the mechanism, which is that light itself activates the repair. The damage being repaired was the thymine dimer that we have already discussed, those fused adjacent bases that form when ultraviolet photons strike DNA and cause neighboring thymine molecules to form improper chemical bonds with each other. These dimers distort the DNA helix and interfere with the normal processes of replication and transcription, and cells have evolved multiple ways of dealing with them. But photoreactivation is perhaps the most direct and the most ancient of these solutions, because it does not remove the damaged bases and replace them with new ones, but instead simply reverses the chemical reaction that created the damage in the first place, restoring the original undamaged structure without any cutting or pasting or resynthesis at all. The enzyme responsible for this remarkable feat is called Photolyase, and understanding its structure requires us to consider the way that proteins can incorporate small non-protein molecules called chromophores, which are specifically designed to absorb light and to convert that light energy into useful chemical work. Photolyase enzymes, as they have come to be understood through decades of careful study, contain not one but two of these light-absorbing components, each playing a distinct role in the overall repair mechanism, and the interplay between them reveals something profound about how evolution has optimized molecular machines for their tasks. The first and most essential of these chromophores is a molecule called flavin adenine dinucleotide, which we can abbreviate as FAD, and which is in fact a common cofactor found in many different enzymes throughout biology. In Photolyase, the FAD molecule exists in an unusual reduced form, carrying extra electrons that make it capable of donating those electrons to other molecules under the right circumstances, and it is this electron donating capacity that ultimately drives the repair reaction. The FAD is held deep within the protein structure, positioned in a cavity that is precisely shaped to accommodate a thymine dimer when the damaged DNA binds to the enzyme. The second chromophore varies between different species of organisms, but it is always a molecule that excels at absorbing light and transferring that absorbed energy to the FAD. In many bacteria and in simple eukaryotes, this second chromophore is a taurine molecule called methenyl tetrahydrofolate, abbreviated as MTHF, while in other organisms it is a diazoflavin molecule called 8-hydroxy-5-diazoflavin, abbreviated as 8-HDF. These antenna chromophores, as they are sometimes called, extend the range of wavelengths at which the enzyme can operate effectively, and they increase the overall efficiency of light capture by absorbing photons and funneling that energy inward toward the catalytic FAD molecule at the heart of the enzyme. The deeper understanding of how Photolyase actually works, at the level of individual chemical steps and electron movements, came through the patient and meticulous research of Aziz Sankar, who pursued this problem for many years, beginning in the 1980s when he was establishing his laboratory at the University of North Carolina at Chapel Hill, and continuing through subsequent decades as the available techniques for studying rapid chemical processes became increasingly sophisticated. Sankar's work on Photolyase, along with his contributions to understanding other DNA repair mechanisms, would eventually earn him a share of the Nobel Prize in Chemistry in 2015, recognition that came more than 30 years after much of his foundational work had been completed, which is perhaps a reminder that scientific understanding accumulates slowly and that important discoveries often require many years before their full significance is properly appreciated. What Sankar and his colleagues demonstrated, through a combination of biochemical experiments and structural studies, was that the repair of thymine dimers by Photolyase proceeds through a mechanism of electron transfer, in which the absorbed light energy is used to move an electron from the reduced FAD chromophore into the thymine dimer itself. This additional electron, arriving in the dimer, disrupts the bonding arrangement that holds the two thymine bases together, causing those bonds to break and allowing the bases to spring back into their normal, separated configuration. The electron, having done its work, then returns to the FAD, leaving the DNA fully repaired and the enzyme ready to perform the same reaction again. This description, while accurate in its broad outlines, does not capture the extraordinary speed at which these events occur, nor the precision with which the quantum mechanical processes must be orchestrated to achieve such efficient repair. For many years, the actual timescales of electron transfer in Photolyase remained uncertain, because the events were simply too fast to measure with the techniques that were available. But advances in ultra-fast spectroscopy eventually made it possible to observe these processes directly, and the results were striking in their implications for how we understand biological chemistry. In 2010, Klaus Brettell and his collaborators published studies demonstrating that the electron transfer from the excited FAD to the thymine dimer occurs in approximately 250 picoseconds, which is to say 250 trillionths of a second, a span of time so brief that light itself, traveling at 300 million meters per second, would cover only about 7.5 centimeters during this interval. This measurement revealed that the repair reaction is extraordinarily fast, occurring on timescales that are characteristic of simple quantum mechanical processes rather than the slower, more complex rearrangements that characterize many enzymatic reactions, and this speed is part of what makes Photolyase so remarkably efficient. Further studies of the repair dynamics, including influential work published in 2016 by Dongping Zhang and his research group, have elaborated the details of this mechanism, showing how the various steps of the reaction are coordinated in time and how the protein structure itself helps to guide and accelerate the repair process. Zhang's work, using sophisticated spectroscopic techniques to follow the reaction as it unfolds, has revealed that the initial electron transfer is followed by bond breaking in the thymine dimer, and then by the return of the electron to the flavin, and that this entire sequence occurs smoothly and efficiently, with very little energy lost to unproductive pathways. One of the most striking features of Photolyase, and one that sets it apart from most other enzymes, is its quantum efficiency, which is a measure of how effectively it converts absorbed photons into successful repair events. For most light-driven processes in biology, quantum efficiency is considerably less than unity, meaning that many absorbed photons fail to produce the desired outcome. But for Photolyase, the quantum efficiency approaches 100%, meaning that essentially every photon absorbed by the enzyme results in the successful repair of a thymine dimer. This extraordinary efficiency suggests that the enzyme has been optimized by billions of years of evolution to minimize every possible source of waste and error in the repair mechanism, and it raises interesting questions about whether quantum mechanical coherence might play a role in achieving such performance. The question of quantum coherence in biological systems is one that occupies a fascinating boundary between physics and biology, and while it remains an area of active research and some controversy, there is evidence suggesting that the electron transfer processes in Photolyase may indeed involve coherent quantum mechanical effects, in which the electron exists in a superposition of states rather than following a single classical pathway from donor to acceptor. Such coherence, if it is maintained long enough to influence the reaction, could help to explain how the enzyme achieves its remarkable efficiency, by allowing the electron to explore multiple pathways simultaneously and to find the most effective route to its destination. The extremely fast timescales observed by Brettell and others are consistent with this possibility, though definitively proving the role of quantum coherence in biological processes remains a challenging experimental problem. Having come to appreciate the elegance and efficiency of Photolyase, you might naturally wonder why humans and other mammals do not possess this enzyme, given how useful it would seem to be for repairing the damage caused by sunlight. The answer to this question involves an interesting evolutionary trade-off, in which the genes that once encoded Photolyase have been repurposed for an entirely different function, One that has nothing to do with DNA repair but everything to do with the internal clocks that regulate our daily rhythms of sleep and wakefulness. Mammals, including humans, retain genes that are clearly descended from the ancient Photolyase genes. But these genes now encode proteins called cryptochromes, which have lost the ability to repair DNA but have gained a new role as components of the circadian clock, the internal timekeeping mechanism that synchronizes our biology to the cycle of day and night. Cryptochromes are light-sensitive proteins that help to reset the circadian clock in response to environmental light, allowing organisms to adjust their internal rhythms to match the external world. And this function has apparently become more important to mammalian survival than the ability to repair thymine dimers using light. The loss of photoreactivation in mammals may be related to our evolutionary history as creatures who were, for millions of years, primarily nocturnal, active mainly at night and sheltered from the sun during the day. Under such conditions, the ability to repair ultraviolet damage using visible light would have been less valuable than it is for organisms that live their lives in constant exposure to sunlight. And other repair mechanisms, such as the nucleotide excision repair pathway that we shall discuss later, would have been sufficient to handle whatever damage did occur. Meanwhile, the circadian functions of cryptochrome would have remained important regardless of lifestyle, providing selection pressure to maintain and optimize those genes even as their repair function was allowed to fade. What makes this evolutionary story particularly remarkable is the extraordinary conservation of Photolyase itself across the rest of the living world, where it has been maintained with relatively little change for an estimated 3 billion years, since the earliest days of life on Earth. Photolyase enzymes from bacteria, archaea, plants, fungi, and animals all share recognizable structural features and mechanistic properties, indicating that this repair system evolved very early in the history of life and has been faithfully preserved ever since. This conservation speaks to the fundamental importance of DNA repair for all living things, and to the exceptional effectiveness of the Photolyase mechanism, which has apparently never been improved upon by subsequent evolutionary innovation. The presence of functional Photolyase in organisms as diverse as the simplest bacteria and complex multicellular plants demonstrates that this enzyme was present in the last common ancestor of all current life forms, and that the basic challenge of protecting genetic information from ultraviolet damage has been a constant throughout the history of life. The solution that evolution found, in those early days when the Earth's atmosphere provided less protection from solar ultraviolet radiation than it does today, was a quantum mechanical machine of extraordinary precision, capable of harnessing light energy to drive chemical reactions with near-perfect efficiency, and that solution has served life well for billions of years. As you contemplate these facts, perhaps letting your thoughts drift toward the gentle boundary between waking and sleep, you might reflect on the way that sunlight, which seems so simple and constant in our daily experience, participates in this intricate dance of damage and repair at the molecular level. The same photons that warm our skin and illuminate our days can break the delicate bonds of our genetic material. Yet in most organisms, those very photons can also be captured and put to work repairing the damage they cause. The enzyme that performs this feat is a molecular embodiment of balance and efficiency, refined by billions of years of evolution to waste nothing and to fail almost never, operating on timescales so brief that they blur the line between chemistry and physics, between the classical world of molecules and the quantum world of electrons and probabilities. And so we find, in the humble Photolyase enzyme, a window into some of the deepest questions about how life operates at its most fundamental level, where the rules of quantum mechanics govern the behavior of the particles and the transfer of energy and the breaking and making of chemical bonds. This is not a dramatic or exciting place, in the ordinary sense of those words, but rather a place of quiet precision and ancient wisdom, where molecular machines perform their tasks with a reliability that puts human engineering to shame, and where the boundary between the living and the non-living dissolves into a continuum of organized chemistry and captured energy. In understanding Photolyase, we understand a little better what it means for matter to be alive, to maintain itself against the constant threat of decay and damage, to preserve information across generations and across billions of years. And that understanding, like the repair of a thymine dimer, happens quietly and efficiently, without fanfare or drama, just the steady accumulation of knowledge in the gentle light of patient inquiry. And so we come now to a quieter kind of story, one that unfolds not in the dramatic moments of mutation but in the ceaseless, patient work of repair, the molecular housekeeping that keeps our genomes intact through the long years of a human life. This is the story of enzymes that act as surgeons at the smallest possible scale, identifying damage among billions of healthy bases and excising it with a precision that borders on the miraculous. And it begins, as so many important stories do, with a single researcher asking a question that others had overlooked. Thomas Lindahl was working at the Karolinska Institute in Stockholm in the early 1970s when he began to wonder about something that seemed almost too simple to matter. Everyone knew that DNA was stable, that it had to be stable to serve as the molecule of heredity, passing information faithfully from generation to generation across the vast stretches of evolutionary time. But Lindahl, with the careful skepticism of a good scientist, decided to measure just how stable DNA actually was, and what he found was quietly revolutionary. DNA, it turned out, was not nearly as stable as everyone assumed. It was decaying all the time, spontaneously and continuously, even in the absence of radiation or chemical mutagens or any external assault. The molecule of life was falling apart, base by base, in a slow but relentless process of chemical degradation. This was a puzzle that demanded explanation. If DNA was constantly accumulating damage, how did cells survive at all, let alone pass their genetic information intact to their descendants? The answer, Lindahl realized, must be that cells possessed repair mechanisms, enzymatic systems dedicated to finding and fixing the constant stream of lesions that arose through normal chemistry. He set out to identify these repair pathways, and over the following decades he and his colleagues mapped out an entire landscape of molecular repair, a set of overlapping and complementary systems that together maintain the integrity of the genome. For this work, Lindahl shared the Nobel Prize in Chemistry in 2015, alongside Paul Modrich and Aziz Sankar, who had elucidated other aspects of DNA repair. The citation recognized their mechanistic studies of DNA repair, a phrase that perhaps understates the profound importance of understanding how life protects itself against its own fragility. The numbers involved in this cellular maintenance are staggering, almost too large to hold comfortably in the mind, and yet they describe processes happening right now, in this very moment, in every cell of your body as you listen to these words. Each human cell sustains somewhere between 10,000 and 100,000 DNA lesions every single day. This is not damage from unusual circumstances, not the result of radiation exposure or chemical poisoning, but simply the consequence of normal cellular chemistry, the background noise of molecular existence. Water molecules attack DNA bases in a process called hydrolysis. Reactive oxygen species. The byproducts of ordinary metabolism oxidize and damage the genetic material. Bases spontaneously lose their amino groups in a process called deamination, converting one nucleotide into something that resembles another. The genome is under constant siege from the very chemistry of life itself, and only the tireless work of repair enzymes keeps this damage from accumulating into catastrophe. Among the most important of these repair pathways is base excision repair, the system that Lindahl helped to discover and characterize. This pathway handles the most common forms of DNA damage, the small chemical modifications to individual bases that arise from oxidation, deamination, and alkylation. The first step in base excision repair is recognition, the identification of a damaged base among the vast sea of normal ones. And this task falls to a remarkable family of enzymes called glycosylases. These molecular surgeons patrol the DNA, somehow distinguishing damaged bases from healthy ones and removing them with exquisite precision, leaving behind a gap that other enzymes then fill with the correct nucleotide. The chemistry of this removal is well understood. The glycosylase cleaves the bond between the damaged base and the sugar backbone of DNA, but the recognition step remains in many ways mysterious, a process that pushes against the limits of what we might expect molecular machines to accomplish. Consider the quantum mechanical challenge that faces a glycosylase enzyme as it moves along the DNA double helix. A human genome contains approximately 3 billion base pairs, 3 billion individual nucleotides arranged in a specific sequence that encodes all the information necessary to build and maintain a human being. Among these billions of bases, the glycosylase must find the handful that have been damaged, perhaps a few thousand per day out of those 3 billion. A task analogous to finding a few particular grains of sand on a beach stretching for miles in either direction. And the damage it seeks is often subtle. A single oxygen atom added here, a hydrogen atom removed there, changes that barely alter the overall structure of the base. How can any molecular machine achieve this level of discrimination, searching through billions of possibilities to find the rare needle in an enormous haystack? Part of the answer lies in a remarkable mechanism called base flipping, which was first visualized in atomic detail through the structural biology work of researchers like Gregory Verdine at Harvard University. Verdine and his colleagues used X-ray crystallography and other techniques to capture glycosylase enzymes in the act of examining DNA, freezing the molecular dance in a single frame that revealed its choreography. What they found was unexpected and beautiful. The glycosylase does not simply slide along the outside of the DNA double helix, reading the bases as they sit in their normal paired configuration. Instead, it actively reaches into the helix and flips individual bases outward, rotating them 180 degrees out of their normal position and into a pocket within the enzyme itself. This base flipping allows the glycosylase to examine each nucleotide individually, holding it in an environment where damage can be detected and acted upon. The process of base flipping is energetically costly, requiring the enzyme to disrupt the hydrogen bonds that hold the base pair together and to overcome the stacking interactions between adjacent bases. And yet glycosylases perform this operation rapidly and continuously, flipping base after base as they scan along the DNA, checking each one before allowing it to snap back into position if it proves to be undamaged. The energy for this constant sampling must come from somewhere, and recent research has suggested that quantum mechanical effects may play a role in making the process efficient enough to be biologically feasible. The enzyme does not simply wait for thermal fluctuations to randomly flip a base outward, a process that would be far too slow to scan the entire genome in a reasonable time. Instead, it appears to actively interrogate the bases, using a combination of mechanical force and electronic interaction to probe their chemical state. One of the most intriguing suggestions from recent research is that electron tunneling may assist in the recognition of damaged bases. When a glycosylase encounters a base, it must determine within a fraction of a second whether that base is damaged or healthy, and the structural differences it must detect are often extraordinarily subtle. Some researchers have proposed that the enzyme uses electron tunneling as a kind of molecular probe, sending electrons through the base and measuring how they behave. A damaged base would have different electronic properties than a healthy one, a slightly different arrangement of electron orbitals, a slightly different tunneling probability, and the enzyme might be able to detect these differences even when the overall shape of the base remains nearly unchanged. This is still an area of active investigation, and the details remain to be fully worked out, but the basic idea, that quantum mechanical sensing assists in damage recognition, has gained increasing support from both theoretical calculations and experimental observations. One of the best-studied examples of this remarkable specificity involves an enzyme called oxoguanine glycosylase, or OGG1 for short, and its recognition of a damaged base called 8-oxoguanine. This lesion, 8-oxoguanine, is the most common form of oxidative DNA damage in human cells, arising when reactive oxygen species attack the base guanine and add an extra oxygen atom at the 8 position. Every day, thousands of guanine bases in every cell undergo this oxidation, and every day, OGG1 must find these damaged bases and remove them before they cause mutations during replication. The challenge is that 8-oxoguanine looks almost exactly like normal guanine. The addition of that single oxygen atom changes the molecular weight by only 16 daltons, adds only one small bump to the overall structure, and barely alters the shape of the base. And yet OGG1 must distinguish between these two forms with extraordinary accuracy, removing the damaged bases while leaving the millions of normal guanines untouched. The structural studies of OGG1 have revealed a sophisticated recognition mechanism that involves multiple checkpoints, each one probing a different aspect of the base's chemistry. When OGG1 flips a base into its active site pocket, it does not simply look at the overall shape. It makes specific contacts with particular atoms, hydrogen bonds that can only form if the base has exactly the right chemical structure. The extra oxygen in 8-oxoguanine creates new possibilities for hydrogen bonding, and OGG1 exploits these possibilities to identify the lesion. But even this is not enough to explain the enzyme's remarkable specificity, because thermal fluctuations and molecular breathing could potentially allow a normal guanine to temporarily mimic some of the contacts expected for 8-oxoguanine. The enzyme needs additional mechanisms to ensure that it only removes truly damaged bases. This is where the work of researchers like Sheila David at the University of California, Davis, has been particularly illuminating. David and her colleagues have studied a related glycosylase called MUT-Y, which faces an even more challenging discrimination problem. MUT-Y must recognize adenine bases that have been mispaired with 8-oxoguanine during replication, removing the adenine so that the correct base, cytosine, can be inserted instead. The challenge is that adenine paired with 8-oxoguanine looks very similar to adenine paired with thymine, the normal Watson-Crick base pair, and MUT-Y must somehow distinguish between these two situations despite their similar overall structures. David's research has revealed that MUT-Y uses a mechanism called kinetic gating, in which the enzyme selectivity emerges not from a simple lock-and-key recognition, but from the dynamics of how the enzyme interacts with different substrates over time. In kinetic gating, the enzyme makes its decision through a series of steps, each with its own rate constant, its own probability of proceeding forward or falling back. A damaged base promotes forward progression through these steps, while a normal base tends to cause the enzyme to release and move on. The overall selectivity emerges from the multiplication of many small preferences, each step contributing a modest discrimination that compounds into the extraordinary specificity observed for the overall process. This kinetic perspective reveals that glycosylase enzymes are not simply molecular calipers, measuring the physical dimensions of bases to determine damage. They are dynamic machines that probe and test and interrogate their substrates through a temporal process, a kind of molecular dialogue that unfolds over microseconds and milliseconds. The quantum mechanical aspects of this kinetic gating are still being explored, but there are reasons to think that quantum effects matter at several stages of the process. The probing of electronic structure that may involve electron tunneling, the subtle rearrangements of hydrogen bonding patterns that distinguish damaged from undamaged bases, the conformational changes in the enzyme that propagate the recognition signal to the catalytic machinery. All of these steps involve molecular motions and electronic rearrangements at scales where quantum mechanics provides the appropriate description. Classical models can capture some aspects of the behavior, but the full picture likely requires attention to the quantum nature of the electrons and protons involved. What emerges from this research is a picture of DNA repair as a quantum mechanical enterprise, not in the sense of exotic quantum computing or macroscopic superposition states, but in the more fundamental sense that the recognition and repair of damaged DNA relies on molecular properties that can only be understood through quantum mechanics. The enzymes that patrol our genomes are products of billions of years of evolution, refined through countless generations to exploit every available physical mechanism in their ceaseless work of maintenance. If quantum tunneling provides even a modest advantage in damage recognition, evolution would have found it and used it. If quantum coherence assists in the coordination of electron movements during catalysis, evolution would have stumbled upon this trick and preserved it in enzyme structure. And so as you settle deeper into rest, perhaps you can take some comfort in the knowledge that your cells are not passive vessels, simply waiting for damage to accumulate until they fail. They are instead hives of constant activity, patrolled by molecular surgeons that work through the night and through the day, finding and fixing the endless stream of lesions that arise from the ordinary chemistry of existence. The glycosylases flip bases outward, examine them in their specialized pockets, probe their electronic structure through mechanisms that may involve quantum tunneling, and then either excise them if damaged or release them if healthy. Other enzymes follow behind, filling the gaps, sealing the backbone, restoring the integrity of the double helix. 10,000 to 100,000 lesions per cell per day, and yet your genome remains largely intact. The information it carries preserved through decades of life, the instructions for your cells copied and recopied with only the rarest errors slipping through. This is the gift of base excision repair and the other maintenance pathways that Thomas Lindahl and his colleagues revealed to us, a testament to the sophistication of molecular machines that operate at the boundary between classical and quantum, between chemistry and physics, between the random thermal motion of molecules and the precise, purposeful activity of life. The work continues in laboratories around the world, Researchers probing ever deeper into the mechanisms of damage recognition and repair, building on the foundations laid by Lindahl and Verdeen and David and so many others. Each new study adds another detail to the picture, another piece to the puzzle of how enzymes achieve their remarkable specificity, their ability to find needles in haystacks of billions, their tireless vigilance in the face of constant chemical assault. And in that work there is something deeply reassuring, not just the knowledge that our cells are protected, but the understanding that nature has found solutions to problems that seem almost impossibly difficult, that life has learned to exploit the deepest features of physical reality in its endless project of self-maintenance and self-renewal. Rest now, and let your glycosylases do their work, flipping bases, probing for damage, excising the lesions that would otherwise accumulate into dysfunction and disease. They have been doing this work for billions of years, since the first cells arose in ancient oceans, and they will continue doing it as you sleep, as you dream, as your body repairs and restores itself through the quiet hours of the night. This is the molecular surgery that keeps you whole, the quantum mechanics of repair that guards the integrity of your genome, one base at a time, 10,000 times a day, in every one of the trillions of cells that together make up the miracle of your living body. And now we come to consider a quieter sort of damage, the kind that accumulates not through sudden violent ruptures in the molecular architecture, but rather through the slow and patient work of oxygen itself, that element we breathe in with such grateful regularity, that gas which sustains our metabolism and permits the burning of fuel within our cells, and which also, in its more reactive forms, participates in a gentle but relentless degradation of the very molecules that carry our hereditary instructions. This is the story of oxidative damage to DNA, a process that occurs with remarkable frequency in every cell of your body even as you rest here listening, a process rooted in the quantum mechanical behavior of electrons as they move through and around the double helix, and it is a story that begins, as many stories in the science of aging do, with a radiologist in California who looked at the damage caused by ionizing radiation and wondered whether something similar might be happening inside us all the time, slowly, incrementally, as a natural consequence of being alive and breathing in an atmosphere rich with oxygen. Denham Harmon was working at the Donner Laboratory of Medical Physics at the University of California, Berkeley, in the early 1950s, studying the effects of radiation on living tissue, when he began to develop an idea that would eventually reshape how we think about aging and cellular damage. The year was 1954, and Harmon proposed what he called the free radical theory of aging, suggesting that the reactive molecular fragments produced during normal metabolism, particularly those involving oxygen, might be responsible for the gradual deterioration we observe in living systems as they grow older. This was a novel proposition at the time, because free radicals were primarily associated with radiation damage and industrial chemistry, not with the quiet workings of ordinary cellular life. Harmon recognized that the very process of using oxygen to generate energy, the process we call aerobic respiration, inevitably produces small quantities of highly reactive oxygen species as byproducts, and that these reactive molecules, though transient and short-lived, might accumulate damage over the years and decades of a lifetime, contributing to the familiar signs of aging and the diseases that accompany advancing years. His theory was initially met with some skepticism, as novel theories often are, but over the following decades, as more and more evidence accumulated regarding the chemistry of oxygen in biological systems, his fundamental insight proved remarkably prescient, and the study of oxidative stress became a major field of biomedical research. To understand why oxygen can be both essential for life and damaging to living molecules, we need to consider the particular chemistry of what scientists call reactive oxygen species, often abbreviated simply as ROS. Ordinary molecular oxygen, the kind that makes up about 21% of the air around you, consists of two oxygen atoms bound together in a relatively stable configuration, content to participate in controlled reactions within the enzyme complexes of your mitochondria where energy is extracted from food. But during the normal operation of these energy-producing systems, and through various other chemical reactions within cells, small amounts of oxygen become converted into more reactive forms that are much less discriminating about what they react with. The first of these is superoxide, which forms when an ordinary oxygen molecule accepts a single extra electron, giving it a negative charge and making it considerably more reactive than its parent molecule. Superoxide is produced in modest quantities during normal mitochondrial respiration, essentially as a kind of inevitable leakage from the electron transport chain, and cells have evolved specific enzymes called superoxide dismutases, whose sole purpose is to convert this reactive species into something less immediately dangerous. The product of superoxide dismutase activity is hydrogen peroxide, which you may recognize as the mild antiseptic found in medicine cabinets, a molecule consisting of two hydrogen atoms and two oxygen atoms arranged in a particular way. Hydrogen peroxide is less reactive than superoxide but still carries oxidizing potential, and cells maintain another enzyme called catalase that breaks hydrogen peroxide down into ordinary water and oxygen. The difficulty arises when these detoxification systems fail to capture all of the reactive species produced, or when hydrogen peroxide encounters certain metal ions, particularly iron or copper, which can catalyze its conversion into something far more dangerous still. This something is the hydroxyl radical, arguably the most reactive molecule commonly found in biological systems, consisting of just one oxygen atom and one hydrogen atom with an unpaired electron that makes it desperately eager to react with almost anything nearby. The hydroxyl radical is so reactive that it typically reacts with the very first molecule it encounters, within nanoseconds of its formation, and this molecule might be a lipid in a cell membrane, or a protein performing some essential function, or, pertinent to our discussion tonight, one of the nucleotide bases in a strand of DNA. When a hydroxyl radical encounters a DNA molecule, it can react with any of the various atoms in the sugar phosphate backbone or in the bases themselves, producing a variety of different chemical modifications depending on exactly where the reaction occurs. Among all the possible products of oxidative damage to DNA, one particular lesion stands out as by far the most common and the most thoroughly studied, and this is the modified base known as 8-oxo-7-8-dihydroguanine, usually abbreviated as 8-oxo-G or sometimes written as 8-oxo-DG when referring to the entire nucleoside including the sugar. This damaged form of guanine results when a hydroxyl radical adds an oxygen atom to the guanine base at a specific position, and it accumulates in cellular DNA at truly remarkable rates. The biochemist Bruce Ames, working at Berkeley some decades after Harman proposed his free radical theory, estimated that each human cell sustains approximately 10,000 to 100,000 oxidative hits to its DNA every single day, and among these, the formation of 8-oxo-G represents a substantial fraction. These numbers may seem almost impossibly large, but they reflect the sheer scale of molecular activity within each cell, the billions of molecules moving and reacting, and the statistical inevitability that some fraction of oxygen metabolism will produce reactive species that escape the normal protective systems and reach the nucleus where the DNA resides. The reason guanine is particularly susceptible to oxidative damage, more so than the other three bases found in DNA, relates directly to its quantum mechanical properties, specifically to a quantity called the ionization potential. The ionization potential of a molecule is the amount of energy required to remove an electron from it completely, to strip away one of its negative charges and leave behind a positively charged ion or radical. Among the 4 DNA bases, guanine has the lowest ionization potential, meaning it holds on to its electrons less tightly than adenine, cytosine, or thymine, and this makes it more willing to participate in electron transfer reactions, more vulnerable to oxidation, more likely to serve as the final resting place for the kind of oxidative damage that permeates our cells. This is not a flaw in the design of DNA, but simply a consequence of guanine's particular electronic structure, the way its atoms are arranged and the energies of its molecular orbitals, properties that are determined by the quantum mechanical equations governing electron behavior and that cannot be changed without fundamentally altering the molecule itself. This brings us to a remarkable discovery about the quantum behavior of electrons in DNA, one that emerged from experiments conducted beginning in 1993 in the laboratory of Jacqueline Barton at the California Institute of Technology. Barton and her colleagues were interested in how electrons and the absence of electrons, called holes, might move through the DNA double helix, and they designed elegant experiments to measure this movement directly. What they found surprised many in the scientific community and opened up an entirely new way of thinking about DNA as more than just an information storage molecule, but also as a medium through which quantum information, in the form of electron transport, could travel. The experiment showed that DNA could act as what Barton evocatively called a molecular wire, conducting electrons along its length over distances that seemed improbably large for a molecule that had not been designed for this purpose. The physical basis for this conductivity lies in the stacking of the base pairs within the double helix, the way adenine-thymine and guanine-cytosine pairs are arranged like a spiral staircase with each flat, ring-shaped base pair sitting above and below its neighbors at a spacing of about 3.4 angstroms. The bases are aromatic molecules, meaning they contain rings of carbon atoms with electrons that are delocalized above and below the plane of the ring in what chemists call pi orbitals. When these aromatic bases stack on top of one another in the regular geometry imposed by the double helix, their pi orbitals can overlap, creating a continuous pathway along which electrons can move from one base pair to the next. This is the phenomenon known as pi stacking, and it provides the electronic coupling that allows the DNA helix to support long-range charge transfer. The Barton laboratory experiments demonstrated that electrons or holes could migrate through DNA over distances exceeding 200 angstroms, which corresponds to roughly 60 base pairs, a distance that spans a substantial portion of a typical gene. This was a remarkable finding because most electron transfer in biological molecules occurs over much shorter distances, typically requiring the donor and acceptor molecules to be in close proximity, and the ability of DNA to conduct charge over such long distances suggested that something more interesting was happening than simple electron hopping from one base to the next. The mechanism that emerged from these studies involves what is called hole transfer, where the thing that moves along the DNA is not an electron itself but rather the absence of an electron, a positive charge that can migrate from one guanine to another through the pi-stacked bases. When an electron is removed from a guanine base somewhere in the DNA helix, either by an oxidizing species or by some other electron acceptor, it leaves behind a hole, a site of positive charge. This hole can then migrate along the helix, moving from one location to another, but it does not move randomly. Because guanine has the lowest ionotation potential among the bases, holes tend to accumulate at guanine sites, particularly at sequences where multiple guanines occur in a row, because these sequences represent energetic valleys where the positive charge can settle most comfortably. This preferential localization of holes at guanine sites helps explain why guanine is so susceptible to oxidative damage, because the charge can essentially funnel toward these sites from elsewhere in the helix, concentrating the oxidative potential at the most vulnerable locations. The quantum mechanical nature of this transfer process is particularly interesting. Studies have shown evidence for quantum coherent transport, meaning that the electron or hole can exist in a superposition of states across multiple bases simultaneously as it moves through the helix, rather than simply hopping from one discrete location to the next in a classical fashion. This coherent transport is facilitated by the regular geometry of the stacked bases and the electronic coupling provided by the overlapping pi orbitals, and it allows for more efficient charge migration than would be possible through purely classical mechanisms. The DNA helix, that familiar twisted ladder of molecular biology textbooks, turns out to be a rather sophisticated quantum system when viewed from the perspective of electron dynamics. The connection between this fundamental chemistry and the diseases of aging becomes clear when we consider where oxidative damage tends to occur and what consequences it might have. The accumulation of 8-oxo-G and other oxidative lesions in cellular DNA can interfere with normal cellular function in several ways. If the lesion occurs in a gene that encodes an important protein, it may lead to mutations when the damaged DNA is replicated, because 8-oxo-G tends to mispair with AID9 rather than cytosine during DNA synthesis, ultimately leading to G to T transversion mutations in the genetic code. If such mutations occur in genes that regulate cell growth and division, they can contribute to the development of cancer, which is fundamentally a disease of uncontrolled cellular proliferation driven by accumulated genetic changes. Among the genes most critically important for preventing cancer is one called P53, often described as the guardian of the genome because of its central role in detecting DNA damage and coordinating the cellular response to such damage. The P53 protein can halt cell division to allow time for repairs, can activate DNA repair pathways, and can trigger programmed cell death if the damage is too severe to be safely repaired. Mutations in P53 are found in roughly half of all human cancers, making it the most commonly mutated gene in cancer, and studies have shown that oxidative damage to the P53 gene occurs at a significant rate in cells experiencing oxidative stress. The quantum mechanical funneling of oxidative damage toward guanine residues means that guanine-rich sequences in critical genes like P53 may be particularly vulnerable to this kind of mutagenic damage, and the long-range electron transfer demonstrated in the Barton experiment suggests that oxidative damage initiated at one location in the genome could potentially propagate to cause damage at distant guanine-rich sites. The numbers estimated by Bruce Ames and others regarding the frequency of oxidative hits to DNA in each cell each day underscore the remarkable challenge faced by our cellular repair systems. If we accept an estimate of 100,000 oxidative lesions per cell per day, and consider that the human body contains roughly 37 trillion cells, the total amount of oxidative DNA damage occurring throughout the body reaches truly astronomical proportions. That we can function normally despite this constant assault reflects the extraordinary efficiency of our DNA repair systems, which evolved over billions of years to deal with exactly this sort of damage. Specialized repair enzymes recognize and remove atoxo-G and other oxidative lesions, replacing them with undamaged bases, working continuously to maintain the integrity of the genome against the ongoing tide of oxidative damage. As you rest there listening, your cells are performing these countless acts of molecular maintenance, detecting and repairing the damage caused by the simple act of breathing, of using oxygen to power the chemistry of life. The quantum electrons that facilitate the transport of oxidative damage through your DNA are also, in a sense, participants in the repair process, as the same electronic properties that make guanine vulnerable also make it recognizable to the repair enzymes that have evolved to find and fix these lesions. It is a continuous and quietly heroic process, this maintenance of the genome, carried out in every cell at every moment, and it continues now as you breathe slowly and prepare to rest, the chemistry of life and the chemistry of repair proceeding together in that gentle equilibrium that sustains us all. The story of how we came to understand that mutations arise spontaneously, without any regard for whether they might prove useful or harmful to the organism, begins in a laboratory during the Second World War, in a collaboration between two scientists whose careful thinking would eventually reshape our understanding of evolution at its most fundamental level. Salvador Luria, an Italian émigré who had fled fascism, and Max Delbruck, a German physicist who had turned his considerable intellect toward biology, found themselves working together at Indiana University, puzzling over a question that had vexed biologists for decades. When bacteria exposed to a virus developed resistance to that virus, were they somehow responding to the challenge by generating useful mutations, or had resistant variants already existed in the population before any exposure occurred, arising through random changes that had nothing whatsoever to do with their eventual usefulness? The experiment they designed in 1943 was elegant in its simplicity and profound in its implications, and it relied on a statistical insight that Luria reportedly conceived while watching a colleague play a slot machine. If mutations arose in response to selection, appearing only when bacteria encountered the virus, then you would expect to find roughly similar numbers of resistant bacteria in each of many parallel cultures grown under identical conditions. The statistical distribution would be narrow, predictable, clustered around some average value. But if mutations occurred randomly during the growth of each culture, arising spontaneously at unpredictable moments without any knowledge of future challenges, then you would see something quite different. A mutation that happened to occur early in the growth of a culture would be copied into many daughter cells, producing a large clone of resistant bacteria, while a mutation occurring late would leave only a small number of descendants. The distribution across many cultures would be enormously variable, with some cultures containing many resistant bacteria and others containing very few. What Luria and Delbruck observed, in experiment after experiment, was precisely this high variability, this fluctuation that gave their test its name. The mutations that conferred resistance to the bacteriophage virus were not arising in response to the virus at all. They were occurring spontaneously, randomly, during ordinary cell division, with no foresight and no purpose, distributed in time according to chance alone. This finding, published in the journal Genetics in 1943, provided the first rigorous experimental proof that mutations are random events with respect to their selective consequences, arising through molecular accidents that have nothing to do with the needs of the organism. The implications rippled outward through biology, confirming Darwin's vision of variation as raw material upon which natural selection acts, rather than some directed process that generates useful changes when they are needed. For this work, along with their many other contributions to understanding the genetics of viruses and bacteria, Salvador Luria and Max Delbruck shared the Nobel Prize in Physiology or Medicine in 1969, together with Alfred Hershey, who had made complementary discoveries about the nature of viral genetics. The prize recognized not merely a single experiment but a transformation in how we think about the origins of biological variation, the understanding that mutations bubble up from the molecular level through processes that are, in their essence, random with respect to fitness, governed by physical and chemical events occurring far below the scale of organismal awareness. In the decades following Luria and Delbruck's foundational experiment, scientists developed increasingly precise methods for measuring just how often these spontaneous mutations occur, and the numbers they obtained are genuinely remarkable for their smallness and their consistency. In the bacterium Escherichia coli, that workhorse of molecular biology, careful measurements have established that the spontaneous mutation rate is approximately 5 times 10 to the negative 10th power per base pair per generation, which is to say roughly 1 mutation for every 2 billion base pairs copied during a single round of cell division. Since the E. coli genome contains approximately 4.5 million base pairs, this translates to roughly 1 mutation every 400 or so generations, meaning that the vast majority of bacterial cell divisions proceed without introducing any new genetic changes whatsoever. The precision of DNA replication that these numbers imply is really quite extraordinary when you pause to consider what is being accomplished. The molecular machinery responsible for copying genetic information must somehow select the correct nucleotide from among 4 possibilities at each of millions of positions, and it must do so with an accuracy exceeding 99.99999%. The processes that achieve this remarkable fidelity involve multiple layers of error correction, proofreading mechanisms that catch mistakes shortly after they are made, and mismatch repair systems that patrol the newly synthesized DNA strand looking for anomalies. But even with all these safeguards in place, some errors inevitably slip through, and these become the mutations that fuel evolutionary change. In humans and other complex organisms, the germline mutation rate, which is to say the rate at which mutations arise in the cells that will give rise to eggs and sperm and thus be passed to the next generation, has been measured with increasing precision through comparison of genome sequences between parents and offspring. The current best estimates place this rate at approximately 1 to 2 times 10 to the negative 8th power per base pair per generation, which is roughly 20 to 50 times higher than the mutation rate in E. coli. Given that the human genome contains approximately 3 billion base pairs, this translates to somewhere between 30 and 100 new mutations in each child compared to their parents, depending on parental age and other factors. Most of these mutations occur in the father's germline, accumulating with each passing year as sperm-producing cells continue to divide throughout adult life, while the mother's contribution is smaller because her egg cells completed most of their divisions before she was born. In 1991, the American geneticist John Drake published a remarkable analysis that revealed an unexpected pattern underlying the seeming diversity of mutation rates across different organisms. When Drake calculated the mutation rate not per base pair but per genome per generation, he found that this quantity remained roughly constant across a wide range of microorganisms, hovering around a value of approximately 0.003 mutations per genome per replication. Whether looking at small viral genomes containing only a few thousand base pairs or at much larger bacterial genomes containing millions of base pairs, the number of mutations per complete genome copy remains surprisingly similar. This constancy suggests that natural selection has tuned the fidelity of DNA replication in each organism to achieve some optimal balance between the costs of maintaining extremely high-fidelity copying machinery and the costs of accumulating too many deleterious mutations. From the perspective of quantum mechanics, the spontaneous mutation rate represents something quite fundamental. The probability that the physical processes of proton tunneling and other quantum phenomena will cause an error in the genetic code during any given replication event. The positions of hydrogen atoms within the double helix are not fixed but rather exist as probability distributions, and there is always some finite chance that a proton will tunnel into an alternative location, creating a tautomeric form of a nucleotide base that pairs incorrectly during replication. These quantum probabilities, integrated over the brief window of time during which the replication machinery passes each base pair, determine the baseline rate at which spontaneous mutations arise. The chemical instability of DNA, which proceeds continuously whether replication is occurring or not, contributes additional opportunities for mutation through lesions that must be correctly repaired if the genetic information is to be preserved. One of the most common forms of spontaneous DNA damage is depurination, the hydrolytic cleavage of the bond connecting purine bases, which are adenine and guanine, to the sugar phosphate backbone of the DNA strand. This reaction occurs with surprising frequency, and current estimates suggest that each human cell loses roughly 5,000 purine bases every single day through this spontaneous chemical process. The resulting gaps in the DNA, called UBA6 sites or AP sites, must be recognized and repaired by the cellular machinery before the next round of replication. Because if a replication fork encounters an UBA6 site, the copying machinery has no template information to guide the selection of the correct nucleotide, and an error becomes nearly inevitable. Another common form of spontaneous damage is the deamination of cytosine, which removes an amino group from this nucleotide base and converts it to uracil. Uracil is not normally found in DNA, belonging instead to the alphabet of RNA, and it pairs with adenine rather than guanine as cytosine should. If this lesion is not repaired before replication, a CG base pair will be converted to a TA base pair in one of the daughter strands, permanently altering the genetic sequence. The rate of cytosine deamination has been estimated at roughly 100 to 500 events per cell per day under normal physiological conditions, and this number increases substantially when DNA is single-stranded or when cytosine residues have been modified by methylation. The cellular repair machinery includes specific enzymes called uracil DNA glycosylases that recognize and remove uracil bases from DNA, initiating a repair process that can restore the correct sequence. But some deamination events inevitably escape repair and become fixed as mutations. These rates of spontaneous damage, the 5,000 depurinations and the hundreds of cytosine deaminations occurring in each cell every day, might seem alarming when stated so baldly. But the efficiency of DNA repair systems means that only a tiny fraction of these lesions ever become permanent mutations. The repair machinery continuously patrols the genome, recognizing damage and initiating the complex multi-step processes that restore the correct sequence, and the overwhelming majority of lesions are corrected without leaving any trace of their former existence. But the small percentage that escape repair, combined with the errors that slip through the proofreading mechanisms during replication itself, produce the steady trickle of spontaneous mutations that we observe in population genetic data. One of the intriguing predictions of the quantum tunneling model of mutation is that spontaneous mutation rates should show a particular kind of temperature dependence, distinct from what would be expected if mutations arose purely through classical over-the-barrier reaction mechanisms. In classical chemistry, reaction rates typically increase exponentially with temperature according to the Arrhenius equation, because higher temperatures provide more molecules with sufficient kinetic energy to overcome activation barriers. Quantum tunneling, by contrast, shows a weaker temperature dependence because particles do not need to surmount the barrier at all. They simply pass through it, and the probability of tunneling depends primarily on the barrier width and height rather than on thermal energy. Experimental measurements of spontaneous mutation rates at different temperatures have provided some support for quantum contributions to mutagenesis, though the interpretation remains contested. The temperature dependence of certain types of mutations, particularly those involving proton transfer between base pairs, appears to be consistent with tunneling predictions rather than purely classical expectations. However, the complexity of biological systems makes clean tests difficult, because temperature affects many aspects of cellular physiology beyond the simple kinetics of chemical reactions, and separating quantum effects from the myriad other temperature-dependent processes remains an ongoing challenge. The field continues to gather evidence bearing on this question, with increasingly sophisticated experimental techniques bringing ever more precise measurements of mutation rates under controlled conditions. The evolutionary biologist Michael Lynch has devoted much of his career to understanding how mutation rates themselves evolve, and his work has revealed fundamental principles governing the fidelity of genetic transmission. Lynch recognized that maintaining extremely accurate DNA replication is costly, requiring the production of elaborate proofreading enzymes and mismatch repair proteins, and that natural selection can only push mutation rates so low as the benefit of further reduction outweighs the cost. In very large populations, where even slightly deleterious mutations are efficiently removed by selection, there is strong evolutionary pressure to minimize the mutation rate, and organisms can afford to invest heavily in replication fidelity. But in small populations, where random genetic drift becomes more powerful relative to selection, the benefits of reducing the mutation rate are diminished because slightly deleterious variants can persist and fix through chance alone regardless of how rare new mutations might be. This reasoning led Lynch to propose the concept of the drift barrier, a fundamental limit below which selection cannot push the mutation rate because further reductions would require investments in fidelity that are themselves too small to be reliably favored by selection in populations of finite size. The drift barrier represents a kind of floor set by population genetics, a minimum mutation rate that persists not because lower rates are physically impossible but because the selective advantage of achieving them is too small to overcome the random fluctuations inherent in finite populations. Lynch's mathematical analyses suggest that observed mutation rates in many organisms may be close to this drift barrier, hovering near the lowest values that selection can maintain given each species' typical population size and other demographic parameters. The quantum mechanical perspective adds another layer to this picture, because the processes of proton tunneling and tautomeric fluctuation that we discussed earlier set their own physical limits on how accurate DNA replication can possibly be. No matter how elaborate the proofreading machinery, no matter how efficient the mismatch repair systems, there remains some irreducible probability that a quantum fluctuation will cause a base to exist in an incorrect form at the critical moment of replication. These quantum probabilities establish a physical floor beneath the drift barrier, a fundamental limit arising from the nature of chemical bonds and the uncertainty inherent in the positions of subatomic particles. Whether this physical limit or the evolutionary drift barrier represents the more stringent constraint likely varies among organisms and perhaps among different types of mutations within the same organism. The interplay between physics and evolution in determining mutation rates illustrates how biological phenomena emerge from the interaction of multiple levels of causation. At the deepest level, quantum mechanics governs the behavior of the electrons and protons that constitute the nucleotide bases, setting the probabilities of tautomeric shifts and the rates of spontaneous chemical damage. At the next level, natural selection shapes the enzymes and repair systems that determine how effectively potential mutations are caught and corrected. At the population level, drift barriers and the effective population size determine how stringently selection can act on small differences in mutation rate. All these factors combine to produce the spontaneous mutation rates that we measure in laboratories and that drive the evolutionary changes we observe in nature. The numbers themselves. A mutation rate of 5 times 10 to the negative 10th per base pair per generation in bacteria, 1 to 2 times 10 to the negative 8th in human germlines, a universal rate near 0.003 mutations per genome per replication across microbes, 5,000 depurinations and hundreds of cytosine deaminations per cell per day, may seem abstract in isolation, but they represent the quantitative foundation upon which all of evolutionary biology rests. These are the rates at which new variation enters populations, the raw material from which natural selection constructs adaptations, and from which genetic drift produces random change. Without spontaneous mutation, evolution would grind to a halt. But if mutation rates were too high, the genetic information accumulated over millions of years would dissolve into meaningless noise. The values we observe represent a balance, maintained by the physical chemistry of DNA and tuned by billions of years of evolutionary adjustment, that permits both the stability necessary for faithful inheritance and the variation necessary for adaptive change. As we have traced the history of mutation rate measurement from Luria and Delbruck's wartime experiments through Drake's universal rate and Lynch's drift barrier to the quantum mechanical interpretations of modern biophysics, we see how a single conceptual thread connects these diverse investigations. Mutations arise spontaneously, randomly with respect to their effects on fitness, through molecular processes governed ultimately by the laws of physics operating at scales far removed from our everyday experience. The clicking of a Geiger counter registering the decay of radioactive atoms and the appearance of a new mutation in a bacterial culture share a common statistical character: the randomness of quantum events that we can predict only in aggregate, never in the individual case. This understanding, hard-won through decades of careful experiment and theoretical analysis, forms the foundation upon which all our knowledge of genetic change must rest. And now we turn our attention upward, far beyond the familiar world of laboratory benches and microscope slides, to consider the messages that arrive continuously from the vast reaches of space, carrying within them energies sufficient to alter the very molecules that define living things. These cosmic visitors have been streaming through the universe since its earliest moments, and they pass through our bodies even now, as you rest quietly and listen to these words, though their effects are so subtle and so distributed across the trillions of cells within you that they cause no immediate sensation whatsoever. Only the occasional quantum mechanical interaction that may, in one cell among billions, leave a small signature upon the double helix of your DNA. The story of how we came to understand these invisible travelers begins in the early 20th century, when scientists noticed something rather puzzling about the electrical conductivity of air. It had been observed that air, even in sealed containers, always seemed to conduct a small amount of electricity, and the prevailing explanation was that this conductivity must be caused by radiation emanating from radioactive elements in the earth beneath our feet. This seemed a perfectly sensible hypothesis, and it suggested a straightforward test. For if the radiation truly came from the ground, then the electrical conductivity of air should decrease as one moved further from the Earth's surface, should diminish steadily as one ascended into the sky. In 1912, an Austrian physicist named Victor Hess decided to investigate this question directly, and he did so in a manner that combined genuine scientific courage with methodical experimental care. Hess undertook a series of balloon flights, ascending to altitudes that were considered quite dangerous at the time, carrying with him sensitive instruments called electrometers that could measure the ionotation of the air around him. He made these ascents both during the day and at night, and even during a solar eclipse, reasoning that if the radiation came from the sun, it should vary with solar conditions. What Hess discovered was precisely the opposite of what the earthly radiation hypothesis predicted, for he found that the ionotation of the air actually increased as he rose higher, becoming several times stronger at an altitude of 5,000 meters than it was at the surface. This finding could only be explained if the ionizing radiation was coming not from below but from above, from somewhere beyond the Earth's atmosphere, and Hess correctly concluded that he had discovered a form of radiation of extraterrestrial origin. He called this phenomenon Hohenstrahlung, which translates roughly as high-altitude radiation, though it soon became known more evocatively as cosmic radiation or cosmic rays. For this discovery, Victor Hess was awarded the Nobel Prize in Physics in 1936, sharing the honor with Carl Anderson, who had discovered the positron. The recognition came more than 2 decades after the original balloon flights, which speaks to the way scientific understanding sometimes unfolds gradually, as the full significance of an observation becomes clear only through years of subsequent investigation. We now know that cosmic radiation consists primarily of particles that have been accelerated to enormous energies by processes occurring throughout the galaxy and beyond, in the magnetic fields surrounding supernova remnants, in the vicinity of black holes, and in other cosmic environments where matter and energy interact on scales almost impossible to comprehend. When these particles enter the Earth's atmosphere, they collide with the atoms of nitrogen and oxygen that make up the air, producing cascades of secondary particles that shower down upon the surface below. The primary cosmic radiation, meaning the particles that arrive from space before interacting with the atmosphere, consists predominantly of protons, which make up approximately 87% of the total flux. Alpha particles, which are helium nuclei consisting of 2 protons and 2 neutrons bound tightly together, account for roughly 12%, and the remaining 1% consists of heavier nuclei, including atoms of carbon, oxygen, iron, and other elements that have been stripped of their electrons and accelerated to velocities approaching the speed of light. These proportions may seem like dry statistics, but they carry significant implications for the biological effects of cosmic radiation, because particles of different masses and charges interact with living tissue in fundamentally different ways. To understand these differences, we must introduce a concept called linear energy transfer, commonly abbreviated as LET, which describes how much energy a particle deposits per unit length as it travels through matter. A particle with high linear energy transfer deposits its energy in a concentrated fashion along a narrow track, while a particle with low linear energy transfer spreads its energy more diffusely over a larger volume. The quantum mechanical basis of linear energy transfer lies in the probability of electromagnetic interactions between the passing charged particle and the electrons of atoms along its path, with these probabilities depending on the particle's charge and velocity in ways that can be calculated from the fundamental equations of quantum electrodynamics. Low-let radiation, such as X-rays and gamma rays, tends to cause damage that is scattered somewhat randomly throughout the cell, with individual ionizations occurring at various points along the trajectory of the photons or the secondary electrons they produce. This scattered pattern of damage is certainly capable of harming DNA, but the cell's repair mechanisms have evolved to deal efficiently with such dispersed injuries, snipping out damaged bases and filling in the gaps with fresh nucleotides as though mending small holes in a piece of fabric. High-let radiation, on the other hand, creates a very different situation. For particles like alpha particles and heavy nuclei carve dense tracks of ionization through biological tissue, depositing enormous amounts of energy in very small volumes. And this concentrated deposition of energy can produce forms of damage that are far more difficult for the cell to repair. The physicist Eleanor Blakely, working at Lawrence Berkeley National Laboratory, devoted much of her career to understanding exactly how heavy ions interact with living cells and the DNA within them. Her research, conducted over many decades beginning in the 1970s, helped establish the scientific foundation for understanding the biological effects of the kinds of radiation that astronauts encounter beyond the protective shield of Earth's atmosphere. Blakely and her colleagues used the particle accelerators at Berkeley to produce beams of heavy ions, atoms of elements like carbon, neon, and iron that had been accelerated to high energies, and they exposed cells and organisms to these beams while carefully measuring the biological consequences. What they found was that heavy ions are remarkably efficient at killing cells and inducing mutations, far more efficient per unit of deposited energy than low-let radiation like X-rays, and this enhanced effectiveness stems from the clustered nature of the damage they produce. The concept of track structure helps illuminate why heavy ion damage is so biologically significant. When a heavy ion passes through a cell nucleus, it does not simply nick the DNA at one or two points along its path but rather creates a corridor of destruction, a track several nanometers wide in which multiple ionizations occur in close proximity. If this track happens to cross a DNA molecule, it may produce not just a single strand break, which the cell can usually repair by using the complementary strand as a template, but a double-strand break in which both sugar-phosphate backbones are severed, leaving the double helix completely fractured. Even more problematic, the clustered ionizations along a heavy ion track can produce multiple double-strand breaks in close proximity, along with oxidized bases, crosslinks between DNA strands, and other forms of damage all jumbled together in what scientists sometimes call a multiplied damaged site or a clustered damage site. These clustered damage sites present the cellular repair machinery with a formidable challenge, for the enzymes that normally excise damaged regions and fill in gaps must somehow contend with a tangle of injuries occurring within a small stretch of DNA, and the probability of error during repair increases substantially when the damage is complex rather than simple. Moreover, when the repair of a double-strand break goes awry, the consequences can include chromosomal aberrations, meaning that pieces of chromosomes become rearranged in ways that may activate oncogenes or inactivate tumor suppressor genes, potentially setting the stage for cancer to develop years or decades later. The relevance of these laboratory findings to human health became dramatically apparent through studies of astronauts who spend extended periods in space, where the Earth's atmosphere and magnetic field no longer provide their usual protection against cosmic radiation. Perhaps the most famous of these studies involved the astronaut Scott Kelly, who spent nearly 1 year aboard the International Space Station from 2015 to 2016, while his identical twin brother Mark Kelly remained on Earth. This twin study provided a unique opportunity to compare biological changes in an astronaut with those in a genetically identical individual who had not been exposed to the space environment. And among the many findings was a notable increase in chromosomal aberrations in Scott Kelly's cells during and after his spaceflight. Chromosomal aberrations are structural changes in chromosomes that can be detected by various laboratory techniques, and they include translocations, in which pieces of one chromosome become attached to a different chromosome, inversions, in which a segment of a chromosome becomes flipped around, and other rearrangements that alter the normal architecture of the genome. The increase in such aberrations observed in Scott Kelly's cells was consistent with the known effects of space radiation, particularly the heavy ion component of galactic cosmic rays. And while the long-term health implications of these changes remain uncertain, the findings underscored the biological reality of cosmic radiation damage in a way that captured considerable public attention. The work of Francis Cucinotta, who spent many years at NASA studying space radiation risks before moving to academic positions, has been instrumental in quantifying the dangers that cosmic radiation poses to astronauts, particularly those who might one day travel beyond low Earth orbit to destinations like Mars. Cucinotta and his colleagues developed models that attempt to predict cancer risk based on the type and amount of radiation exposure, incorporating the biological effectiveness of different particles and the uncertainties inherent in extrapolating from laboratory data to actual human risk. Their analyses have shown that a round trip to Mars, which might involve several years of exposure to galactic cosmic rays without the protection of Earth's magnetic field, could significantly increase an astronaut's lifetime risk of developing cancer, with the heavy ion component of the radiation being particularly concerning because of its ability to produce the kinds of clustered DNA damage that are most likely to lead to carcinogenesis. Even in low Earth orbit, astronauts encounter regions where cosmic radiation exposure is elevated, and one of the most notable of these is the South Atlantic anomaly, a region over the southern Atlantic Ocean and parts of South America where the Earth's inner Van Allen radiation belt dips unusually close to the surface. This anomaly results from the fact that the Earth's magnetic field is not perfectly aligned with its rotational axis and is not perfectly symmetrical. And in this particular region, the magnetic field is weaker than elsewhere at comparable altitudes, allowing charged particles from the radiation belt to penetrate to lower altitudes. Spacecraft passing through the South Atlantic anomaly experience elevated radiation levels, and the effects are noticeable in ways both biological and electronic. The sensitive electronics aboard spacecraft are vulnerable to the energetic particles encountered in the South Atlantic anomaly, and engineers must design systems to withstand or recover from the occasional bit flips and glitches that occur when a charged particle deposits its energy in a microchip. Similar concerns apply to the DNA of astronauts passing through this region, for the same particles that can corrupt a computer's memory can also ionize molecules within a living cell, and though the momentary passage through the anomaly is brief, astronauts on the International Space Station traverse it multiple times per day as the station circles the Earth in its orbit. Yet we need not travel to space to encounter cosmic radiation and its biological effects, for one particularly important source of high-let radiation exposure exists in many homes and buildings on the Earth's surface. Radon is a radioactive gas that forms as part of the decay chain of uranium, which is present in varying concentrations in rocks and soil throughout the world. Being a gas, Radon can seep up from the ground and accumulate in enclosed spaces like basements and lower floors of buildings. And being radioactive, it decays by emitting alpha particles, those same helium nuclei that constitute 12% of primary cosmic radiation. When radon gas is inhaled, it enters the lungs along with ordinary air, and when radon atoms decay within the respiratory tract, they release alpha particles directly into the delicate tissue lining the bronchi and bronchioles. These alpha particles have high linear energy transfer, depositing their energy in dense tracts through the cells they traverse, and they can produce the same kinds of clustered DNA damage that we have been discussing, including double-strand breaks and multiply damaged sites. The connection between radon exposure and lung cancer has been established through extensive epidemiological studies, beginning with observations of elevated lung cancer rates among uranium miners and extending to studies of residential radon exposure in the general population. Today, radon is recognized as the leading cause of lung cancer among people who have never smoked, and the second leading cause overall after smoking itself, accounting for an estimated 20,000 or more lung cancer deaths per year in the United States alone. The alpha particles emitted by radon and its decay products damage the DNA of bronchial epithelial cells, inducing mutations that can accumulate over years and decades of exposure. And some of these mutations may eventually affect genes that control cell division, leading to the uncontrolled proliferation that characterizes cancer. The probability that an alpha particle or any other ionizing particle will interact with a DNA molecule and cause a particular type of damage can be expressed in terms of a quantity called a cross-section, which has units of area and represents, in a sense, the effective target size that the DNA presents to the incoming particle. This cross-section is not simply the geometrical size of the DNA molecule, but rather a quantum mechanical quantity that depends on the energy and charge of the particle, the electronic structure of the target molecule, and the specific type of interaction being considered. Calculating these cross-sections requires the full apparatus of quantum mechanics, including treatments of how the electromagnetic field of the passing charged particle couples to the bound electrons of the target atoms, how energy is transferred in these interactions, and what the probabilities are for various outcomes ranging from simple ionizations to complex bond breakages. The results of such calculations, often performed using sophisticated computational methods, help explain why different types of radiation have different biological effectiveness, why a given amount of energy deposited by alpha particles causes more biological damage than the same amount of energy deposited by beta particles or gamma rays, and why the heavy ions found in cosmic radiation are so efficient at producing the kinds of clustered DNA damage that pose the greatest challenge to cellular repair mechanisms. As you rest now, cosmic ray particles are passing through your body at a rate of roughly 1 particle per square centimeter per minute at sea level, most of them secondary particles produced when primary cosmic rays collide with the upper atmosphere. The vast majority of these particles pass through without interacting with anything important, for your body is mostly empty space at the atomic level, and even your DNA, stretched end to end, would form a thread so thin that random particles are unlikely to strike it. Yet occasionally, inevitably, an interaction does occur, and a small quantum of damage is registered somewhere in your genome, a testament to the fact that we live in a universe suffused with energetic particles, remnants of stellar explosions and processes occurring in distant reaches of space. And so the study of cosmic radiation and its effects on DNA connects the smallest scales of quantum mechanical interaction to the largest scales of astrophysical phenomena, reminding us that the molecules of life exist within a cosmic context, shaped by forces and particles that have traveled across the galaxy to reach us, carrying messages from the stars that are written, occasionally and indelibly, into the very language of our genes. And so we come now to what is perhaps the most challenging question in all of quantum biology, the question that skeptics rightly raise and that proponents must carefully address, which is simply this: how can delicate quantum effects possibly survive in the warm, wet, chemically turbulent environment of a living cell? This is not a minor technical objection, but rather a fundamental concern that strikes at the very heart of whether quantum mechanics can play any meaningful role in biological processes. And we shall spend some time with this question, turning it over gently, examining it from various angles, because understanding the challenge of decoherence helps us appreciate just how remarkable biological systems truly are, and perhaps, if the evidence continues to accumulate, how cleverly evolution may have learned to work with the quantum world rather than against it. The fundamental problem can be stated quite simply, though its implications are profound. Your body maintains a temperature of approximately 310 kelvins, which is about 37 degrees Celsius, and at this temperature molecules are in constant thermal motion, vibrating and rotating and colliding with their neighbors billions of times per second. This thermal agitation creates what physicists call noise, random fluctuations that jostle and disturb any delicate quantum state. A quantum superposition, in which a particle exists in multiple states simultaneously, is an extraordinarily fragile thing, rather like a soap bubble that can be popped by the slightest disturbance. When a quantum system interacts with its warm environment, information about its state leaks out into the surroundings, and the superposition collapses into a single definite state. This process is called decoherence, and in warm wet systems it happens astonishingly quickly, typically in femtoseconds, which are quadrillionths of a second, or even faster. The thermal energy available at body temperature is substantial compared to the energy scales of quantum effects, and this thermal energy acts like a constant barrage of tiny measurements, each one threatening to destroy whatever quantum coherence might be trying to establish itself. This challenge was addressed directly and rather devastatingly in a paper published in the year 2000 by the physicist Max Tegmark, who is now at the Massachusetts Institute of Technology but was then working on questions at the intersection of physics and neuroscience. Tegmark's paper was concerned specifically with a proposal by Roger Penrose and Stuart Hameroff that quantum effects in structures called microtubules, which are protein scaffolding inside neurons, might be responsible for consciousness. Tegmark performed careful calculations of how quickly quantum superpositions would decohere in microtubules at brain temperature, and his results were sobering for proponents of quantum consciousness. He found that decoherence times would be on the order of 10 to the minus 13 seconds or faster, far too brief for the proposed quantum computations to occur before being destroyed by environmental noise. The paper was influential and widely cited, and for many physicists it seemed to close the door on quantum effects playing any significant role in warm biological systems. If quantum coherence could not survive in microtubules, the reasoning went, then surely it could not survive in DNA either, which operates in the same warm cellular environment. The implications of Tegmark's analysis for DNA were taken seriously by researchers in the emerging field of quantum biology, and estimates of decoherence times for quantum effects in DNA are indeed extremely short. For electronic coherence, in which electrons exist in superpositions of different orbital states, Decoherence times are typically estimated at 10 to the minus 13 to 10 to the minus 14 seconds, which is to say 100 femtoseconds to 10 femtoseconds. These are almost inconceivably brief intervals of time. Light itself travels only about 30 micrometers in 100 femtoseconds, roughly the width of a few cells. It would seem that any quantum effect would be destroyed almost instantaneously, long before it could influence chemical reactions or biological processes. And yet, curiously, this apparent impossibility did not cause researchers to abandon the field. Instead, it prompted a deeper investigation into whether biological systems might have found ways to work within these constraints, or perhaps even to exploit them. One mechanism by which quantum states might be preserved despite environmental noise is known as the quantum Zeno effect, named after the ancient Greek philosopher Zeno of Elea, who proposed paradoxes about motion and change. The quantum Zeno effect refers to the somewhat counterintuitive phenomenon whereby frequent measurements or observations of a quantum system can actually prevent it from changing state. In its simplest form, if you measure a quantum system repeatedly and rapidly enough, you can freeze it in its initial state, because each measurement collapses any incipient evolution back to the starting point. This might seem like a curiosity of measurement theory, but it has been demonstrated experimentally and may have biological relevance. If certain molecular configurations act as natural observers of quantum states, repeatedly interacting with quantum particles in specific ways, they might help to stabilize those states against decoherence. The protein environment around DNA is precisely such a structured molecular configuration, and while we do not yet know whether Zeno-like effects play a role in protecting quantum states in DNA, it remains a theoretical possibility that researchers continue to investigate. A more surprising and perhaps more significant development came from the laboratory of Martin Plinio at the University of Ulm in Germany, who along with his colleagues proposed something quite remarkable, that noise and decoherence might not simply be enemies of quantum effects in biology, but might actually be essential ingredients that help quantum processes work more efficiently. This idea, sometimes called noise-assisted quantum transport or environment-assisted quantum transport, turned the conventional wisdom on its head. Rather than viewing the warm, wet environment as purely destructive of quantum coherence, Plinio and his collaborators suggested that a certain amount of environmental noise might help quantum particles navigate complex molecular landscapes more effectively than they could in either a perfectly coherent quantum regime or a purely classical regime. The noise, in effect, helps to unstick quantum particles when they become trapped in unproductive states, while enough quantum coherence remains to allow efficient wave-like exploration of multiple pathways. This Goldilocks principle, in which the noise level is neither too high nor too low but just right, might explain how biological systems achieve remarkable efficiencies in processes like photosynthesis and, potentially, in the chemistry of DNA. The work on noise-assisted transport emerged partly from studies of photosynthesis, where experiments had shown that quantum coherence persists in light harvesting complexes for much longer than expected, hundreds of femtoseconds rather than the few femtoseconds predicted by simple decoherence models. These experiments, conducted on bacterial photosynthetic systems and later on plant systems, suggested that the protein scaffolding surrounding the light-absorbing molecules was not simply a passive container but rather an active participant in preserving and channeling quantum coherence. The proteins create what is called a structured environment, in which the noise is not random and undifferentiated but rather has specific frequencies and correlations that can interact constructively with quantum states. This insight from photosynthesis research has been eagerly adopted by researchers studying DNA, because the double helix is also surrounded by a structured environment of proteins, water molecules, and ions that might serve a similar protective or channeling function. The protein scaffolding around DNA is extensive and highly organized. DNA in living cells is not a naked molecule floating freely in solution, but rather is associated with numerous proteins that regulate its activity, maintain its structure, and repair its damage. In the nuclei of eukaryotic cells like our own, DNA is wrapped around protein complexes called histones, forming a structure called chromatin. Even in simpler organisms like bacteria, DNA is associated with various proteins that help to compact it and control its function. These proteins create a local environment around the DNA that differs significantly from bulk water. The water molecules immediately adjacent to DNA and its associated proteins are more ordered and less mobile than water in the surrounding cytoplasm. This structured water shell might help to shield quantum states from some of the thermal noise of the broader environment, creating a kind of quantum protective layer. Additionally, the proteins themselves might have vibrational modes that couple constructively with quantum processes in the DNA, although this remains largely speculative at our current level of understanding. At the University of Surrey in England, the physicist Jim Al-Khalili and the molecular geneticist John Joe McFadden have been developing theoretical frameworks for understanding how quantum effects might operate in DNA despite the challenge of decoherence. Their work has emphasized that the relevant question is not whether quantum coherence can persist indefinitely in DNA, because clearly it cannot, but rather whether it can persist long enough to influence chemical reactions. A chemical reaction such as proton transfer or electron tunneling occurs on timescales that are themselves very fast, often in the femtosecond to picosecond range. If quantum coherence can survive for even a few femtoseconds, it might be sufficient to influence the outcome of a chemical process before decoherence destroys the superposition. Al-Khalili and McFadden have proposed that quantum tunneling of protons in DNA base pairs might be rapid enough to compete with decoherence, allowing quantum effects to influence mutation rates even in the warm cellular environment. Their calculations suggest that while the window for quantum effects is extremely narrow, it may not be closed entirely. An important conceptual distinction that has emerged from this work is between what researchers call trivial and non-trivial quantum effects in biology. Trivial quantum effects are those that are necessary to explain the basic structure and properties of biological molecules, but do not require any special biological adaptation. The fact that atoms form chemical bonds, that molecules have specific shapes, that light is absorbed at particular wavelengths, all of these depend on quantum mechanics, and in this sense all of biology is quantum mechanical at its foundation. However, these are considered trivial in the sense that they do not require living systems to do anything special to exploit them. Non-trivial quantum effects, by contrast, are those that require biological systems to actively maintain or protect quantum coherence, channeling it to achieve functions that would not be possible classically. The evidence for non-trivial quantum effects in biology remains controversial, though it has strengthened considerably in recent years, particularly in the areas of photosynthesis, enzyme catalysis, and magnetoception in birds. For DNA specifically, the question of whether quantum effects are trivial or non-trivial remains open. The quantum tunneling of protons in tautomeric mutations might be considered trivial in the sense that it is simply a consequence of the quantum nature of protons and does not require any special biological adaptation. However, If it turns out that the protein environment around DNA has been evolutionarily optimized to allow certain quantum processes while suppressing others, then we would be in the realm of non-trivial quantum biology. Similarly, if the repair enzymes that fix DNA damage turn out to use quantum effects to locate damage with greater efficiency than classically possible, this would represent a non-trivial exploitation of quantum mechanics. The distinction matters because it helps us understand whether quantum effects in DNA are simply background physics or whether they represent something that evolution has actively shaped and refined over billions of years. The numerical estimates of decoherence times in DNA, while extremely short, are not necessarily fatal to the idea of quantum effects in genetic processes. The key insight from recent theoretical work is that biological systems may have evolved to work within the constraints of decoherence rather than against them. Rather than trying to maintain long-lived quantum coherence, which would indeed be impossible at body temperature, DNA chemistry may exploit brief quantum fluctuations that occur naturally and continuously. Every proton in every hydrogen bond of every base pair is constantly subject to quantum uncertainty, and this uncertainty manifests as a perpetual flickering between different tautomeric states. Most of these fluctuations are quickly damped out by the environment, but occasionally one might persist long enough to be captured by DNA replication, resulting in a mutation. In this view, decoherence is not the enemy of quantum effects in DNA, but rather is part of the mechanism by which quantum fluctuations are converted into classical genetic changes that can be inherited. We might think of it this way, if a gentle analogy may be permitted. Imagine a vast car motion in which tiny waves are constantly forming and disappearing, their energy quickly dissipated by the viscosity of the water. Most of these waves never grow large enough to be noticed, but occasionally, by chance, one forms at just the right moment and in just the right place to be captured and amplified by some larger process. The decoherence of quantum states in DNA might be similar, a constant background of quantum fluctuations, most of which decay harmlessly, but some of which are captured at critical moments to produce lasting changes in the genetic code. This picture is speculative but consistent with what we know about the physics of warm biological systems, and it suggests that quantum effects in DNA may be fundamentally statistical rather than deterministic, contributing to the randomness of mutation rather than to any directed process. The challenge of maintaining quantum effects in the warm-wet environment of the cell is thus both formidable and fascinating. The skepticism exemplified by Tegmark's paper was well-founded and remains an important reminder that extraordinary claims about quantum biology require extraordinary evidence. Yet the subsequent developments in our understanding of noise-assisted transport, structured environments, and the interplay between coherence and decoherence have opened new possibilities that were not appreciated 2 decades ago. The protein scaffolding around DNA, the structured water shell, the rapid timescales of relevant chemical reactions, all of these factors might combine to create a window, however narrow, through which quantum effects can influence the chemistry of heredity. Whether this window is sufficient for quantum effects to play a biologically significant role remains one of the central questions in quantum biology, a question that researchers are actively pursuing through ever more sophisticated experiments and theoretical models. For now, we can say only that the door has not been closed, and that the warm-wet challenge, while severe, may not be insurmountable. And so we arrive now at a topic that concerns the remarkable speed of molecular repair, the way in which enzymes manage to achieve reaction rates that seem, at first consideration, almost impossible, and how quantum mechanics provides a partial explanation for this extraordinary catalytic power that keeps your DNA intact through countless chemical challenges each day. Enzymes, you see, are proteins that accelerate chemical reactions, and they do so with an efficiency that defies ordinary intuition. When we speak of enzyme catalysis, we are speaking of rate enhancements that can reach factors of 10 to the power of 17, which is to say that an enzyme can make a reaction proceed 10,000 trillion times faster than it would if left to occur spontaneously in solution. This number is difficult to comprehend in everyday terms, but perhaps you might imagine a process that would naturally take longer than the current age of the universe being completed instead in a fraction of a second. And this gives some sense of the magnitude of acceleration we are discussing, the sheer improbability of it when viewed through the lens of classical chemistry alone. The theoretical framework for understanding how enzymes achieve these remarkable speeds was developed in the 1930s, and it remains foundational to our understanding today, though it has been considerably refined and extended in the decades since. Henry Eyring, an American theoretical chemist working at Princeton and later at the University of Utah, developed what we now call transition state theory, a way of understanding chemical reaction rates in terms of an energy barrier that reactants must surmount to become products. In Eyring's formulation, which drew upon the new quantum mechanics that had recently been developed in Europe, the rate of a chemical reaction depends on the height of this energy barrier and on the temperature of the system, with higher temperatures providing more thermal energy to help molecules overcome the barrier, and lower barriers naturally allowing faster passage from reactants to products. The quantum mechanical foundations of this theory are essential to its accuracy, because Eyring understood that the behavior of atoms and electrons during chemical transformations cannot be adequately described by classical physics alone. The making and breaking of chemical bonds involves the rearrangement of electron density, the quantum mechanical phenomenon of zero-point energy, and the wave-like behavior of particles that allows them to exist in states that would be forbidden under classical mechanics. Eyring's transition state theory incorporated these quantum considerations in a way that allowed chemists to calculate reaction rates with reasonable accuracy, and it earned him widespread recognition, though not, as some felt it should have, the Nobel Prize itself. Now, within the context of DNA repair, we encounter enzymes whose catalytic power is particularly impressive, because the chemical transformations they must accomplish are both difficult and urgent. Consider the ERV-RABC nucleotide excision repair system, which operates in bacteria such as Escherichia coli to remove bulky lesions from the DNA double helix, the sort of damage caused by ultraviolet light or by certain chemical mutagens that distort the regular structure of the helix. This repair system consists of multiple protein components that work together in an elegant choreography, with ERV-RA proteins first scanning the DNA to detect distortions, then recruiting ERV-B proteins that verify the damage and prepare the site for incision, and finally ERV-C proteins that make precise cuts on either side of the lesion, allowing the damaged section to be removed and replaced with correct nucleotides. The speed and accuracy with which this system operates is remarkable, particularly when one considers the vast amount of DNA that must be surveyed and the relatively rare occurrence of damage at any particular site. The enzymes must scan millions of base pairs, recognize subtle distortions in helix geometry, and then execute precise chemical reactions to cut the DNA backbone at exactly the right positions, all while avoiding inappropriate cuts that would create more damage than they repair. The kinetics of this process, the rates at which each step occurs, reflect an evolutionary optimization that has tuned these enzymes to near perfection over billions of years of natural selection. Understanding how enzymes achieve such catalytic power at the molecular level requires computational methods of considerable sophistication, and it was for developing such methods that Henri Worshell, along with Michael Levitt and Martin Karplus, received the Nobel Prize in Chemistry in 2013. Worshell, who was born in Israel and has spent much of his career at the University of Southern California, pioneered techniques for simulating enzyme catalysis using computer models that combine quantum mechanical treatments of the chemical reaction itself with classical mechanical treatments of the surrounding protein environment. This hybrid approach, often called quantum mechanics molecular mechanics or QMMM, allows researchers to calculate the energy landscapes that govern enzyme reactions with accuracy that would be impossible using either quantum or classical methods alone. What these computational studies revealed, and what Worshell's work demonstrated with particular clarity, is that enzymes achieve their remarkable rate enhancements not through any single mechanism but through a combination of effects that work together synergistically. The enzyme active site, the pocket or cleft where the chemical transformation occurs, provides an environment that stabilizes the transition state of the reaction, the fleeting high-energy configuration through which reactants must pass to become products. By binding more tightly to the transition state than to either reactants or products, the enzyme effectively lowers the energy barrier for the reaction, accelerating the rate according to the principles of Ehring's transition state theory. Additionally, the enzyme positions the reactive groups in precisely the correct orientation, excludes water molecules that might interfere with the reaction, and provides catalytic residues that participate directly in the chemistry through acid-base reactions or through temporary covalent bond formation. But even with all these factors accounted for, researchers found that classical transition state theory could not fully explain the observed rates of certain enzyme-catalyzed reactions, and this led to a renewed appreciation for the role of quantum mechanical tunneling in enzyme catalysis, a phenomenon that allows particles to pass through energy barriers rather than having to climb over them. The evidence for tunneling in enzymatic reactions came first from studies of enzymes other than those involved in DNA repair, but the principles established by this work apply broadly to any enzyme that catalyzes reactions involving hydrogen transfer, which includes many of the polymerases and repair enzymes that operate on DNA. Alcohol dehydrogenase, an enzyme found in the liver that oxidizes ethanol to acetaldehyde, provided some of the earliest and clearest evidence for hydrogen tunneling in enzyme catalysis. This enzyme transfers a hydrogen atom, technically a hydride ion consisting of a proton and 2 electrons, from alcohol to a cofactor molecule, and researchers noticed that the kinetics of this reaction showed unusual features that could not be explained by classical over-the-barrier mechanisms alone. When the normal hydrogen was replaced with deuterium, a heavier isotope of hydrogen, the reaction slowed down more than classical theory predicted it should, a phenomenon called an anomalous kinetic isotope effect that is characteristic of tunneling contributions. The lighter hydrogen particle, with its smaller mass, has a longer de Broglie wavelength and therefore a greater probability of tunneling through the energy barrier, and this quantum effect contributes substantially to the overall reaction rate. The work of Nigel Scruton and his research group at the University of Manchester has been particularly illuminating in quantifying the contribution of tunneling to enzyme catalysis. Through careful kinetic measurements and isotope effect studies on a variety of enzymes, Scruton and colleagues have shown that hydrogen tunneling can contribute 50% or more of the total catalytic rate in certain cases, meaning that without tunneling, These enzymes would operate at less than half their observed speed. This is not a minor quantum correction to an essentially classical process, but rather a fundamental aspect of how these biological catalysts function, a feature that evolution has apparently optimized over countless generations of natural selection for faster and more efficient enzymes. One of the most intriguing concepts to emerge from this research is the idea of tunneling-ready active site conformations, protein configurations that are particularly favorable for quantum mechanical tunneling to occur. Enzymes are not rigid structures but rather dynamic molecules that constantly fluctuate between different conformational states, some more open and some more closed, some with reactive groups positioned close together and others with them farther apart. The tunneling-ready conformation is one in which the donor and acceptor atoms for the hydrogen transfer are positioned at just the right distance, neither too close nor too far, and at just the right orientation for the quantum mechanical wave function of the hydrogen to overlap efficiently with the destination site. When the protein fluctuates into this tunneling-ready state, the probability of tunneling increases dramatically, and the reaction can proceed rapidly through the barrier rather than waiting for enough thermal energy to accumulate to push the system over the top. The coupling between protein dynamics and quantum tunneling events represents a fascinating intersection of classical and quantum phenomena, with the large-scale thermal motions of the protein, which follow classical statistical mechanics, creating the conditions that allow small-scale quantum effects to manifest in the chemical reaction. This coupling means that the protein is not merely a static scaffold holding reactive groups in place, but rather an active participant in the catalysis, a dynamic machine whose fluctuations are tuned by evolution to optimize the quantum mechanical aspects of the reaction it catalyzes. The timescales of these protein motions, typically in the range of picoseconds to nanoseconds, match well with the timescales relevant for tunneling, suggesting that this coupling is not accidental but rather a designed feature of enzyme catalysis. The research of Judith Klinman at the University of California, Berkeley, has been particularly important in establishing that enzyme evolution optimizes tunneling contributions to catalysis, that natural selection has acted on protein sequences to create active sites where quantum effects are maximized. Klinman's work on a variety of enzymes has shown that mutations which disrupt the precise geometry of the active site, even mutations that do not directly contact the reactive groups, can substantially reduce tunneling efficiency and thereby slow the reaction. This observation implies that the entire architecture of the enzyme, including residues far from the active site, contributes to creating an environment where tunneling can occur efficiently, and that evolution has explored sequence space to find solutions that optimize this quantum mechanical aspect of catalysis along with all the classical factors that contribute to rate enhancement. One of the diagnostic features used to identify tunneling contributions to enzyme catalysis is the temperature dependence of the reaction rate, or more precisely, the degree to which that temperature dependence deviates from the predictions of classical transition state theory. In classical over-the-barrier reactions, higher temperatures provide more thermal energy and therefore faster rates, following the exponential relationship described by the Arrhenius equation, a formula that relates reaction rate to temperature through the activation energy of the process. Tunneling, however, is fundamentally temperature-independent in its quantum mechanical essence, because the probability of tunneling through a barrier depends on the shape and height of the barrier and on the mass of the particle, but not directly on the temperature of the system. When tunneling contributes substantially to an enzyme reaction, the overall temperature dependence becomes weaker than classical theory predicts, and careful measurement of this deviation can allow researchers to estimate the fraction of the reaction that proceeds through the tunneling pathway. Applying these concepts to DNA polymerase, the enzyme responsible for copying genetic information during cell division, we encounter a system where both speed and accuracy are essential, and where quantum effects may contribute to both. DNA polymerase achieves error rates of approximately 10 to the power of -9, meaning that only about 1 in a billion nucleotides is incorporated incorrectly, and this extraordinary fidelity is essential for preserving genetic information across generations. The enzyme accomplishes this feat through a combination of mechanisms, including careful selection of the correct nucleotide based on complementary base pairing, a conformational change that checks the geometry of the nascent base pair before allowing the chemical reaction to proceed, and a proofreading exonuclease activity that can remove incorrectly incorporated nucleotides shortly after they are added. The chemical step of DNA synthesis, the formation of a phosphodiester bond that extends the growing DNA chain by one nucleotide, involves the transfer of a phosphate group and is facilitated by metal ions in the enzyme active site. While this reaction does not involve hydrogen transfer in the same way that alcohol dehydrogenase does, there are aspects of the catalysis where quantum effects may still be relevant, including the precise positioning of atoms in the transition state and the dynamics of the conformational changes that accompany catalysis. The extraordinary precision with which DNA polymerase positions its substrates, achieving geometric accuracy measured in tenths of angstroms, creates conditions where quantum mechanical phenomena might contribute to the discrimination between correct and incorrect nucleotides, though this remains an active area of research. The broader lesson from studies of quantum effects in enzyme catalysis is that the boundary between classical and quantum mechanical behavior in biological systems is not as sharp as one might initially assume. Enzymes operate at a scale where both classical and quantum effects are relevant, and evolution has apparently found ways to harness quantum phenomena to enhance catalytic efficiency. The proteins that maintain and replicate your DNA are products of billions of years of optimization, and that optimization has tuned them to exploit every available mechanism for achieving the speed and accuracy that life requires, including mechanisms that depend on the wave-like behavior of hydrogen atoms and the probability of tunneling through energy barriers. As you rest now, perhaps drifting towards sleep, these enzymes continue their work within your cells, scanning DNA for damage, recognizing distortions, executing precise chemical reactions at rates that would be impossible without the quantum mechanical effects that evolution has woven into their structure. The proteins flex and fluctuate, sampling conformational space until they find the tunneling-ready states that allow chemistry to proceed at speeds approaching the physical limits of what is possible. And all of this occurs without any conscious effort on your part. A background hum of molecular repair and replication that maintains the integrity of your genetic information through each cycle of day and night. The mathematics of transition state theory and tunneling probability that we have discussed are the language in which these processes are described by scientists, the formal framework that allows quantitative predictions to be compared with experimental observations. But the underlying reality is simply that your DNA repair enzymes are very, very good at their jobs, accomplishing chemical transformations with a speed and precision that reflects the accumulated wisdom of evolutionary time, a wisdom that includes, it seems, an intuitive grasp of quantum mechanics encoded in the shapes and dynamics of folded proteins. And with that comforting thought, of molecular machines optimized across billions of years working diligently on your behalf, you might allow yourself to rest a little deeper, knowing that the chemistry of repair proceeds regardless of whether you are awake to contemplate it. In the year 1956, in a laboratory at Washington University in St. Louis. A biochemist named Arthur Kornberg was engaged in a rather quiet and methodical pursuit, one that would eventually illuminate the very machinery by which life copies itself across generations. Kornberg, who had trained as a physician but found himself drawn inexorably toward the fundamental chemistry of living systems, had set himself the task of understanding how cells manufacture new DNA, and in particular, whether this process might be carried out by a specific enzyme that could be isolated and studied in a test tube. At the time, the double helical structure of DNA had only recently been described by Watson and Crick, and the question of how this elegant molecule actually replicated remained largely mysterious, a problem that many scientists were approaching from various angles with varying degrees of success. Kornberg's approach was characteristically patient and systematic, involving the careful fractionation of bacterial extracts and the testing of each fraction for the ability to incorporate nucleotide building blocks into growing DNA chains. After considerable effort, he and his colleagues succeeded in purifying an enzyme from Escherichia coli that could indeed synthesize DNA in the test tube, provided it was given a template strand to copy and a supply of the 4 nucleotide triphosphates that serve as raw materials for new DNA. This enzyme, which came to be known as DNA Polymerase I, was the first of many such copying enzymes to be discovered, and its isolation represented a fundamental advance in our understanding of the molecular basis of heredity. For this work, Kornberg was awarded the Nobel Prize in Physiology or Medicine in 1959, sharing the honor with Severo Ochoa, who had made parallel discoveries concerning the synthesis of RNA. What makes this story particularly relevant to our discussion of quantum mechanics and genetic change is that DNA polymerase, in all its various forms across all domains of life, must accomplish something rather remarkable, which is to select the correct nucleotide partner for each position along the template strand with extraordinary accuracy, and it must do so while operating at speeds that can exceed 1,000 nucleotides per second in some bacterial systems. The question of how this accuracy is achieved, and the extent to which quantum mechanical effects contribute to this achievement, has occupied researchers for decades and continues to yield insights that are both subtle and profound. To appreciate the precision involved, it helps to consider the error rate at each level of the replication process, for cells have evolved not one but several successive mechanisms for ensuring fidelity, each contributing its own reduction in the overall error rate. The first level involves the initial selection of the correct nucleotide by the polymerase enzyme itself, and at this stage, the enzyme typically discriminates against incorrect bases with an accuracy of roughly 1 in 10,000, which we might write as 10 to the power of -4. This means that for every 10,000 nucleotides incorporated, approximately 1 incorrect nucleotide might slip through based on selection alone, which may seem reasonably accurate until one considers that a human genome contains roughly 6 billion base pairs that must be copied each time a cell divides. The second level of fidelity arises from a proofreading function that is built into many polymerases, a kind of molecular backspace key that allows the enzyme to recognize when it has made a mistake and excise the offending nucleotide before continuing. This proofreading activity typically provides an additional 100-fold improvement in accuracy, reducing the error rate by a factor of 10 to the power of -2. The third level involves a system of enzymes that patrol newly synthesized DNA after replication is complete, searching for mismatched base pairs that somehow escaped both selection and proofreading, and this mismatch repair system contributes yet another 100-fold reduction in errors. When all 3 levels work together, The combined error rate drops to approximately 1 mistake in every billion base pairs copied, or 10 to the power of -9, which is a truly astonishing degree of precision and one that is essential for maintaining the integrity of genetic information across the countless cell divisions that occur over the lifetime of an organism. The quantum mechanical basis of base pair recognition lies at the heart of the first level of fidelity, the initial selection of the correct nucleotide by the polymerase. And this recognition depends critically on the geometry and electronic properties of hydrogen bonds. When adenine pairs with thymine, or when guanine pairs with cytosine, the two bases are held together by hydrogen bonds that form only when the shapes and charge distributions of the molecules are precisely complementary, much like puzzle pieces that fit together in only one correct orientation. These hydrogen bonds are inherently quantum mechanical phenomena, arising from the sharing of proton density between electronegative atoms and involving the delicate interplay of electronic wave functions that we discussed in earlier sections. Thomas Kunkel, who has spent much of his career at the National Institutes of Health studying the mechanisms of polymerase fidelity, has made substantial contributions to our understanding of how these enzymes achieve their remarkable accuracy. His work has shown that polymerases do not simply sit passively and wait for the correct nucleotide to arrive, but rather actively participate in the selection process through a mechanism that is both elegant and instructive. When a nucleotide enters the active site of the polymerase, the enzyme undergoes a conformational change, a shift in its three-dimensional shape that brings certain amino acid residues into close contact with the incoming base and the template base to which it must pair. This conformational change is sensitive to the geometry of the base pair, and if the geometry is incorrect, the conformational change cannot proceed properly, and the nucleotide is rejected rather than incorporated. This process, known as the induced fit mechanism, has important quantum mechanical aspects that are worth considering in some detail. The conformational change that the polymerase undergoes involves the coordinated motion of many atoms, and this motion is governed by the potential energy surface of the protein, which is itself determined by the quantum mechanical interactions among all the atoms involved. The fidelity of the enzyme depends on the fact that the correct and incorrect substrates produce different responses in this complex energy landscape, with the correct substrate leading to a pathway that proceeds smoothly toward catalysis while incorrect substrates encounter energy barriers that slow or prevent their incorporation. Myron Goodman, working at the University of Southern California, has spent many years studying a special class of polymerases known as translesion polymerases, which have the remarkable ability to replicate past damaged sites in DNA where normal polymerases would stall. These translesion polymerases have deliberately lower fidelity than their more accurate counterparts, and this lower fidelity is actually essential for their function, because the damaged templates they must copy often cannot form proper Watson-Crick base pairs with any of the four standard nucleotides. The study of these error-prone polymerases has provided valuable insights into the structural features that determine fidelity, because by understanding what makes a polymerase less accurate, we can better understand the mechanisms that make other polymerases more accurate. Goodman's work has shown that the active sites of high-fidelity and low-fidelity polymerases differ in subtle but important ways, particularly in the tightness with which they grip the incoming nucleotide and the template base. High-fidelity polymerases have active sites that are snugly fitted to the geometry of correct Watson-Crick base pairs, leaving little room for the distortions that would be required to accommodate incorrect pairings. Lower fidelity polymerases have somewhat more spacious active sites that can tolerate a wider range of geometries, which allows them to function on damaged templates but at the cost of reduced accuracy on undamaged DNA. A critical feature of all polymerase active sites is the presence of metal ions, typically magnesium, though in some cases manganese or other divalent cations may be involved. These metal ions play an essential role in the chemistry of nucleotide incorporation, helping to position the incoming nucleotide correctly, activating the attacking hydroxyl group, and stabilizing the negative charges that develop during the transition state of the reaction. The quantum chemical behavior of these metal ions is complex and fascinating, involving the coordination of water molecules and protein side chains in arrangements that are precisely tuned to facilitate catalysis while maintaining fidelity. The magnesium ions in the polymerase active site typically coordinate to the phosphate groups of the incoming nucleotide triphosphate and to specific aspartate residues in the enzyme, creating a rigid geometric framework that positions the reactive groups with great precision. Quantum mechanical calculations have shown that the electronic structure of these metal complexes is exquisitely sensitive to the distances and angles involved, and even small perturbations can significantly affect the rate of the catalytic reaction. This sensitivity helps to explain how polymerases can discriminate against incorrect substrates, because the geometric distortions required to accommodate a mismatched base pair would necessarily perturb the metal coordination, disrupting the optimal electronic arrangement for catalysis. Another factor that contributes to polymerase fidelity is the exclusion of water from the active site during the incorporation reaction. Water molecules are excellent proton donors and acceptors, and their presence in the active site could facilitate unwanted proton transfer reactions that might stabilize incorrect tautomeric forms or otherwise compromise fidelity. High-fidelity polymerases undergo conformational changes upon nucleotide binding that close off the active site from the surrounding solvent, creating a relatively dry microenvironment in which the geometry of hydrogen bonding can be more precisely controlled. This water exclusion has important consequences for the quantum dynamics of protons in the active site, because in the absence of competing water molecules, the proton positions within the hydrogen bonds of the nascent base pair are determined primarily by the electronic properties of the bases themselves. There is considerable evidence that tautomeric forms of the nucleotide bases contribute to polymerase errors, and this connection brings us full circle to the fundamental quantum mechanics that we discussed at the beginning of our journey. Recall that each base can exist in multiple tautomeric forms, with different arrangements of protons along the hydrogen bonding edges, and that these forms interconvert through quantum mechanical proton transfer. If an incoming nucleotide happens to be in a rare tautomeric form at the moment when the polymerase makes its selection, it may be able to pair with the wrong template base and be incorporated incorrectly. Because tautomeric interconversion is a quantum mechanical process involving proton tunneling, the rate at which errors of this type occur depends on fundamentally quantum properties such as barrier heights, tunneling distances, and the vibrational frequencies of the protons involved. Studies using nucleotide analogues that are locked into particular tautomeric configurations have provided direct evidence that tautomeric mismatches can indeed be incorporated by polymerases, and that the error rates observed in these experiments are broadly consistent with the expected populations of rare tautomers under physiological conditions. These findings support the idea that a significant fraction of spontaneous mutations may arise from the quantum mechanical fluctuations in proton positions that occur naturally within nucleotide bases, a conclusion that has profound implications for our understanding of genetic change and evolution. The evolutionary optimization of error rates is a topic that deserves careful consideration, for one might naively assume that the ideal polymerase would be one that makes no errors whatsoever. But this assumption turns out to be incorrect, and there are good reasons to believe that natural selection has fine-tuned polymerase fidelity to a level that is neither too high nor too low, but rather optimally suited to the biological needs of the organism. If error rates were too high, organisms would accumulate deleterious mutations faster than selection could remove them, leading to a phenomenon known as mutational meltdown, in which the genetic quality of the population deteriorates over time. This is clearly disadvantageous and explains why selection has favored mechanisms that reduce error rates to very low levels. But if error rates were too low, organisms might lose the genetic variation that is essential for adaptation and long-term survival. In a changing environment, populations that cannot generate new variants are unable to evolve and may become extinct when conditions shift beyond their tolerance. The optimal error rate therefore represents a balance between the short-term cost of deleterious mutations and the long-term benefit of evolutionary adaptability. And different organisms have settled on different points along this spectrum depending on their population sizes, generation times, and the environmental challenges they face. It is worth noting that the error rate of roughly 1 in a billion base pairs, which we mentioned earlier as the combined outcome of selection, proofreading, and mismatch repair, is not a universal constant but varies among organisms and even among different regions of the genome within a single organism. Some viruses, particularly RNA viruses, have error rates that are many orders of magnitude higher, which allows them to evolve rapidly in response to immune defenses or antiviral drugs, but also means that many of their offspring carry lethal mutations and fail to reproduce. At the other extreme, some archaea living in extreme environments appear to have evolved exceptionally high-fidelity replication systems, perhaps because their harsh habitats leave little room for error. Within complex organisms such as ourselves, there is evidence that error rates may vary across the genome, with some regions being copied more carefully than others. This could arise from differences in chromatin structure, the activity of repair enzymes, or the presence of sequence motifs that are inherently more or less prone to certain types of errors. The full picture of how mutation rates are regulated across the genome remains an active area of research, with new data emerging regularly from large-scale sequencing projects that catalog the precise locations and frequencies of mutations in various cell types and conditions. As we reflect on the extraordinary accuracy of DNA polymerases, achieved through the coordinated action of multiple quantum mechanical mechanisms operating at the molecular scale, it is perhaps appropriate to feel a certain sense of quiet wonder at the sophistication of these ancient molecular machines. They have been refined by billions of years of evolution, tested and improved by the relentless filter of natural selection, and they continue to function with remarkable reliability in every dividing cell of every living organism on Earth. Yet they are not perfect, and their imperfection is, in a very real sense, the engine of evolutionary change, the source of the variation upon which selection acts to produce the endless forms most beautiful and most wonderful that Darwin described at the close of his great work. The proofreading activity of polymerases, which we have touched upon only briefly, deserves a moment of additional consideration before we move on. This activity is carried out by a separate domain of the polymerase enzyme, positioned adjacent to the active site where nucleotides are incorporated, and it functions by recognizing when the growing DNA strand has a mismatched terminal nucleotide. When a mismatch is detected, the strand is shifted from the polymerase active site to the proofreading active site, where the incorrect nucleotide is removed by hydrolysis, and the strand is then returned to the polymerase active site for another attempt at correct incorporation. This back-and-forth motion between incorporation and proofreading occurs on timescales of milliseconds to seconds, and the decision about whether to proofread depends on the kinetic competition between continued polymerization and the rate of strand transfer to the proofreading site. The quantum mechanics of the proofreading reaction itself involves the breaking of the phosphodiester bond that links the incorrect nucleotide to the growing strand, a process that requires the activation of a water molecule to serve as the nucleophile and the stabilization of the leaving group through coordination to metal ions. The details are intricate and have been the subject of extensive computational studies, but the overall picture is one of remarkable molecular choreography, with multiple active sites working in concert to achieve levels of fidelity that would be impossible for any single mechanism acting alone. And so we come to the end of this section on the proofreaders, those vigilant molecular sentinels that stand guard over the integrity of our genetic information. They work quietly and continuously, in every cell of our bodies and in every living thing around us, reducing the error rate of DNA replication to levels so low that they are almost unimaginable by everyday standards. Yet they cannot eliminate errors entirely, and perhaps they should not, for the errors that slip through are the raw material of evolution, the source of the diversity that allows life to adapt and persist through all the vicissitudes of a changing world. In this delicate balance between fidelity and flexibility, between conservation and innovation, we see reflected the same tension that runs through all of biology, the same creative tension that has given rise to every species that has ever lived, including ourselves. The story of how cells came to understand the boundaries of self and other, of what genetic material belongs within and what must be kept without, begins in a rather unexpected place, in a modest laboratory in London during the late 1920s, where a British medical officer named Frederick Griffith was studying the bacterium responsible for pneumonia. Griffith was not searching for the substance of heredity, not seeking to unravel the mysteries of genetic transfer, but rather was engaged in the practical work of understanding how disease-causing bacteria could vary in their virulence, and how the immune system might be trained to recognize them. His experiments involved 2 strains of Streptococcus pneumoniae, one smooth and deadly, surrounded by a protective capsule that allowed it to evade the defenses of the body, and one rough and harmless, lacking this protective coat and therefore easily destroyed by the immune system. The smooth strain killed mice with terrible efficiency, while the rough strain caused no illness at all, and heat killed smooth bacteria. Their proteins denatured and their membranes disrupted, were similarly harmless when injected into experimental animals. What Griffith observed next would quietly revolutionize our understanding of heredity, though the full implications would not become clear for many years. When he mixed heat-killed smooth bacteria with living rough bacteria and injected this combination into mice, the animals sickened and died, and from their tissues he recovered living smooth bacteria, complete with their protective capsules, virulent and deadly. Something from the dead bacteria had transformed the living rough strain, had conveyed to them the instructions for building a capsule they had never possessed, had permanently and heritably altered their nature. Griffith called this the transforming principle, and he puzzled over its identity, suspecting perhaps some protein or some complex of cellular components. But the tools of 1928 were not sufficient to isolate and identify this mysterious substance. The transformation was stable, passed from generation to generation of bacteria, a permanent change in the hereditary material itself, and yet the mechanism remained obscure. a gentle mystery waiting patiently for the technology and insight needed to reveal its nature. 16 years would pass before Oswald Avery, working at the Rockefeller Institute in New York, along with his colleagues Colin MacLeod and Macklin McCarty, would apply the meticulous techniques of biochemistry to the question of the transforming principle's identity. Their approach was one of systematic elimination, of careful purification and enzymatic destruction, testing whether the transforming activity could survive treatment with enzymes that degraded proteins, enzymes that degraded lipids, enzymes that degraded ribonucleic acid. Each of these treatments left the transforming activity intact, the rough bacteria still capable of being converted to smooth, the hereditary information still flowing from dead cells to living ones. Only when they treated their purified extracts with enzymes that specifically degraded deoxyribonucleic acid did the transformation cease. Only then did the rough bacteria remain rough, unchanged by their exposure to the remnants of smooth cells. The transforming principle was DNA, was the long chain of nucleotides that had seemed too simple, too repetitive, too chemically monotonous to carry the complexity of hereditary information. Avery's paper, published in 1944, stated these conclusions with characteristic caution, noting simply that the evidence favored DNA as the carrier of genetic specificity, but the implications were profound, suggesting that cells possessed mechanisms for taking up foreign DNA and incorporating it into their own hereditary material, that the boundaries between organisms were more permeable than anyone had imagined. The phenomenon that Griffith discovered and Avery explained is now understood as one of three major pathways by which bacteria can acquire genetic material from their environment. A process called horizontal gene transfer that operates alongside the vertical transmission of DNA from parent to offspring. Transformation is the simplest of these pathways, involving the direct uptake of naked DNA from the surrounding medium, the absorption of genetic information released by dead or dying cells, or sometimes actively secreted by living neighbors. Transduction represents a second pathway, in which bacterial viruses called bacteriophages accidentally package fragments of host DNA during their reproductive cycle, then inject this bacterial DNA into new host cells during subsequent infections, inadvertently transferring genetic material between cells that may never have been in direct contact. Conjugation is the third pathway, requiring physical connection between donor and recipient cells through a structure called a pilus, through which single strands of DNA are threaded directly from one bacterium to another, a form of bacterial intimacy that can transfer large plasmids or even portions of the main chromosome. Each of these pathways has been shaped by evolution to balance the potential benefits of acquiring new genetic capabilities against the dangers of incorporating harmful or incompatible sequences, and each involves molecular machinery of remarkable sophistication, machinery that must distinguish between desirable and undesirable DNA, between genetic material that will enhance survival and genetic material that threatens cellular integrity. The uptake of foreign DNA across the bacterial cell membrane represents a significant thermodynamic challenge, a process that cannot occur spontaneously but requires the investment of cellular energy and the coordinated action of multiple protein complexes. The cell membrane is a lipid bilayer, a structure whose hydrophobic interior presents a formidable barrier to the passage of large, highly charged molecules like DNA, and the crossing of this barrier requires specialized machinery that can transiently disrupt the membrane's integrity, can create channels or pores through which DNA can thread, can couple the unfavorable energetics of membrane crossing to favorable reactions that drive the process forward. In Streptococcus pneumoniae, the organism that Griffith studied nearly a century ago, the uptake of transforming DNA has been analyzed in exceptional detail by researchers including Donald Morrison, whose careful genetic and biochemical studies have revealed a complex system of proteins that must work in concert to bind DNA at the cell surface, to transport it across the cell wall and membrane, and to protect it from degradation during this vulnerable journey. The bacterium enters a specialized physiological state called competence, a transient condition in which the genes encoding the transformation machinery are expressed, in which the cell becomes receptive to foreign DNA, and this competent state is itself carefully regulated, induced only under specific environmental conditions, only when the potential benefits of acquiring new genetic information outweigh the costs and risks of opening the cellular boundary to external material. The quantum mechanical barriers that foreign DNA must overcome during transformation are subtle but significant, involving the precise energetics of protein-DNA interactions, the delicate balance of hydrogen bonds and electrostatic forces that must be formed and broken as DNA moves through the uptake machinery, the conformational changes in both protein and nucleic acid that must occur with exquisite timing and coordination. Each step in the transformation process involves a transition between states of different energy, and while the energy differences may be small, on the order of a few kilocalories per mole, the accumulated effect of many such transitions determines whether uptake will proceed efficiently or stall at some intermediate stage. The proteins that mediate DNA uptake must bind their substrates with sufficient affinity to capture passing molecules, but not so tightly that release becomes impossible, must thread double-stranded DNA through narrow channels where it is often converted to single strands, must protect the DNA from the nucleuses that would otherwise degrade foreign sequences as a defense against invasion. The quantum tunneling of protons may play a role in some of these processes, particularly in the enzymatic reactions that process DNA during uptake, and the precise geometries required for optimal hydrogen bonding between protein and nucleic acid reflect the quantum mechanical nature of these interactions at the atomic scale. Once foreign DNA has entered a bacterial cell, it must still evade the immune systems that have evolved to protect the cell against precisely this kind of invasion. Systems that recognize and destroy DNA from viruses and other sources that would exploit the cellular machinery for their own replication. The most celebrated of these immune systems, and surely the most thoroughly studied in recent years, is the CRISPR-Cas system, whose acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats, a name that reflects the unusual structure of the genomic loci that encode this remarkable defense. The fundamental insight that CRISPR functions as a bacterial immune system emerged gradually during the first decade of the 21st century, as researchers noticed that the short sequences between the repeats often matched sequences from bacteriophages and other mobile genetic elements, suggesting that bacteria were somehow recording the identities of past invaders in their own chromosomes, creating a genetic memory of infection that could guide future defenses. The breakthrough that transformed this observation into a powerful technology came from the work of Jennifer Doudna and Emmanuel Charpentier, whose collaboration beginning around 2011 revealed how the CRISPR-Cas9 system from Streptococcus pyogenes could be reprogrammed to target essentially any DNA sequence, could be directed by a simple guide RNA to find and cut specific sites in a genome, could be adapted as a tool for precise genetic engineering in organisms from bacteria to humans. For this work, Doudna and Charpentier received the Nobel Prize in Chemistry in 2020, recognition of both the fundamental biological insight and the transformative practical applications of their discovery. The mechanism by which CRISPR-Cas recognizes foreign DNA exemplifies the quantum precision of biological recognition, the extraordinary selectivity achieved through the simple chemistry of base pairing between complementary nucleic acid strands. The Cas9 protein, the molecular scissors at the heart of the type II CRISPR system, carries with it a small RNA molecule called the guide RNA, whose sequence is complementary to the target DNA that must be cut, and recognition occurs through Watson-Crick base pairing between this guide and the potential target, through the same hydrogen bonding interactions that hold the two strands of the double helix together. But perfect complementarity is not sufficient for cleavage, for the system must also distinguish between foreign DNA that should be destroyed and the CRISPR locus itself, which contains matching sequences that must not be cut, and this discrimination is achieved through a short sequence called the protospacer adjacent motif, or PAM, that must be present next to the target for cleavage to occur, a sequence that is absent from the CRISPR array itself. The energetics of guide RNA binding to target DNA involve the progressive formation of hydrogen bonds as the guide anneals to its complement, the gradual displacement of the non-target DNA strand as the R-loop structure forms, the conformational changes in the protein that couple target recognition to activation of the nucleus domains that will cleave both strands of the DNA. Each base pair contributes only a few kilocalories per mole of binding energy, but the accumulated effect of 12 to 20 matching base pairs creates a highly specific interaction, one that can distinguish a perfect match from sequences differing by even a single nucleotide, at least under optimal conditions. The discrimination between self and non-self in bacteria extends beyond CRISPR to include systems based on DNA methylation, the covalent attachment of methyl groups to specific bases that marks DNA as belonging to the cell rather than to an invader. Restriction modification systems, widespread among bacteria, encode both a restriction enzyme that cuts DNA at specific recognition sequences and a methyltransferase that modifies those same sequences in the host chromosome, protecting them from cleavage. Foreign DNA entering the cell lacks these protective methylation marks and is therefore subject to rapid degradation by the restriction enzyme, a simple but effective defense that can eliminate most invading DNA before it has a chance to replicate or integrate. The quantum mechanical details of how restriction enzymes distinguish between methylated and unmethylated substrates involve the precise geometry of the active site, the way in which the methyl group, a small addition of 1 carbon and 3 hydrogen atoms, nonetheless disrupts the network of hydrogen bonds and van der Waals contacts that position the DNA for cleavage, the way in which this tiny chemical change translates into a profound difference in enzymatic activity. The methyltransferases themselves catalyze a remarkable reaction, transferring a methyl group from the cofactor S-adenosyl methionine to a specific position on an adenine or cytosine base, a reaction that involves the transient formation of a covalent bond between enzyme and DNA, the quantum tunneling of the methyl group to its new position, the precise alignment of molecular orbitals that permits this otherwise improbable chemistry to proceed. When foreign DNA has survived the gauntlet of nucleuses and immune systems, has been transported across the membrane and into the cytoplasm intact, it must still be integrated into the host chromosome to achieve permanent transformation, to become part of the cell's hereditary endowment that will be passed to future generations. This integration typically occurs through homologous recombination, a process that exchanges segments of DNA between molecules sharing similar or identical sequences, that allows incoming DNA to replace or supplement corresponding regions of the host chromosome. The central player in bacterial homologous recombination is a protein called REC-A, which forms long helical filaments on single-stranded DNA and then searches through double-stranded DNA molecules for sequences complementary to the bound strand, catalyzing strand invasion when a match is found, initiating the complex series of molecular rearrangements that will ultimately result in genetic exchange. The formation of the REC-A filament is itself a cooperative process, with each REC-A monomer binding 6 nucleotides of single-stranded DNA, with the binding of each monomer facilitating the binding of the next, with the growing filament stretching the DNA to roughly 1.5 times its normal length, a conformation that appears to facilitate the search for homology. The strand invasion catalyzed by REC-A requires a quantum precision of base pairing that is truly remarkable, for the filament must distinguish between sequences that match perfectly and sequences that differ by even 1 or 2 nucleotides, must sample millions of potential pairing partners in seconds, must stabilize productive interactions while rejecting those that would lead to inappropriate recombination. The thermodynamics of this process have been studied extensively, revealing that REC-A does not simply accelerate the spontaneous hybridization of complementary strands, but actively promotes pairing between sequences that would not readily anneal on their own. That couples the energy of ATP binding and hydrolysis to the work of strand exchange. The transition from the initial sampling of homology, a fleeting and unstable interaction, to the formation of a stable joint molecule, committed to completing the recombination reaction, involves a series of conformational changes in the REC-A filament, changes that are contingent on the presence of sufficient sequence complementarity, that ensure productive recombination occurs only when the incoming and resident sequences are adequately matched. The quantum mechanical underpinnings of this fidelity include the precise energetics of base stacking and hydrogen bonding in the 3-stranded intermediate, the way in which mismatches destabilize this structure and trigger rejection of unsuitable partners, the remarkable sensitivity of the system to even small imperfections in sequence alignment, the boundaries that cells enforce around their genetic material, the mechanisms by which they decide what DNA to accept and what to reject, reflect billions of years of evolutionary refinement, of balancing the need for genetic stability against the benefits of acquiring new capabilities, of protecting against viral invasion while remaining open to potentially useful horizontal transfer. Each component of these systems, from the competence machinery that takes up transforming DNA to the CRISPR arrays that remember past infections, from the restriction enzymes that degrade foreign sequences to the REC-A filaments that integrate acceptable DNA into the chromosome, operates through the quantum mechanical principles that govern all molecular interactions, through the delicate balance of hydrogen bonds and electrostatic forces, through the precise geometries of protein-DNA contacts, through the thermodynamic barriers that must be overcome for each step to proceed. As you rest now, consider how every cell maintains these boundaries with tireless vigilance, how the decision to accept or reject each fragment of foreign DNA is made through molecular machinery of extraordinary sophistication, machinery that has been refined over evolutionary timescales to achieve a balance that serves the interests of cellular survival. The work of Griffith and Avery opened our eyes to the permeability of genetic boundaries, to the flow of hereditary information between organisms, and the subsequent decades have revealed the intricate mechanisms that control this flow, that permit beneficial exchange while limiting harmful intrusion, that operate at the quantum mechanical boundary between chemistry and biology, where the fundamental laws of physics find expression in the ancient art of cellular self-preservation. And so we come now to a quieter layer of inheritance, one that sits upon the sequence itself like morning dew upon a blade of grass, present and influential yet somehow separate from the structure beneath. This is the realm of epigenetics, where the letters of DNA remain unchanged and yet their meaning shifts, where genes may be silenced or awakened without any alteration to their underlying code. And at the heart of this gentle modulation lies a small chemical modification, the addition of a single methyl group, 3 hydrogens bonded to a carbon, attached to one of the 4 nucleotide bases. It is a tiny change, almost imperceptible in the grand architecture of the double helix, and yet it carries profound consequences for which genes are read and which remain dormant. And as we shall see, the transfer of this small chemical group relies upon the same quantum mechanical principles we have been exploring throughout our time together. The discovery of this modification predates our understanding of the double helix itself, which gives some sense of how slowly and carefully the story of DNA has been assembled from separate threads of observation and experiment. In 1948, a biochemist named Rollin Hotchkiss was working at the Rockefeller Institute, using paper chromatography to separate the components of nucleic acids extracted from calf thymus tissue. He noticed something unexpected in his separations, a 5th spot on his chromatography paper where there should have been only 4, corresponding to a modified form of cytosine that he identified as 5-methylcytosine. The methyl group was attached to the 5th carbon of the cytosine ring, jutting out into the major groove of the helix like a small molecular flag. Hotchkiss published his findings and noted the presence of this modified base, though at the time its function remained entirely mysterious, a chemical curiosity awaiting explanation. It would take several more decades before researchers began to understand that this methylation of cytosine was not random chemical noise, but rather a carefully controlled system for regulating gene expression, a layer of information written above the sequence itself. The modification occurs predominantly at cytosines that are followed by guanine in the sequence, forming what we call CpG dinucleotides, where the P represents the phosphate linkage between the 2 bases. This specificity is quite remarkable and emerges from the detailed architecture of the enzymes responsible for adding methyl groups to DNA, enzymes known as DNA methyltransferases or DNMTs. Understanding how these enzymes achieve their specificity and catalyze the transfer of methyl groups requires us to examine the quantum mechanical basis of their reaction mechanism. The donor of methyl groups in this process is a molecule called S-adenosylmethionine, commonly abbreviated as SAM, which serves as a universal methyl donor throughout biology, participating in hundreds of different methylation reactions in cells. SAM carries a methyl group attached to a sulfur atom in what chemists call a sulfonium center, and this arrangement makes the methyl group particularly reactive, ready to be transferred to suitable acceptor molecules. When a DNA methyltransferase encounters an unmethylated cytosine in the appropriate sequence context, it brings SAM into close proximity with the target base and facilitates the transfer of the methyl group from the sulfur of SAM to the 5th carbon of cytosine. The mechanism of this transfer involves several steps, each requiring the precise quantum mechanical behavior of electrons and protons. First, the enzyme flips the target cytosine out of the DNA helix, rotating it 180 degrees so that it sits in a special pocket within the enzyme where the reaction can occur. This base flipping is itself a remarkable feat of molecular recognition. and the enzyme must somehow identify the correct base in the correct sequence context before committing to this dramatic conformational change. Once the cytosine is positioned in the active site, a cysteine residue in the enzyme donates electrons to the 6th carbon of the cytosine ring through a process called nucleophilic attack. This creates what chemists call a covalent intermediate, where the enzyme is temporarily bonded to the DNA base, and this intermediate activates the 5th carbon, making it receptive to the incoming methyl group. The transfer of the methyl group from SAM to the activated cytosine involves the movement of electrons across chemical bonds, and as with all chemical bond formation, this process is fundamentally quantum mechanical in nature. The electrons do not simply jump from one location to another, but rather exist in superposition states during the transition, with probability amplitudes that determine the likelihood of successful transfer. The transition state of this reaction, the fleeting configuration at the top of the energy barrier between reactants and products, involves partial bonds both to the sulfur of SAM and to the carbon of cytosine, with the methyl group suspended between its origin and its destination. Research by Xiaodong Cheng and colleagues at Emory University has illuminated the detailed structure of DNA methyltransferases, particularly the DNMT3 family of enzymes that establish new methylation patterns during development. Using X-ray crystallography, Cheng's group has captured snapshots of these enzymes in complex with DNA and SAM analogues, revealing the precise architecture of the active site and the positions of key amino acid residues involved in catalysis. Their work shows how the enzyme cradles the flipped cytosine in a hydrophobic pocket, positioning it with angstrom-level precision relative to the methyl group of SAM. The distance between the sulfur of SAM and the target carbon of cytosine in these structures is approximately 3 angstroms, close enough for orbital overlap but separated by what would classically be an insurmountable energy barrier. It is in overcoming this barrier that quantum mechanical effects become particularly relevant. Computational studies of SAM-dependent methyltransferases suggest that the transfer of the methyl group may involve quantum tunneling, where the probability wave of the transferring atoms extends through the energy barrier rather than over it. This tunneling contribution would be most significant for the proton transfers that occur during the reaction, as protons are light enough for their quantum wavelengths to be substantial relative to the barrier width. The reaction mechanism involves at least 2 proton transfers in addition to the methyl transfer, one to abstract a proton from the nitrogen of cytosine and another to remove the proton that was added during the nucleophilic attack. Each of these proton movements could potentially involve tunneling through energy barriers that would otherwise slow the reaction considerably. The evidence for quantum tunneling in enzymatic methylation reactions comes from kinetic isotope effect studies, where researchers replace hydrogen atoms with deuterium, a heavier isotope of hydrogen, and observe changes in reaction rates. If proton transfer occurred purely by classical thermal activation, replacing hydrogen with deuterium would slow the reaction by a predictable factor related to the mass difference. But if tunneling is involved, the isotope effect can be larger than classical predictions, because the heavier deuterium nucleus has a shorter quantum wavelength and tunnels less efficiently. Several SAM-dependent methyltransferases show kinetic isotope effects consistent with tunneling contributions, suggesting that quantum mechanics plays a functional role in their catalytic mechanism. This quantum biochemistry operates during some of the most dramatic moments in development, including the epigenetic reprogramming that occurs in early embryos. Shortly after fertilization, the methylation patterns inherited from both parents are largely erased, wiping clean the epigenetic slate so that the new organism can establish its own patterns appropriate for different cell types and tissues. This global demethylation involves the removal of methyl groups from millions of cytosines across the genome, and it must occur with sufficient accuracy to erase parental marks while preserving certain regions that need to maintain their methylated state. The quantum mechanical fidelity of the enzymes involved in both adding and removing methyl groups is therefore crucial for proper development. The pathway of demethylation was clarified by the discovery in 2009 of a modified base called 5-hydroxymethylcytosine, where a hydroxyl group is added to the methyl group of methylated cytosine. This discovery, made independently by two research groups led by Anjana Rao and Nathaniel Heinz, revealed that demethylation does not occur in a single step but rather through a series of oxidation reactions that progressively modify the methyl group until it can be removed. The enzymes responsible for these oxidations belong to the TET family, named for the 1011 translocation that first brought them to attention in cancer research. TET enzymes are iron and alpha-ketoglutarate-dependent dioxygenases, which is to say they use iron atoms and a small organic molecule called alpha-ketoglutarate to activate molecular oxygen for insertion into substrate molecules. The active site of TET enzymes contains an iron atom coordinated by two histidine residues and one aspartate residue, forming what is called a facial triad that is common to this family of enzymes. During catalysis, the iron cycles between different oxidation states, generating a highly reactive iron oxo intermediate that can abstract hydrogen atoms from otherwise stable carbon-hydrogen bonds. The iron oxo intermediate at the heart of TET catalysis is one of the most reactive species in biology, capable of breaking bonds that would be stable under ordinary conditions. Its formation involves the transfer of electrons from iron to oxygen through quantum mechanical processes that depend on the overlap of their electron orbitals, and its reactivity derives from the unpaired electrons that create what chemists call a high-spin state. When this iron oxo species encounters the methyl group of 5-methylcytosine, it abstracts a hydrogen atom through a process that likely involves quantum tunneling, given the light mass of the hydrogen and the narrow width of the energy barrier for abstraction. The resulting carbon radical then captures a hydroxyl group, yielding 5-hydroxymethylcytosine. TET enzymes can continue oxidizing this product, first to 5-formylcytosine and then to 5-carboxylcytosine, each step involving the same iron oxo chemistry. These further oxidized bases can then be removed by the base excision repair pathway, the same system we discussed earlier for removing damaged bases, ultimately replacing the modified cytosine with an unmodified one. Thus the erasure of epigenetic marks relies upon both oxidation chemistry catalyzed by TET enzymes and the repair mechanisms that maintain genomic integrity, all operating through quantum mechanical processes at the level of electron and proton transfer. The connection between DNA methylation and cancer has been recognized for several decades, with aberrant methylation patterns being a hallmark of tumor cells. In many cancers, the promoter regions of tumor suppressor genes become hypermethylated, silencing genes that would otherwise restrain cell proliferation. This epigenetic silencing can be as effective as mutation in disabling protective genes, and it occurs without any change to the underlying DNA sequence. The quantum mechanical fidelity of DNA methyltransferases is thus relevant to cancer biology, because errors in methylation pattern maintenance could lead to inappropriate silencing or activation of genes. Some researchers have proposed that oxidative stress in tumor microenvironments might perturb the delicate quantum mechanical processes in DNMT and TET enzymes, contributing to the epigenetic chaos observed in cancer cells. The inheritance of methylation patterns from one cell generation to the next requires a remarkable feat of quantum mechanical copying. When DNA replicates, the two daughter duplexes each contain one methylated strand from the parent and one unmethylated strand that is newly synthesized. A specialized methyltransferase called DNMT1 recognizes these hemimethylated sites and adds methyl groups to the new strand, copying the pattern from the template. This process must be extremely accurate, because errors in methylation inheritance would cause epigenetic drift over many cell divisions, potentially altering gene expression patterns in ways that could be harmful. The quantum mechanical precision required for this copying is in some ways analogous to the fidelity requirements for DNA sequence replication, though operating through a different set of enzymes and mechanisms. The study of quantum effects in DNA methylation remains an active area of research, with many questions still unanswered about the extent to which tunneling and other quantum phenomena contribute to the efficiency and accuracy of these reactions. What is clear is that the chemistry of epigenetic modification, like all chemistry, is fundamentally quantum mechanical, and that understanding these modifications fully requires engaging with the quantum description of electrons, protons, and the bonds they form. The methyl groups that silence genes and the oxidation reactions that awaken them again are all consequences of electron probability distributions and the tunneling of light particles through energy barriers. And so we see that the inheritance of biological information extends beyond the sequence of bases in DNA to include these chemical modifications that sit upon the sequence, modulating its expression without altering its content. The quantum mechanics of methylation and demethylation form a second layer of inheritance, one that allows organisms to respond to their environment and regulate their development through mechanisms that complement the digital code of the genome. The small methyl group, just 4 atoms in total, carries with it the full quantum mechanical complexity of chemical bonding, its transfer governed by the same principles that determine the behavior of electrons in atoms and the tunneling of particles through barriers. It is a gentle reminder that even the most subtle biological processes rest upon the foundations of physics, and that the inheritance of traits across generations involves not just the copying of sequences, but the maintenance of chemical modifications through quantum mechanical reactions of exquisite precision and reliability. And now we turn our attention to something rather beautiful in its quietness. The way that scientists have learned to watch individual molecules as they go about their work, one at a time, in the soft glow of carefully focused light. This is the story of how we came to observe the very smallest dancers in the molecular world, and how these observations revealed that the processes governing your genetic material are not the smooth, continuous motions one might imagine, but rather a series of discrete quantum events, each one separate and particular, like raindrops falling one by one into a still pond. For many decades, the study of biological molecules was necessarily a study of crowds. When a biochemist wished to understand how an enzyme worked, or how DNA replicated itself, they would gather together billions upon billions of molecules in a test tube and measure the average behavior of the entire population. This was rather like trying to understand how a single person dances by observing a stadium filled with thousands of dancers and measuring only the collective sway of the crowd. You might learn something about the general rhythm, certainly, but the individual flourishes, the momentary pauses, the particular way one dancer might hesitate before a turn, all of this would be lost in the averaging. And it is precisely in these individual moments, these quantum mechanical decisions made by single molecules, that the true nature of DNA dynamics reveals itself. The ability to watch single molecules emerged from a convergence of technologies, each one developed with patient care over many years. Among the most important of these was the optical trap, sometimes called optical tweezers, a device that uses light itself to hold and manipulate objects far too small to touch with any physical instrument. The theoretical possibility of using light to exert force on matter had been understood since the time of Maxwell, who showed that electromagnetic radiation carries momentum. But the practical realization of this principle for manipulating microscopic objects came through the careful work of Arthur Ashkin, a physicist working at Bell Laboratories in New Jersey, who spent many quiet years refining the technique until it became a tool of extraordinary precision. Ashkin began his investigations in the 1970s, and he discovered that a tightly focused laser beam could create a gentle well of force, a kind of optical valley into which small particles would naturally settle and remain. The physics of this trapping involves the way that light bends when it passes through an object with a different refractive index than its surroundings. And because light carries momentum, this bending creates a force that pushes the particle toward the region of highest light intensity at the beam's focus. The forces involved are almost unimaginably small, measured in piconewtons, which are millionths of the force you might use to press a key on a keyboard. And yet these piconewton forces are precisely the scale at which individual molecular bonds operate, the scale at which a single hydrogen bond forms or breaks, the scale at which the quantum mechanical interactions within DNA take place. Ashkin was awarded the Nobel Prize in Physics in 2018 for this work, more than 4 decades after his initial experiments, a recognition of how fundamentally his patient investigations had changed our ability to observe the molecular world. Around the same time that optical tweezers were being developed, another technique was emerging that would prove equally important for observing individual molecules: atomic force microscopy, which uses a tiny physical probe to feel the contours of surfaces at the atomic scale, was invented in 1986 by Gerd Binnig, Calvin Quate, and Christoph Gerber. This microscope works by dragging an extremely sharp tip, often just a few atoms wide at its point, across a surface while measuring the tiny forces that push and pull on the tip as it encounters individual atoms and molecules. The forces measured by atomic force microscopy are again in the piconewton range, and the spatial resolution can reach fractions of a nanometer, allowing researchers to see the helical grooves of individual DNA molecules and even, in some cases, to distinguish one base pair from another. The development of these techniques meant that for the first time, scientists could reach down into the molecular world and not merely observe, but touch, hold, and gently manipulate individual strands of DNA. Among the pioneers who applied these new tools to understanding DNA was Stephen Bloch, a biophysicist at Stanford University who began his career studying the motors that move cargo within cells and then turned his attention to the molecular machines that copy and transcribe genetic information. Bloch and his colleagues developed methods to attach single DNA molecules to tiny beads that could be held in optical traps, allowing them to stretch the DNA like a very fine thread and measure its mechanical properties with exquisite precision. They could feel the force required to extend the double helix, the way it stiffened and relaxed under different conditions, the sudden changes in length when proteins bound to it or when the helix unwound. These experiments revealed that DNA is not a passive storage medium but a mechanically active molecule whose physical properties are intimately connected to its function. The forces that block measured, typically in the range of a few piconewtons, correspond to the energy scales of individual hydrogen bonds and base stacking interactions, the quantum mechanical forces that hold the double helix together and allow it to be read, copied, and repaired. The measurement of piconewton forces deserves a moment of contemplation, for it represents a remarkable achievement in the detection of the very small. A piconewton is 1/1,000,000th of a newton, and to place this in some perspective, the force of gravity on a single bacterium is roughly 1 piconewton, and the force required to break a single hydrogen bond is also in this range. The optical traps and atomic force microscopes used in single-molecule studies can measure forces with a precision of fractions of a piconewton, which means they can detect the forming and breaking of individual molecular bonds as they happen. This sensitivity brings us directly into the quantum mechanical realm, where the discrete nature of energy and force manifests itself in the behavior of individual molecules. At the University of California, Berkeley, Carlos Bustamante built upon these foundations to study the molecular machines that replicate and transcribe DNA at the single-molecule level. Bustamante and his group developed techniques to watch individual DNA polymerase enzymes as they copied genetic information, one nucleotide at a time, and to measure the forces these enzymes generated as they worked. They discovered that the process of copying DNA is not smooth and continuous but proceeds in discrete steps, with each nucleotide added in a distinct event that involves the binding of the incoming nucleotide, a conformational change in the enzyme, and the formation of a new chemical bond. The timing of these steps is stochastic, meaning that each individual event occurs with a certain probability per unit time rather than on a fixed schedule, and this stochastic nature reflects the quantum mechanical reality of molecular processes, where thermal fluctuations and quantum uncertainties determine when and how each reaction occurs. Another technique that proved essential for observing quantum events in DNA is Foster resonance energy transfer, commonly known as FRET, which allows researchers to measure distances between specific points on a molecule with a precision of a few angstroms, which is to say a few tenths of a nanometer. The technique is named for Theodor Foster, a German physical chemist who worked out the theory of this phenomenon in the 1940s, and it works by attaching two different fluorescent molecules, called a donor and an acceptor, to the points whose distance you wish to measure. When the donor is excited by light, it can transfer its energy to the acceptor without emitting a photon, but only if the acceptor is close enough, and the efficiency of this transfer depends very sensitively on the distance between the two molecules. By measuring how much energy is transferred, researchers can determine the distance between the donor and acceptor, and by watching how this transfer changes over time, they can observe the molecule's conformational dynamics as they happen. The quantum mechanical basis of FRET lies in the phenomenon of dipole-dipole coupling, which describes how two oscillating electric dipoles can exchange energy when they are close together. The excited donor molecule acts as an oscillating dipole, and if the acceptor molecule is nearby and has the right electronic properties, it can absorb this oscillating electromagnetic field and become excited itself. The efficiency of this coupling falls off very steeply with distance, following an inverse sixth power law, which is what gives FRET its remarkable sensitivity to small changes in molecular separation. The distances over which FRET operates efficiently, typically between 1 and 10 nanometers, are precisely the length scales relevant to conformational changes in DNA and proteins. making this a powerful tool for observing the quantum mechanical dance of biological molecules. Tekjip Ha, working first at Stanford and later at Johns Hopkins University, became one of the masters of single-molecule FRET and applied it with great effect to understanding the conformational dynamics of DNA and the proteins that interact with it. Ha and his colleagues developed methods to observe individual DNA molecules as they bent, twisted, and changed shape in response to the binding of proteins, and they discovered that these conformational changes often occur in discrete steps rather than as smooth, continuous motions. The time resolution of these experiments reached into the sub-millisecond range, allowing the observation of events that occur on timescales of hundreds of microseconds, and this revealed a world of molecular motion that had been invisible to earlier techniques. The DNA molecule was found to be constantly fluctuating, sampling different conformations as thermal energy jostled its atoms, and the proteins that read and repair DNA were found to exploit these fluctuations, waiting for the right conformation to appear before binding and carrying out their functions. One of the most remarkable observations made possible by single-molecule techniques was the direct visualization of base flipping, the process by which repair enzymes flip individual nucleotides out of the DNA helix to examine them for damage. This process had been predicted from structural studies, but watching it happen in real time required the sensitivity and time resolution of single-molecule FRET. Researchers observed that when a repair enzyme encounters a damaged base, it does not immediately recognize the damage while the base remains stacked in the helix, but rather it induces the base to flip out, breaking the hydrogen bonds that hold it in place and rotating it through the major groove until it sits in a pocket on the enzyme surface where it can be inspected. The flipping process was found to be dynamic and reversible, with bases flipping in and out multiple times before the enzyme makes its decision about whether to initiate repair. This stochastic sampling process reflects the quantum mechanical nature of molecular recognition, where thermal fluctuations allow molecules to explore different configurations until they find one that is energetically favorable. Antoine van Oijen, a Dutch biophysicist who has worked in the Netherlands and Australia, developed techniques to observe DNA replication at the single-molecule level with unprecedented detail. Van Oijen and his colleagues created what might be called molecular movies of individual replication forks, the Y-shaped structures where the two strands of the double helix separate and are copied by DNA polymerase enzymes. They discovered that the replication process is remarkably uneven, with the enzyme sometimes pausing for extended periods and other times racing ahead at hundreds of nucleotides per second. This variability is not a sign of disorder but rather reflects the stochastic nature of the chemical reactions involved, where each nucleotide addition is a separate event with its own quantum mechanical probability. The timing of these events follows statistical distributions that can be understood in terms of the energy barriers that must be overcome and the thermal fluctuations that provide the energy to cross them. These single-molecule studies have collectively revealed that the processes governing DNA are not the deterministic, clockwork mechanisms that one might imagine from looking at textbook diagrams, but rather they are stochastic quantum events, each one subject to the uncertainties and fluctuations inherent in the molecular world. The polymerase enzyme does not copy DNA at a constant rate but rather adds each nucleotide with a certain probability per unit time, and this probability depends on quantum mechanical factors such as the energy required to break and form hydrogen bonds, the tunneling of protons between tautomeric states, and the thermal fluctuations that allow molecules to sample different configurations. The repair enzymes do not scan DNA in a smooth, continuous motion but rather hop and slide in discrete steps, pausing to examine suspicious bases by flipping them out of the helix in a process governed by the same stochastic physics. What emerges from these observations is a picture of DNA as a dynamic, fluctuating molecule whose behavior is fundamentally quantum mechanical in nature. The forces that hold the double helix together, the hydrogen bonds between base pairs and the stacking interactions between adjacent bases, are quantum mechanical forces arising from the electronic structure of the atoms involved. The transitions between different states, such as the flipping of a base or the addition of a nucleotide, are governed by energy barriers that are crossed through thermal fluctuations and, in some cases, through quantum tunneling. The timing of these events is stochastic, following probability distributions rather than fixed schedules. And this stochasticity is not noise or error but rather a fundamental feature of how molecules behave at the quantum scale. The development of single-molecule techniques thus represents not merely a technical advance but a conceptual shift in how we understand the molecular basis of life. By watching molecules one at a time, scientists have been able to see the discrete, quantum nature of biological processes that was previously hidden in the averaging of bulk measurements. Each hydrogen bond that forms or breaks, each nucleotide that is added to a growing DNA strand, each damaged base that is flipped out for inspection, is a separate quantum event, occurring with its own probability and timing. And yet from these myriad individual events, carried out by billions of molecules in every cell, emerges the reliable transmission of genetic information from generation to generation, a remarkable consistency arising from the statistical averaging of quantum uncertainties. As you rest now and let these ideas settle into quietness, you might imagine the molecules within your own cells going about their work in this gentle, stochastic way, not rushing or forcing anything but simply allowing each quantum event to occur when the time is right, when the thermal fluctuations align and the energy barriers are crossed. There is something peaceful in this picture, a kind of molecular patience that has no need for urgency or drama but simply continues, moment by moment, as it has continued for billions of years since the first DNA molecules began their quiet dance in the ancient seas of Earth. The techniques that allow us to watch this dance, the optical tweezers and atomic force microscopes and FRET experiments, are themselves testimonies to human patience and careful work, developed over decades by researchers who wanted nothing more than to see what had never been seen before, to observe the quantum mechanical heart of life itself. And so we come now to a quieter corner of this story, one that unfolds not in the wet spaces of laboratory benches or the hushed reading rooms of crystallography facilities, but rather in the humming warmth of computer servers and the patient accumulation of calculations that can take days or even weeks to complete. This is the realm of computational quantum chemistry, where researchers have learned to simulate the behavior of electrons and nuclei with such precision that they can predict, from first principles, how molecules will behave without ever needing to synthesize them or observe them directly. It is a remarkable development in the history of science, this ability to calculate the properties of matter using nothing more than the fundamental equations of quantum mechanics, and when applied to the molecules of life, to the base pairs of DNA and their delicate propensity for change, these methods have opened windows onto processes that would otherwise remain entirely invisible to experimental observation. The story of how we learned to calculate the quantum properties of molecules is itself a long and winding one, stretching back to the earliest days of quantum mechanics when scientists first began to grapple with the mathematical complexity that arises whenever more than two particles interact with one another. The hydrogen atom, with its single electron orbiting a single proton, can be solved exactly using Schrödinger's equation, its energy levels and wave functions written down in closed mathematical form. But the moment you add a second electron, as in the helium atom, the problem becomes intractable to exact solution, because each electron not only feels the attraction of the nucleus but also the repulsion of the other electron. And this repulsion depends on where the other electron happens to be at any given moment, which in turn depends on where the first electron is, creating a tangled web of interdependencies that cannot be unraveled through simple algebra. The first major breakthrough in dealing with this complexity came in the 1920s and 30s, when Douglas Hartree, a physicist working at Cambridge, developed an approach that would bear his name for decades to come. Hartree's insight was to treat each electron as though it were moving in an average field created by all the other electrons, rather than trying to track the instantaneous position of every particle simultaneously. This self-consistent field approach, as it came to be known, allowed calculations to proceed iteratively, with each electron's wave function being refined based on the average behavior of all the others, and then that refinement being fed back into the calculation, over and over, until the solution stopped changing and a stable answer emerged. Vladimir Fock, a Soviet physicist working in Leningrad, later refined this approach to properly account for the quantum mechanical requirement that electrons be indistinguishable from one another, leading to what we now call the Hartree-Fock method, which remained the workhorse of quantum chemistry for many decades and which, in various modified forms, continues to find application even today. The Hartree-Fock method was a triumph of practical approximation, allowing chemists to calculate molecular geometries, bond energies, and electronic properties with reasonable accuracy for small to medium-sized molecules. But it had limitations, the most significant being that it treats electron correlation only in an average way, missing the subtle dance by which electrons avoid one another instant by instant rather than merely on average. For many chemical problems, and particularly for the weak interactions that govern so much of biochemistry, this missing electron correlation proved to be critically important, and researchers spent decades developing increasingly sophisticated methods to capture it. Methods with names like configuration interaction and coupled cluster theory that promise greater accuracy at the cost of dramatically increased computational expense. It was in this context that Walter Kohn, an Austrian-born physicist who had fled the Nazis as a child and eventually settled in California, developed an entirely different approach to the quantum mechanical problem, one that would earn him the Nobel Prize in Chemistry in 1998 and would transform the landscape of computational chemistry in ways that continue to reverberate today. Kohn's insight, developed together with Liu Ju Sham in the 1960s, was that instead of trying to calculate the full many-electron wave function with all its exponentially growing complexity, one could instead work with the electron density, a much simpler quantity that describes merely how much electronic charge exists at each point in space, without worrying about which specific electrons are contributing to that charge or how they correlate with one another. This approach, known as density functional theory, or DFT, rested on a remarkable theorem that Kohn had proven with Pierre Hohenberg in 1964, demonstrating that the electron density alone contains all the information needed to determine every property of the ground state of a quantum mechanical system. If you know the density, in principle you know everything. And Kohn and Sham showed how to set up equations that would allow the density to be calculated in a practical way, with computational costs that scaled far more gently with system size than traditional wave function methods. The catch, and there is always a catch in computational quantum chemistry, was that the exact form of the crucial exchange correlation functional, The mathematical object that encapsulates how electrons interact with one another, remained unknown and had to be approximated, leading to a proliferation of different functional forms with acronyms like B3LYP and PBE that populate the literature to this day. Despite the inherent approximations, density functional theory proved remarkably successful at predicting molecular structures and energetics, accurate enough for many practical purposes and efficient enough to be applied to systems containing hundreds or even thousands of atoms, sizes that would be utterly unthinkable with more rigorous but more expensive methods. It was this combination of reasonable accuracy and manageable computational cost that made DFT the method of choice for studying biological molecules, including, crucially, the base pairs of DNA and their tautomeric variants. Among the pioneers who applied these computational methods to DNA was Jerzy Leszczynski, a Polish-born chemist who established a remarkably productive research program at Jackson State University in Mississippi, turning that historically black university into an unexpected center of excellence for computational chemistry. Leszczynski and his collaborators used density functional theory and related methods to systematically study the tautomerization of DNA bases, calculating with careful precision the energy differences between canonical forms and their rare variants. These calculations confirmed what experimentalists had long suspected but could never directly measure, that the rare tautomers of adenine, guanine, cytosine, and thymine lie roughly 10 to 15 kilocalories per mole above their canonical counterparts, an energy difference that corresponds, through the Boltzmann distribution, to roughly one rare tautomer for every 10,000 to 100,000 canonical forms at body temperature. To put these numbers in perspective, 10 kilocalories per mole is not a large energy in absolute terms, perhaps a tenth of the energy released when a typical covalent bond breaks, but it is large enough that thermal fluctuations alone will rarely provide sufficient energy to push a base from one tautomeric form to another. This is why the rare tautomers are rare, because the energetic barriers and final state energies conspire to make them statistically unlikely, and yet not so unlikely that they never occur. The calculations thus provide a quantitative framework for understanding how often mispairing events might be initiated by tautomerization, though the connection between static energy differences and actual mutation rates involves many additional factors that we will consider in due course. The quantum chemical calculations also revealed subtleties that had not been anticipated from simpler considerations. Different tautomers of the same base can have quite different energy gaps depending on their environment, on whether they are isolated in the gas phase or embedded in the polar environment of water or nestled in the hydrophobic interior of the DNA helix. The presence of neighboring bases, of the backbone phosphate groups, of water molecules and metal ions. All of these environmental factors can shift the delicate balance between tautomeric forms, sometimes stabilizing the rare variants more than would be expected from gas phase calculations alone. Understanding these environmental effects became a major focus of computational research, requiring methods that could somehow capture both the quantum mechanical details of the bases themselves and the classical mechanical effects of the surrounding environment. This need gave rise to the development of hybrid methods, approaches that treat different parts of a molecular system with different levels of theory, applying the full rigor of quantum mechanics only where it is most needed while describing the remainder with simpler, faster methods based on classical physics. These quantum mechanics and molecular mechanics hybrid approaches, known by the abbreviation QMMM, partition a system into a small quantum region, perhaps a single base pair or a damaged nucleotide, embedded within a much larger classical region that includes the surrounding DNA, water molecules, and perhaps protein components. The quantum region feels the electrostatic influence of the classical environment, and sometimes the boundary is handled with sophisticated link atom schemes that ensure the electron density behaves properly where quantum and classical regions meet. Pavel Hobza, working primarily at the Institute of Organic Chemistry and Biochemistry in Prague, became one of the leading practitioners of these computational approaches, with a particular focus on the stacking interactions between adjacent base pairs in DNA. These stacking interactions, which arise from the subtle interplay of dispersion forces and electrostatic multipole interactions between the flat aromatic rings of the bases, are individually weak, perhaps 2 to 3 kilocalories per mole for each stacking contact, but they sum up over the length of the helix to provide a major contribution to DNA stability. Hobza's calculations showed that these stacking interactions are exquisitely sensitive to the relative geometry of neighboring bases, to the rise, twist, slide, and roll that describe how one base pair sits atop another, and that small changes in these parameters can significantly alter the energetic landscape that governs tautomerization and proton transfer. The proton transfer events that interconvert tautomeric forms are inherently dynamical processes, not static energy differences but actual chemical reactions in which a hydrogen nucleus moves from one potential energy well to another. Calculating the barriers to such reactions requires methods that go beyond simply optimizing molecular geometries and computing energies. Methods that can sample the transition states and reaction pathways that connect reactants to products. Ab initio molecular dynamics, which propagates the nuclear positions through time while computing the forces from quantum mechanical calculations at each time step, allows researchers to watch these proton transfers unfold in silico, to see the hydrogen waver at the top of the barrier before committing to one well or the other, and to compute the rate at which such transfers occur at different temperatures. These dynamical simulations have proven particularly valuable for understanding how proton transfer couples to the motion of surrounding water molecules and to the breathing fluctuations of the DNA helix itself. A proton transfer that would face a formidable barrier in an isolated base pair might become much more favorable when the surrounding water molecules reorganize to stabilize the emerging charge distribution, or when the helix breathes open slightly to relieve steric strain. The quantum chemistry of DNA cannot be divorced from the classical mechanics of its environment, and the most sophisticated modern calculations attempt to capture both levels of description simultaneously. Leonid Gorb, who collaborated extensively with Leshinsky at Jackson State, turned these computational methods toward understanding a different kind of DNA damage, the kind produced not by tautomerization but by ionizing radiation. When high-energy photons or particles strike DNA, they can knock electrons loose from the bases, creating radical cations that are chemically reactive and can undergo further transformations. Gorb's calculations traced the pathways by which these radiation-damaged bases can fragment, rearrange, or react with neighboring molecules, providing insight into the chemical mechanisms of radiation damage that would be nearly impossible to extract from experiment alone. The calculations showed how certain damaged bases might be more mutagenic than others, how some radical intermediates might be rapidly repaired while others persist long enough to cause lasting genetic change. The great challenge that runs through all of these computational studies is the treatment of the environment. The thousands of water molecules, the protein factors, the counterions, the thermal fluctuations that make biological systems so different from the pristine gas phase conditions that quantum chemical methods can most accurately describe. Various approaches have been developed to address this challenge. from continuum solvation models that represent water as a uniform dielectric medium, to explicit microsolvation where a handful of water molecules are included in the quantum region, to the full QM-MM treatment where vast numbers of water molecules are propagated classically while interacting with the quantum core. Each approach involves trade-offs between accuracy and computational cost, between physical realism and practical feasibility. In recent years, a new tool has entered the computational chemist's arsenal, one that would have been scarcely imaginable to the pioneers of quantum chemistry, and that is the application of machine learning methods to accelerate and extend the reach of quantum calculations. Neural networks and related algorithms can be trained on libraries of quantum chemical calculations, learning to recognize patterns in how electronic structure relates to molecular geometry, and then applying those learned patterns to predict the properties of new molecules without needing to repeat the expensive quantum calculations from scratch. For DNA systems, where the same basic building blocks appear over and over in different sequence contexts and different conformations, these machine learning approaches have proven remarkably effective, allowing researchers to screen vast numbers of potential mutation sites and predict which ones might be most susceptible to tautomerization or other forms of chemical change. The combination of machine learning with quantum mechanics is not a replacement for rigorous calculation, but rather an amplifier, a way of extending the insights gained from careful quantum chemical studies to larger systems and longer timescales than would otherwise be accessible. A neural network trained on DFT calculations of base pair tautomerization can predict tautomerization energies for base pairs in novel sequence contexts with nearly the same accuracy as a full quantum calculation, but in a fraction of a second rather than hours or days. This acceleration opens up new possibilities for understanding how mutation propensity varies along the genome, how certain sequences might be mutation hotspots while others remain resolutely stable. Another technological development that has transformed the field is the adoption of graphics processing units, originally designed for rendering video game graphics, as platforms for quantum chemical calculations. These GPUs, as they are known, contain thousands of simple processing cores that can execute identical operations simultaneously, a style of parallel computing that turns out to be ideally suited for many of the matrix operations that dominate quantum chemistry. Codes like terachemistry, developed originally by Todd Martinez at Stanford, have been written from the ground up to exploit GPU acceleration, achieving speed-ups of orders of magnitude over traditional processors and enabling quantum chemical calculations on entire DNA strands that would have been unthinkable a decade ago. With these accelerated methods, researchers can now perform molecular dynamic simulations in which the electronic structure is computed fresh at every time step, not for isolated base pairs or small model systems, but for full turns of the DNA helix containing hundreds of atoms, all solvated in explicit water with thousands more. These simulations can be run for long enough to observe rare fluctuations into tautomeric forms, to watch proton transfers happen spontaneously, to see how the dynamic breathing of the helix modulates the barrier heights for these chemical transformations. The computational microscope has become powerful enough to observe, at least in principle, the moment when a genetic mutation first appears. And yet, even with all of these advances, there remains a humbling awareness within the computational chemistry community of how much is still unknown, of how approximate our density functionals remain, of how many assumptions are buried within our molecular mechanics force fields, of how the full complexity of the cellular environment may harbor effects that we have not yet imagined. The calculations provide insight and understanding, They generate hypotheses and suggest experiments, but they do not yet provide the kind of quantitative predictive power that would allow us to say with certainty when and where mutations will arise. The machinery of computational quantum chemistry continues to be refined. The methods grow more accurate and more efficient with each passing year, and the dream of a fully predictive theory of genetic change, one that could tell us from first principles how DNA will change over generations, remains a guiding light for researchers in this field. For now, though, the calculations provide a window onto processes that would otherwise remain entirely opaque, and that is no small thing, to be able to see in the mind's eye, through the patient accumulation of numbers and the colorful renderings of electron densities and energy surfaces, the quantum mechanical origins of genetic change. And so we come now to a rather beautiful convergence, a place where the smallest phenomena in nature, those flickering quantum events we have been considering so gently throughout our time together, meet the grandest story biology has ever told, which is of course the story of evolution itself, the slow and patient unfolding of life across the immense stretches of geological time. And as we settle into this topic, you might find it pleasant to consider how these two scales of existence, the subatomic and the evolutionary, are not as separate as they might first appear, but are instead connected by the steady and reliable ticking of quantum processes deep within the molecular structure of DNA. Charles Darwin, that most patient and observant of naturalists, published his great work on the origin of species in 1859. And in doing so he provided the world with a mechanism for understanding how the diversity of life could arise through the gradual accumulation of small changes over vast periods of time. And yet Darwin worked in a kind of beautiful ignorance, for he had no knowledge whatsoever of the physical basis of heredity, no understanding of genes or chromosomes or the molecular structure that carries information from one generation to the next. And this gap in his knowledge troubled him greatly, as he readily admitted in his writings, because he could not explain how variations arose in the first place, nor how they were transmitted so faithfully from parent to offspring, and so he was left to speak of variation as something that simply appeared, as if by some process he could not name, which of course was precisely the case. For the discoveries that would illuminate these mysteries lay many decades in the future. The monk Gregor Mendel, working quietly in his monastery garden in what is now the Czech Republic, had in fact discovered the fundamental laws of inheritance at almost exactly the same time Darwin was publishing his theory. But Mendel's work remained obscure and unread for decades. And it was not until the turn of the 20th century that his experiments with pea plants were rediscovered and their significance appreciated. And even then, for many years, there seemed to be a tension between the Mendelian view of inheritance, with its discrete units of heredity, and the Darwinian view of gradual continuous change. And this tension was not resolved until the 1930s and 40s. When a group of brilliant biologists and mathematicians, including figures such as Ronald Fisher, J.B.S. Haldane, and Sewell Wright, developed what came to be known as the modern synthesis, a theoretical framework that showed how Mendelian genetics and Darwinian evolution were not only compatible but were in fact deeply complementary, each illuminating and strengthening the other. And this synthesis remains the foundation of evolutionary biology to this day, a reliable and well-tested framework within which we can now begin to consider the quantum mechanical underpinnings of genetic change. For several decades after the modern synthesis, the prevailing view among evolutionary biologists was that natural selection was the dominant force shaping the genetic makeup of populations. That most genetic changes that spread through a population did so because they conferred some advantage upon their bearers, however slight. And this selectionist view held sway until 1968, when a Japanese geneticist named Moto Kimura proposed something rather surprising and, at the time, quite controversial, which was the neutral theory of molecular evolution. And Kimura's insight, which he developed with characteristic mathematical rigor, was that the vast majority of genetic changes that become fixed in populations are not beneficial at all, nor are they harmful, but are instead selectively neutral, meaning that they have no effect whatsoever on the survival or reproduction of the organisms that carry them, and these neutral changes drift through populations by chance alone, accumulating steadily over time like a fine dust settling on a windowsill. And this idea, which initially met with considerable resistance, has since been supported by a great weight of molecular evidence and is now accepted as a fundamental principle of evolutionary genetics. The neutral theory has a rather remarkable implication, one that had been hinted at a few years earlier by the biochemist Emil Zuckerkandl and the chemist Linus Pauling, who proposed in 1962 the concept of the molecular clock. And the idea is beautifully simple. For if neutral mutations accumulate at a more or less constant rate over time, then the number of genetic differences between two species should be proportional to the time since they last shared a common ancestor. And this means that by counting the molecular differences between species, one can estimate how long ago they diverged, much as one might estimate the age of a sedimentary layer by the thickness of dust upon it. And this molecular clock has proven to be an extraordinarily useful tool for reconstructing the history of life, allowing biologists to date evolutionary events even in the absence of fossil evidence, and providing a kind of independent chronometer against which the geological and paleontological records can be calibrated. But for the molecular clock to work at all, there must be something keeping time, some underlying process that ticks along at a steady rate regardless of the particular species or environment in question. And here is where our quantum mechanical perspective becomes rather illuminating. For as we have discussed in earlier sections of our journey together, the fundamental process of mutation, the alteration of genetic information at the molecular level, is governed in part by quantum tunneling, that gentle and probabilistic process by which protons and electrons can occasionally find themselves on the opposite side of an energy barrier from where classical physics would expect them to be. And quantum tunneling rates are determined by the fundamental constants of nature, by the masses of the particles involved and the heights and widths of the energy barriers they must traverse. And these parameters do not change from one species to another or from one geological era to the next. And so if quantum tunneling contributes substantially to the baseline mutation rate, then we might expect that rate to be relatively constant across the tree of life, which is precisely what the molecular clock requires. This connection between quantum mechanics and the molecular clock is not merely speculative, for researchers have calculated that the tunneling rates for proton transfer in DNA base pairs are indeed consistent with the observed rates of spontaneous mutation. And while the correspondence is not perfect, and while other factors certainly contribute to mutation rates, the quantum mechanical component provides a kind of fundamental background rate, a floor below which mutation rates cannot easily fall, and this floor may explain why the molecular clock, despite the many biological and environmental factors that might be expected to perturb it, nevertheless keeps reasonably good time across such a vast range of organisms and timescales. Of course, mutation rates are not perfectly uniform, and as researchers have examined the genome in ever greater detail, they have discovered that some regions are far more prone to mutation than others, creating what are sometimes called hot spots and cold spots of mutability. And these variations in mutation rate are themselves quite interesting from a quantum mechanical perspective. for they suggest that local features of the DNA sequence can either enhance or suppress the tunneling processes that underlie spontaneous mutation. And one of the most well-documented examples of a mutation hotspot involves a particular 2-nucleotide sequence called a CpG dinucleotide, where a cytosine is followed immediately by a guanine. And in vertebrate genomes, these CpG sites are frequently modified by the addition of a methyl group to the cytosine, creating what is called 5-methylcytosine. And this methylated cytosine is chemically unstable in a rather specific way, being prone to a reaction called deamination in which it loses an amino group and is converted to thymine. And this conversion, if not repaired, results in a mutation from a CG base pair to a TA base pair. And the rate of this deamination reaction is substantially higher than the rate of other spontaneous mutations, which is why CpG sites are such prominent mutation hotspots. The chemistry of methylcytosine deamination involves processes that are themselves subject to quantum mechanical effects. For the breaking and forming of chemical bonds, the transfer of protons and electrons, and the tunneling of particles through energy barriers all play roles in determining how quickly this reaction proceeds. And so the elevated mutation rate at CpG sites is not a violation of the quantum mechanical framework we have been discussing, but rather a specific instance of it. A case where the local chemical environment shifts the quantum mechanical parameters in such a way as to increase the probability of a mutagenic event. And this example illustrates how the baseline quantum tunneling rate can be modulated by the molecular context without ever escaping the fundamental constraints that quantum mechanics imposes. The use of molecular clocks to date evolutionary events has been refined considerably over the decades since Zucker Kandel and Pauling first proposed the concept. And one of the leading figures in this refinement has been Sudhi Kumar at Temple University, who has developed sophisticated statistical methods for estimating divergence times while accounting for the variation in mutation rates that we have been discussing. And Kumar's work has shown that by carefully calibrating molecular clocks against well-dated fossil evidence, and by accounting for rate variation across different parts of the genome and different lineages, it is possible to achieve remarkably accurate estimates of when major evolutionary events occurred. Estimates that have transformed our understanding of the history of life on Earth. And while these methods are complex and require many careful assumptions, they ultimately rest upon the reliability of the underlying mutation process, a process whose quantum mechanical foundations provide a kind of ultimate stability that makes the entire enterprise possible. There is another rather elegant observation that emerges when we consider mutation rates from a quantum mechanical perspective. And this is the curious fact that the mutation rate per genome per generation is roughly similar across a very wide range of organisms, from bacteria to fruit flies to humans, despite the enormous differences in genome size and generation time among these species. And this approximate constancy suggests that there may be something fundamental about the mutation rate, some underlying physical limit that evolution cannot easily transgress. And the quantum mechanical tunneling rate provides a natural candidate for such a limit, for it sets a floor on how infrequently spontaneous mutations can occur. A flaw determined not by biological adaptation but by the basic physics of proton and electron behavior in molecular systems. The evolutionary biologist Michael Lynch has written extensively about what he calls the mutation rate paradox, which is the observation that mutation rates are typically much higher than would seem optimal from a purely selective standpoint. For most mutations are either neutral or harmful, and only a very small fraction are beneficial, which means that organisms are constantly burdened by a rain of deleterious changes that must be filtered out by natural selection at considerable cost. And one might naively expect that evolution would favor ever more accurate DNA replication and ever more vigilant repair systems, driving mutation rates down toward zero. But this has not happened, and mutation rates remain surprisingly high across all forms of life. And Lynch has argued that this reflects a kind of evolutionary equilibrium in which the benefits of further reducing mutation rates outweighed by the costs of maintaining ever more elaborate replication and repair machinery. And from our quantum mechanical perspective, we might add that there may also be a physical limit below which mutation rates simply cannot fall, a limit set by the irreducible probability of quantum tunneling events in the DNA double helix. This brings us to a rather philosophical consideration, which is that the relationship between mutation rate and evolutionary adaptation is not straightforward. For while too many mutations lead to genetic disorders, cancer, and the accumulation of deleterious changes that can ultimately drive a population to extinction, too few mutations would also be problematic, leaving a population unable to adapt to changing environments, unable to generate the raw material upon which natural selection operates, and so life exists in a kind of delicate balance, with mutation rates high enough to permit adaptation but low enough to prevent mutational meltdown. And this balance is maintained not only by the biological machinery of DNA replication and repair but also, at the deepest level, by the quantum mechanical processes that govern the spontaneous alteration of genetic information. The quantum effects we have been discussing thus set what might be called the ultimate limits on the rate of evolutionary change. For no matter how efficient the repair machinery becomes, no matter how carefully the cell proofreads its DNA, there will always be some irreducible background of quantum tunneling events that cannot be prevented, that slip beneath the threshold of detection and become incorporated into the germline, passed on to future generations as permanent changes in the genetic record. And this quantum background is not a bug but a feature, in the sense that it ensures a steady supply of genetic variation upon which evolution can act. And it may be that this quantum contribution to mutation is not merely tolerated by life but is in some sense necessary for its long-term survival and adaptation. As we rest with these ideas, letting them settle gently like leaves upon still water, we might reflect on the remarkable continuity that connects the fleeting quantum events within a single DNA molecule to the vast sweep of evolutionary history, the billions of years during which life has diversified and adapted and spread across every corner of our planet. And at every step along this journey, the quiet processes of quantum tunneling have been at work, introducing variations, testing possibilities, providing the raw material for natural selection to shape and refine. And while Darwin could not have known any of this, could not have imagined the subatomic world that underlies the variations he observed so carefully in his finches and orchids and barnacles, we can now see that his theory and the quantum theory are deeply intertwined, that the one flows into the other across the scales of nature, connecting the very small to the very large in a single continuous fabric of cause and effect. And so the molecular clock ticks on, its rhythm set by the fundamental constants of physics, by the masses of protons and the heights of energy barriers. And as we listen to that gentle ticking, as we imagine the slow accumulation of changes across millions and billions of years, we might find ourselves feeling a quiet sense of wonder, a gentle appreciation for the elegant way in which the universe has arranged for life to change and grow and explore the endless possibilities of existence, all through the patient and unhurried workings of quantum mechanics, deep within the spiraling strands of DNA. And so we come now to a more somber consideration, though we shall approach it with the same quietude and measured pace that has carried us through our earlier explorations. For even when we speak of things gone wrong, of cells that have lost their way and begun to multiply without restraint, there is a certain comfort to be found in understanding in tracing the molecular pathways that lead from the tiniest quantum fluctuation to the larger disruptions we call disease, and in recognizing that the same beautiful repair mechanisms we have been discussing, when they falter or fail, can set in motion a cascade of changes that accumulates over years and decades, eventually giving rise to what we know as cancer. The idea that cancer might arise from changes within the cell itself, rather than from some external miasma or imbalance of humors, began to take shape in the early years of the 20th century, when a German zoologist named Theodor Boveri, working at the University of Würzburg, made careful observations of sea urchin eggs that had been fertilized by two sperm simultaneously, a condition that resulted in cells with abnormal numbers of chromosomes, and these cells, he noted, behaved erratically, dividing in disordered ways and failing to develop properly. And from these observations Boveri drew a remarkable conclusion that he published in 1914, proposing that cancer cells might similarly arise from disturbances in the chromosomal complement of normal cells, that somewhere within the nucleus lay the instructions for orderly growth and division, and that damage to these instructions might release a cell from its normal constraints, allowing it to proliferate without regard for the needs of the organism as a whole. Boveri's hypothesis, which we now call the somatic mutation theory of cancer, suggested that tumors were not infections, not invasions from outside, but rather the consequence of changes occurring within the body's own cells, mutations that accumulated in the somatic tissues, meaning the ordinary cells of the body rather than the germ cells that give rise to offspring. And while Boveri could not have known the molecular nature of these changes, could not have imagined the double helix or the four-letter alphabet of nucleotides, his insight proved remarkably prescient, establishing a framework that would guide cancer research for the century to follow. And you might find it soothing to reflect that even a century ago, patient observation of sea urchin eggs could illuminate principles that remain central to our understanding today. More than 50 years after Boveri's work, an American physician and geneticist named Alfred Knudsen, working at the University of Texas and later at the Fox Chase Cancer Center in Philadelphia, turned his attention to a rare childhood cancer of the eye called retinoblastoma, which occurs in two distinct patterns, sometimes appearing in just one eye and sometimes in both, sometimes arising in children with a family history of the disease and sometimes appearing spontaneously in families where no one had been affected before. And Knudsen, analyzing the statistics of these different presentations with careful mathematical reasoning, proposed in 1971 what has come to be known as the two-hit hypothesis, suggesting that retinoblastoma required two separate mutations to occur, both copies of a particular gene needing to be inactivated before a retinal cell would lose its normal growth control and begin to form a tumor. In the hereditary form of the disease, Knudsen proposed, a child inherits one damaged copy of the gene from an affected parent, so that every cell in the body already carries the first hit, and then a second, random mutation occurring in any one of the millions of retinal cells completes the process. And because there are so many opportunities for this second hit to occur, children with inherited susceptibility typically develop tumors in both eyes and at younger ages, whereas in the sporadic, non-hereditary form, both hits must occur by chance in the same cell, a far less likely event that typically results in tumors appearing later and in only one eye. And this elegant model, derived from statistical analysis of clinical data, predicted the existence of tumor suppressor genes years before such genes were molecularly identified, and the gene responsible for retinoblastoma, now called RB1, was eventually isolated in 1986, confirming Knudsen's hypothesis with molecular precision. the concept of tumor suppressor genes, and their partner category of oncogenes, which are genes that when mutated actively drive cell proliferation, has proven essential to our understanding of how cancer develops. And among all the genes implicated in human cancer, none has proven more important than one called TP53, which encodes a protein known simply as P53, sometimes called the guardian of the genome for its central role in detecting DNA damage and orchestrating the cell's response, either halting the cell cycle to allow time for repair or, when damage is too severe, triggering programmed cell death to eliminate the damaged cell before it can pass its mutations to daughter cells. And this protein, P53, is found to be mutated or inactivated in approximately half of all human cancers, an astonishing proportion that speaks to its fundamental importance in preventing the uncontrolled growth that characterizes malignancy. When we consider the molecular details of how P53 works, we find a protein that binds directly to DNA, recognizing specific sequences and activating the transcription of genes involved in cell cycle arrest, DNA repair, and apoptosis. And when DNA damage occurs, whether from radiation, from chemical carcinogens, or from the spontaneous quantum fluctuations we have been discussing, P53 levels rise in the cell. The protein accumulates in the nucleus, and it switches on a program of protective responses. But when P53 itself is mutated, when the guardian is disabled, the cell loses its ability to respond appropriately to damage. Mutations accumulate unchecked, and the multi-step path toward cancer advances more rapidly. And you might picture P53 as a careful proofreader who has been reading manuscripts for many years, catching errors that would otherwise slip through. And when this proofreader retires or becomes incapacitated, the quality of the final product inevitably suffers. Another pair of genes whose mutations have proven particularly consequential are BRCA1 and BRCA2, which encode proteins involved in the repair of double-strand DNA breaks through the high-fidelity pathway of homologous recombination that we discussed earlier. And women who inherit mutations in either of these genes face substantially elevated risks of breast and ovarian cancer, with lifetime breast cancer risks approaching 60 to 70% in some studies, compared to approximately 12% in the general population. Because when homologous recombination is impaired, cells must rely on more error-prone repair pathways to fix double-strand breaks, leading to the accumulation of chromosomal rearrangements and the gradual erosion of genomic stability that characterizes cancer cells. And the discovery of these genes, and the development of genetic testing to identify carriers, represents one of the genuine triumphs of cancer genetics, allowing individuals at elevated risk to pursue enhanced surveillance or preventive measures. Let us now return to the quantum mechanical perspective, for when we ask how carcinogens actually damage DNA, how the substances we recognize as cancer-causing agents exert their effects at the molecular level, we find ourselves once again in the realm of electron orbitals and chemical bonds and the quantum mechanical principles that govern molecular interactions. And one of the most thoroughly studied carcinogens is a compound called benzo(A)-pyrene, which is found in tobacco smoke, in charred food, in diesel exhaust, and in various industrial emissions. A polycyclic aromatic hydrocarbon consisting of 5 fused benzene rings that forms when organic matter undergoes incomplete combustion. Benzo(A)-pyrene itself is not actually the agent that damages DNA, for the body treats it as a foreign substance to be eliminated, and in the process of metabolizing this compound, enzymes in the liver convert it through a series of steps into more reactive forms, including a highly electrophilic species called benzo(A) pyrene diol epoxide. And this metabolite, with its strained three-membered ring and its positive charge, is extraordinarily reactive toward the nucleophilic sites in DNA, particularly the amino group of guanine. And when benzo-A pyrene diol epoxide encounters the double helix, it forms a covalent bond with guanine, creating what is called a DNA adduct, a bulky chemical modification that distorts the structure of the helix and interferes with normal replication and transcription. The quantum mechanics here involves the redistribution of electron density as the epoxide ring opens and a new bond forms with the DNA base, a process governed by the wave functions of the interacting electrons and the overlap of molecular orbitals, and the specificity of this reaction, the preference for guanine over other bases, reflects the quantum mechanical properties of each molecule, the energetics of electron transfer, the geometry of approach, and the stability of the final product. And when we speak of carcinogenesis at this level, we are describing events that unfold on the scale of angstroms and femtoseconds, individual molecules colliding and reacting according to the laws of quantum chemistry, each adduct formation representing a discrete quantum event with probabilistic rather than deterministic outcomes. A Croatian-American molecular biologist named Miroslav Radman, working at the University of Paris and other institutions, made important contributions to our understanding of how mutations accumulate in cancer cells, particularly through his studies of what he called mutator phenotypes, which are states in which cells exhibit dramatically elevated mutation rates due to defects in their DNA repair or replication fidelity mechanisms. And Radman recognized that cancer cells often display genomic instability, producing new mutations at rates far exceeding those of normal cells. And this instability, while potentially dangerous for the cancer cell itself, provides the raw material for evolutionary adaptation, allowing tumors to diversify and develop resistance to treatments. One of the most clinically important examples of a mutator phenotype arises from defects in mismatch repair, the system we discussed earlier that catches and corrects replication errors where the wrong base has been inserted, and when mismatch repair is deficient, whether through inherited mutations or through silencing of repair genes during tumor development. The result is a dramatic increase in the mutation rate, particularly at repetitive sequences called microsatellites, which are short stretches of DNA where the same 1, 2, or 3 nucleotide motif is repeated many times. And these repetitive sequences are particularly prone to errors during replication, as the polymerase can slip and misalign the template and daughter strands, producing insertions or deletions that alter the length of the repeat. When mismatch repair is functioning normally, these slippage errors are caught and corrected, but when repair is deficient, they accumulate, producing a distinctive pattern of changes in microsatellite lengths that we call microsatellite instability, and tumors showing this pattern, often designated MSI high, have particular clinical characteristics and, interestingly, tend to respond unusually well to certain immunotherapy treatments, perhaps because their high mutation burden creates many abnormal proteins that the immune system can recognize as foreign. And Lynch syndrome, also known as hereditary non-polyposis colorectal cancer, is a condition caused by inherited mutations in mismatch repair genes, conferring substantially elevated risks of colorectal, endometrial, and other cancers. And individuals with Lynch syndrome benefit from enhanced surveillance that can detect cancers at early, more treatable stages. The role of oxidative stress in cancer brings us back to the quantum events we discussed when considering reactive oxygen species. those molecules with unpaired electrons that can damage DNA through processes governed by free radical chemistry. And the most common oxidative lesion in DNA, atoxoguanine, which forms when oxygen radicals attack the guanine base, has been measured at elevated levels in many tumor types, reflecting the increased metabolic activity and oxidative stress that characterize cancer cells. And this lesion, if not repaired, causes the characteristic G to T transversion mutation during replication, adding to the burden of genetic changes that accumulate as cancer progresses. And you might find it interesting that some cancer treatments, including certain forms of radiation therapy and chemotherapy, work partly by overwhelming cancer cells with oxidative damage, exploiting the very vulnerability that contributed to their initial development. Perhaps no human condition illustrates the importance of DNA repair more poignantly than xeroderma pigmentosum, a rare inherited disorder caused by defects in the nucleotide excision repair pathway, the system responsible for removing bulky lesions like those caused by ultraviolet light. And individuals with this condition cannot effectively repair the thymine dimers and other damage that sunlight produces in their skin cells. And as a result, they experience extreme sensitivity to sunlight and a 1,000-fold increase in the risk of skin cancers compared to the general population, developing numerous squamous cell carcinomas and melanomas often beginning in childhood. And these patients must take extraordinary precautions to avoid sun exposure, sometimes living essentially nocturnal lives. And while their condition is tragic, it demonstrates with painful clarity the essential role that DNA repair plays in protecting us from the damaging effects of our environment. The concept of tumor mutation burden, the total number of mutations present in a cancer's genome, has emerged in recent years as an important predictor of response to immunotherapy, particularly to a class of drugs called immune checkpoint inhibitors, which work by releasing the brakes on the immune system's ability to attack cancer cells. And tumors with high mutation burdens, such as those arising from mismatch repair deficiency or from heavy carcinogen exposure like smoking-related lung cancers, tend to produce more abnormal proteins, called neoantigens, that can be recognized by immune cells as foreign. And clinical studies have confirmed that patients whose tumors have high mutation burdens are more likely to respond to checkpoint inhibitors, though the relationship is probabilistic rather than absolute. And mutation burden represents just one of many factors that influence treatment outcomes. And so we arrive at a synthesis, recognizing that the quantum tunneling events and chemical reactions we have been discussing throughout this narration, the tautomeric shifts, the proton transfers, the spontaneous depurinations and deaminations, the formation of adducts and the generation of reactive oxygen species, all represent the molecular origins of the genetic changes that, when they accumulate in the right combination and evade the repair systems meant to correct them, can initiate the multi-step journey toward cancer, a journey that typically unfolds over decades, requiring multiple independent mutations in genes that control cell growth, death, and genomic stability, each mutation representing a discrete molecular event with quantum mechanical underpinnings. The path from a single quantum fluctuation to a diagnosable tumor is long and uncertain, passing through many checkpoints and surveillance mechanisms, most of which function with remarkable reliability, and the vast majority of potentially harmful changes are caught and corrected, or result in cell death rather than transformation. But occasionally, rarely, a change slips through, and then another, and then another, and over many years the accumulated damage crosses some threshold, and a cell begins to divide without restraint, and its descendants accumulate further changes, and a tumor emerges. And understanding this process at the molecular level, understanding the quantum events that initiate it and the repair failures that permit its progression, represents one of the great achievements of modern biology, offering not only insight but also the foundation for treatments that target specific molecular abnormalities. And while we shall not dwell on outcomes or prognoses, for this is not that kind of narration, we might simply note that knowledge, even knowledge of difficult things, carries its own quiet comfort, the comfort of understanding what was once mysterious. And with that thought we shall allow ourselves to rest, breathing slowly, knowing that even as we sleep, countless faithful enzymes patrol our DNA, catching errors, making repairs, maintaining the integrity of our genomes with tireless molecular devotion. And so we come now to the very beginning, to a time so distant that the numbers themselves become almost dreamlike, a span of nearly 4 billion years separating us from the first stirrings of molecular replication on a young and restless Earth. This is a gentle story, a patient unfolding of chemistry and chance, and we shall move through it slowly, allowing each detail to settle softly before moving on to the next. In 1986, a molecular biologist named Walter Gilbert, who had shared the Nobel Prize in Chemistry just 6 years earlier for his work on DNA sequencing, wrote a short paper in the journal Nature in which he proposed a simple and rather elegant phrase to describe a possible early chapter in the history of life. He called it the RNA world, and with those 3 words he gave a name to an idea that had been forming quietly in the minds of researchers for some years: the notion that before there was DNA, before there were proteins as we know them today, there existed a simpler molecular system in which RNA served both as the carrier of genetic information and as the catalyst for chemical reactions. This was a tidy solution to a puzzle that had long troubled those who thought carefully about origins, because in modern cells, DNA requires proteins to replicate, and proteins require DNA to encode their sequences, and so one seems always to need the other, a circular dependency that made it difficult to imagine how either could have arisen first. But RNA, Gilbert suggested, might have done both jobs alone, and this possibility opened a door through which much subsequent research would pass. Of course, naming a world is not the same as building one, and the problem of how the first nucleotides, the building blocks of RNA, might have formed on the early Earth remained stubbornly difficult. A nucleotide, you see, is not a simple molecule. It consists of 3 parts joined together in a precise arrangement: a sugar called ribose, a phosphate group, and a nitrogenous base. And each of these components must be synthesized and then connected in just the right way. In 1953, the same year that Watson and Crick published their famous paper on the structure of DNA, a young graduate student at the University of Chicago named Stanley Miller performed an experiment that captured the imagination of scientists and the public alike. Working under the supervision of Harold Urey, Miller filled a flask with gases thought to resemble the atmosphere of the early Earth: methane, ammonia, hydrogen, and water vapor, and he passed electrical sparks through this mixture to simulate lightning. After a week, he analyzed the contents and found, to widespread astonishment, that the flask now contained a variety of amino acids, the building blocks of proteins. It seemed, for a hopeful moment, that the chemistry of life might arise with surprising ease from simple beginnings. But the Miller experiment, for all its elegance, had limitations that became clearer as researchers probed further. The amino acids it produced were relatively simple molecules, and attempts to generate nucleotides using similar methods proved far less successful. The sugar ribose is fragile and prone to decomposition. The bases are difficult to form in the right configurations, and bringing all three components together in the correct arrangement seemed to require conditions that were hard to reconcile with any plausible early Earth environment. For decades, this problem, sometimes called the nucleotide synthesis problem, stood as one of the great obstacles to understanding how life might have begun, and many thoughtful researchers wondered whether there might be some piece of the puzzle that everyone was missing. That missing piece, or at least one version of it, emerged in 2009 from a laboratory at the University of Cambridge, where a chemist named John Sutherland and his colleagues published a paper that approached the problem from an entirely new angle. Rather than trying to build nucleotides by assembling their components separately and then joining them, Sutherland's team discovered a pathway in which the sugar and the base could form together, their synthesis intertwined rather than sequential. Using simple starting materials, including cyanamide, cyanoacetylene, glycolaldehyde, and phosphate, and subjecting them to a series of reactions that included exposure to ultraviolet light, they were able to produce activated pyrimidine nucleotides, the letters C and U in the RNA alphabet, in a form that was ready for further chemistry. This was a breakthrough of considerable importance, because it showed that the nucleotide synthesis problem, while not fully solved, was perhaps not as insurmountable as it had seemed. What makes Sutherland's work particularly interesting from our present perspective is the role played by ultraviolet light in the synthetic pathway. The early Earth, before the rise of atmospheric oxygen and the formation of the protective ozone layer, would have been bathed in far more intense ultraviolet radiation than the surface receives today. This same radiation, as we have discussed in earlier sections, is capable of damaging DNA by exciting electrons and inducing the formation of lesions such as cyclobutane pyrimidine dimers. Yet here, in the prebiotic context, that very same quantum mechanical process, the absorption of photons and the excitation of electrons to higher energy states, appears to have played a constructive role, driving chemical reactions that would not otherwise proceed. It is a quiet irony, really, that the same quantum phenomenon that modern cells must constantly defend against may have been essential for the origin of the genetic molecules in the first place. The wavelengths of light that threaten the integrity of our genomes today may once have been the spark that set the whole process in motion. We should pause here to consider what it means for a molecule to replicate, and what level of accuracy is required for that replication to support the kind of gradual change that we call evolution. In modern cells, as we have seen, the copying of DNA is extraordinarily precise, with error rates as low as 1 mistake per billion nucleotides copied. But the first replicators, whatever they were, would have had no enzymes to proofread their work, no elaborate repair machinery to correct their mistakes. They would have been simple, imperfect, prone to error. And this raises a question that the German biophysicist Manfred Eigen addressed with mathematical rigor in the 1970s, in work that remains foundational to our understanding of early evolution. Eigen showed that for a population of replicating molecules to maintain coherent genetic information over many generations, the error rate could not exceed a certain threshold, which depended on the length of the genetic sequence being copied. If the error rate was too high, the information would degrade with each generation, a process he called error catastrophe, in which the offspring of any given molecule would be so different from their parent that no meaningful inheritance could occur. 4 primitive replicators with no repair mechanisms, Agin calculated that this threshold lay somewhere in the range of 1 to 10% errors per nucleotide per replication, a figure that corresponds to a maximum genome length of perhaps 100 to a few thousand nucleotides. Longer genomes would require more accurate replication, but more accurate replication would seem to require longer genomes to encode the necessary enzymes. And so there appeared to be a catch, a limit on the complexity that could evolve without some way of improving fidelity first. This threshold has important implications for our understanding of the RNA world, because it suggests that the earliest ribozymes, RNA molecules capable of catalyzing chemical reactions, must have been rather short, perhaps just a few dozen to a few hundred nucleotides in length. And yet such molecules would need to perform at least one essential function, namely, replicating themselves or assisting in their own replication. Studies by Gerald Joyce and his colleagues at the Scripps Research Institute have explored this question through elegant experiments in directed evolution, in which populations of RNA molecules are subjected to selection for catalytic activity over many generations in the laboratory. Joyce's work has shown that ribozymes can indeed evolve to perform a variety of functions, including the copying of RNA templates, and that this evolution can proceed through the accumulation of small improvements even with the high mutation rates characteristic of RNA replication. These experiments do not recreate the conditions of the early Earth, of course, but they demonstrate that the basic principles are sound, that RNA molecules can carry information, undergo mutation, experience selection, and evolve increasing complexity over time. The transition from RNA to DNA, which occurred at some point in the early history of life, represents a significant shift in the chemistry of genetic information storage. DNA differs from RNA in two main ways: the presence of deoxyribose rather than ribose as the sugar component, and the substitution of thymine for uracil as one of the bases. The absence of a hydroxyl group at the 2' position of the sugar makes DNA considerably more stable than RNA, less prone to hydrolysis, and therefore better suited for the long-term storage of genetic information. The reasons for this increased stability are quantum mechanical in nature, relating to the electronic structure of the sugar and its effects on the reactivity of the phosphodiester backbone. In RNA, the 2' hydroxyl group can participate in reactions that lead to strand cleavage, whereas in DNA, this pathway is blocked. Thus, while RNA may have been the first genetic molecule, DNA proved to be the better archivist, and natural selection, once it had the enzymatic machinery to produce deoxyribose and incorporate it into nucleotides, would have favored the transition. There is also the question of how, in the crowded and chaotic chemistry of the early Earth, the right molecules might have been selected from among the vast number of possible compounds. Here we enter territory that is more speculative, but no less interesting for that. Some researchers have suggested that quantum coherence, the phenomenon in which quantum mechanical states remain correlated over time and space, may have played a role in the early chemical selection of biomolecules. The idea is that certain molecules, those with particular electronic structures and arrangements of atoms, might have been able to maintain coherent quantum states long enough to gain some advantage in chemical reactions, perhaps by allowing more efficient energy transfer or by favoring certain reaction pathways over others. This remains an area of active investigation, and the evidence is not yet conclusive, but it is intriguing to consider that the molecules which eventually became the basis of life may have been, in some subtle quantum mechanical sense, especially well suited to the task. we should also consider the physical environment in which these early chemical processes took place. In recent years, the biochemist Nick Lane and his collaborators, building on earlier work by Michael Russell, have developed a detailed hypothesis concerning the role of alkaline hydrothermal vents in the origin of life. These vents, which form where seawater reacts with certain types of rock on the ocean floor, produce structures riddled with tiny pores and labyrinthine channels, natural compartments that could have served as primitive cells. More importantly, these vents generate a gradient of protons, a difference in hydrogen ion concentration between the alkaline fluid emerging from the rock and the more acidic ocean water. Lane has argued that this natural proton gradient could have provided a source of energy for early chemical reactions, analogous to the proton gradients that power the synthesis of ATP in modern cells. The chemistry involved is delicate and specific, involving the flow of protons across thin mineral walls and the coupling of this flow to the formation of reactive molecules. But it offers a plausible account of how the first metabolic processes might have arisen without the need for complex enzymes or genetic information. If Lane's hypothesis is correct, then the origin of life was not a single event but a gradual process, a slow accumulation of chemical systems that fed on the energy provided by the Earth itself, by the heat of its interior and the chemistry of its rocks and waters. The first replicators would have emerged within this context, shaped by the quantum mechanical properties of their constituent atoms, constrained by the error thresholds that govern their ability to maintain information, and gradually gaining in complexity and sophistication over millions of years. There is no drama here, no sudden spark of creation, only the patient working out of chemistry and physics over spans of time that dwarf all human experience. And so the molecules that would eventually become DNA, the very substance whose quantum mechanical behavior we have been exploring throughout these sections, began their long journey under conditions that we can only dimly reconstruct. The same ultraviolet light that today threatens the integrity of the double helix once drove the synthesis of its precursors. The same quantum effects that now cause mutations and require elaborate repair systems were present at the very beginning, shaping the chemistry of the first replicators and setting the stage for everything that followed. It is a pleasing continuity, a thread that connects the ancient chemistry of the prebiotic Earth to the intricate molecular machinery of the cells that now compose our bodies. The story of those first replicators is, in many ways, the story of how information came to be preserved and transmitted, how the quantum mechanical properties of atoms and electrons were harnessed, through no conscious design but simply through the relentless logic of selection and chance, to create systems capable of evolution. It is a gentle story, a quiet accumulation of chemical possibilities, and it continues still, in every cell of every living thing, as DNA unwinds and copies itself, letter by patient letter, carrying the information of the past into the future. You may close your eyes now, if you have not already, and let these thoughts settle softly, these images of ancient seas and warm volcanic vents, of nucleotides forming under ultraviolet skies, of ribozymes stumbling toward replication in a world without enzymes or oversight. The chemistry proceeds without urgency, the quantum effects unfold without fanfare, and the story continues in its own time, as it has for nearly 4 billion years, patient and unhurried and boundlessly slow. And so we come now, gently and without any particular urgency, to consider what lies ahead, to the quiet unfolding of discoveries that may reshape our understanding of life itself over the coming decades. The field we have been exploring throughout these restful hours, This intersection of quantum physics and the molecular machinery of heredity has only recently emerged from the shadows of scientific skepticism to take its place as a legitimate area of inquiry, and we might spend some time now tracing how this transformation occurred and where it may lead us, all at a pace suitable for the drifting mind. In the year 2018, a rather significant gathering took place in London, hosted by the Royal Society, that venerable institution founded in 1660 and dedicated to the advancement of natural knowledge. The meeting brought together physicists, biologists, chemists, and researchers from various disciplines, all converging to discuss the nascent field of quantum biology. What made this particular assembly noteworthy was not any single dramatic announcement, but rather the quiet acknowledgement it represented, an acceptance by the broader scientific community that quantum effects in biological systems had moved beyond the realm of speculation into something worthy of serious, sustained investigation. The discussions were measured and careful, as scientific discussions tend to be, covering topics from photosynthesis to avian navigation to enzyme catalysis, and yes, to the quantum mechanical processes that might influence DNA mutation and repair. The meeting did not resolve every question or settle every debate, but it served as a kind of watershed moment, after which quantum biology could be discussed in mainstream scientific circles without the raised eyebrows or gentle skepticism that had sometimes greeted such ideas in earlier decades. The implications of this growing acceptance extend naturally into the practical realm of medicine, where an understanding of quantum processes in DNA might eventually inform the design of therapeutic interventions. Consider for a moment the process of drug development as it currently exists, a lengthy and expensive endeavor involving the screening of countless molecular candidates, the testing of their interactions with biological targets, the gradual refinement of chemical structures to improve efficacy while minimizing unwanted effects. Much of this work proceeds through a combination of empirical trial and established chemical principles, treating molecules as classical objects that interact through well-understood forces. But if the binding of repair enzymes to damaged DNA involves quantum tunneling, if the recognition of mutations depends on the subtle redistribution of electron density that quantum mechanics describes, then a deeper appreciation of these effects might open new avenues for therapeutic design. One could imagine, in principle, drugs that are engineered not merely to occupy a particular binding site but to exploit the quantum mechanical properties of that interaction. Molecules whose electron configurations are optimized to enhance or inhibit specific quantum processes involved in DNA repair or mutation recognition. This brings us quite naturally to the subject of quantum computing, that much-discussed technology that has progressed steadily from theoretical curiosity to practical reality over recent years. Companies such as IBM and Google have invested considerable resources in developing quantum processors, machines that exploit superposition and entanglement to perform certain calculations far more efficiently than classical computers can manage. In 2019, Google announced that their quantum processor had achieved what they termed quantum supremacy, completing a calculation in minutes that would have required thousands of years on the most powerful classical supercomputers then available. While the particular calculation involved was of limited practical significance, the demonstration served as proof of principle, an indication that quantum computers might eventually tackle problems of genuine scientific importance. Among the most promising applications for such machines is the simulation of quantum systems themselves, a task at which classical computers struggle mightily as the number of particles increases. A molecule of even moderate size involves so many interacting quantum states that accurately modeling its behavior becomes computationally intractable through classical means. This limitation has significant implications for our understanding of DNA, where the quantum mechanical interactions between thousands of atoms determine the precise energetics of mutation, the tunneling probabilities that influence proton transfer, the electronic structures that guide enzyme recognition and repair. A sufficiently powerful quantum computer could, in principle, simulate these processes with a fidelity that classical approaches cannot achieve, revealing details of DNA quantum mechanics that remain hidden from our current computational methods. Researchers at IBM and various academic institutions have already begun applying quantum computing techniques to problems in chemistry and biochemistry, starting with relatively simple molecules and gradually working toward more complex systems. The simulation of DNA quantum effects represents a more distant goal, requiring advances in both hardware and algorithms. But the trajectory of progress suggests that such simulations may become feasible within the lifetimes of many people listening to these words. Another area where quantum understanding might inform practical application involves the gene editing technology known as CRISPR, which has transformed biological research since its development in the early 2010s. The system relies on a molecule of guide RNA that directs an enzyme to a specific location in the genome, where it introduces a targeted cut in the DNA strand. The precision of this targeting depends on the binding interaction between the guide RNA and its complementary DNA sequence, a process governed ultimately by quantum mechanical forces. The hydrogen bonds that form between base pairs, the stacking interactions between adjacent nucleotides, the conformational changes that occur as recognition proceeds, all of these involve the same quantum effects we have been discussing in other contexts. A more complete understanding of the quantum mechanics underlying guide RNA binding might enable researchers to design more specific and efficient CRISPR systems, reducing off-target effects and improving the accuracy of genetic modifications. This represents perhaps a less dramatic application of quantum biology than some we might imagine, but it illustrates how fundamental physical understanding can propagate through to practical technologies in ways that benefit human health and scientific capability. The detection of mutations, particularly those rare changes at single points in the genome that can initiate disease, remains a challenge for current technologies. Quantum sensors, devices that exploit quantum mechanical effects to achieve measurements of extraordinary sensitivity, might eventually enable the detection of such mutations with unprecedented precision. These sensors rely on phenomena such as quantum entanglement and superposition to overcome the noise limitations that constrain classical measurement techniques. Researchers have already demonstrated quantum sensors capable of detecting single molecules, and the application of such technologies to genetic analysis represents an active area of investigation. One could envision, at some point in the coming decades, diagnostic tools that identify cancer-initiating mutations in their earliest stages, or screens that detect inherited genetic variants with perfect accuracy, all enabled by the same quantum physics that underlies the mutations themselves. The symmetry of such an application is rather pleasing to contemplate, using quantum mechanics to observe the quantum mechanical processes that shape our genomes. Among the researchers who have contributed to our understanding of quantum effects in biological systems, the work of Vlatko Vedral at Oxford deserves particular mention in this context. Vedral has explored the boundaries of quantum theory with notable creativity, investigating how quantum effects manifest in various physical systems, including those of biological relevance. His theoretical work has helped to establish the frameworks through which quantum coherence and entanglement in warm, wet biological environments can be understood and quantified. The contributions of Vedral and researchers like him have been instrumental in moving quantum biology from the margins toward the mainstream, providing the mathematical and conceptual tools necessary for rigorous investigation of these phenomena. The variability among individuals in their susceptibility to mutation, a phenomenon influenced by genetic differences in DNA repair capacity, suggests another avenue for the application of quantum biological understanding. If quantum tunneling rates depend on the precise molecular structure of DNA and its associated proteins, and if small variations in these structures alter the probabilities of proton transfer and other quantum processes, then individuals might differ systematically in their quantum mechanical mutation rates. This could inform approaches to personalized medicine, where treatments and preventive measures are tailored to the specific biological characteristics of each patient. Someone whose genetic makeup results in elevated quantum tunneling rates in certain genomic regions might benefit from targeted interventions, while another person with different molecular properties might require an entirely different approach. The practical implementation of such personalized quantum medicine remains distant, requiring advances in both our understanding of individual quantum variation and our ability to translate that understanding into therapeutic action. But the conceptual framework is already taking shape. Synthetic biology, the field concerned with designing and constructing biological systems with novel properties, represents yet another domain where quantum understanding might prove valuable. If certain quantum effects are beneficial for genetic stability, and if we can identify the molecular features that promote such effects, then we might design synthetic DNA sequences optimized for particular quantum mechanical properties. Genetic circuits engineered to minimize mutation rates through enhanced quantum coherence, or synthetic genomes constructed to exploit beneficial tunneling effects. These ideas remain largely speculative at present, but they illustrate the kinds of possibilities that quantum biology opens. The design of life itself might eventually proceed not only through the traditional tools of molecular biology, but through an understanding of quantum mechanics at the most fundamental level. The origins of genetic disease in quantum mechanical processes suggest corresponding possibilities for intervention. If certain inherited conditions result from elevated mutation rates in specific genes, and if those elevated rates stem from particular quantum properties of the affected DNA sequences, then targeted interventions might be designed to modulate those quantum effects. Small molecules that alter the energy landscape for proton tunneling, environmental modifications that shift the equilibrium between tautomeric forms, or genetic modifications that change the local molecular environment around mutation-prone sites. All of these represent conceivable approaches to addressing genetic disease at its quantum mechanical roots. The path from conceptual possibility to practical therapy would be long and uncertain, requiring advances on multiple fronts. But the direction of inquiry is becoming clearer. Beyond the earthly applications we have discussed, quantum biology may have implications for human activities in space, where the radiation environment differs markedly from that encountered on Earth's surface. Astronauts traveling beyond the protective shield of Earth's magnetic field are exposed to cosmic rays and solar particles that can damage DNA in ways distinct from terrestrial radiation exposure. Understanding how these radiations interact with DNA at the quantum level, and how the resulting damage is recognized and repaired, could inform the development of protective measures for long-duration space missions. If certain quantum effects influence the susceptibility of DNA to radiation damage, or the efficiency with which repair enzymes recognize and correct such damage, then interventions targeting those effects might enhance the resilience of astronaut genomes. As humanity contemplates missions to Mars and beyond, the protection of astronaut health becomes increasingly important, and quantum biology may contribute to the solutions we develop. We might venture, with appropriate caution, into more speculative territory now, considering the possible connections between quantum effects in DNA and the process of aging itself. The gradual accumulation of mutations over a lifetime contributes to the decline in cellular function that characterizes aging, and if quantum mechanical processes influence mutation rates, then they may also influence the pace of aging. Some researchers have speculated that interventions targeting quantum effects in DNA could potentially slow the accumulation of age-related mutations, though such ideas remain firmly in the realm of hypothesis at present. The complexity of aging, involving far more than mutation accumulation alone, suggests that quantum interventions would represent at most one component of any comprehensive approach to extending healthy lifespan. Still, the connections between quantum mechanics and the most fundamental biological processes open conceptual space for considering how aging might eventually be understood and perhaps influenced at the most basic physical level. Looking forward across the next 50 years, we can anticipate a progressive deepening of our understanding of quantum effects in DNA, accompanied by a gradual translation of that understanding into practical applications. The pace of such progress is difficult to predict with any confidence, as scientific advancement proceeds through periods of rapid discovery interspersed with longer stretches of consolidation and incremental refinement. But the trajectory seems clear, toward more precise knowledge of the quantum processes underlying mutation and repair, toward computational tools capable of simulating these processes with ever greater accuracy, toward technologies that detect and manipulate quantum effects in biological systems, and eventually toward medical interventions informed by quantum mechanical understanding. The students entering graduate programs today may witness transformations in our comprehension of life's physical basis that we can only dimly imagine from our current vantage point. Throughout this exploration, we have maintained the perspective that these developments, however significant they may prove, unfold within the ordinary course of scientific progress, without drama or sudden revelation. The quantum mechanics of DNA is not a secret waiting to be unlocked by some flash of insight, but rather a territory to be mapped gradually through patient observation, careful experimentation, and rigorous theoretical work. The researchers who contribute to this mapping are not heroes in any conventional sense, but simply people doing their work with diligence and curiosity, adding their small pieces to a puzzle that will be assembled over generations. This is how science typically proceeds, and there is something rather comforting in that steadiness, in the knowledge that understanding accumulates even when no individual breakthrough captures public attention. And so we leave these considerations of the future with a sense of quiet optimism, an appreciation for the questions that remain and the generations who will address them. The quantum mechanical processes at work within our cells continue their microscopic operations regardless of our awareness. Protons tunneling between energy states, electrons redistributing through molecular orbitals, repair enzymes recognizing damage through mechanisms we are only beginning to understand. This activity proceeds now, as it has throughout the history of life on Earth, forming the physical foundation upon which heredity and evolution rest. Our growing ability to comprehend these processes in quantum mechanical terms represents simply another step in the long human project of understanding the natural world. A project that will continue long after the particular questions we have discussed are settled and new questions take their place. The night continues around you as these words fade, and the science we have explored together becomes part of whatever dreams may come. Perhaps fragments of quantum tunneling and DNA repair woven into the strange narratives of sleep. The molecules of your own cells carry on their work, untroubled by any need for your conscious attention, maintaining the integrity of your genome through mechanisms both classical and quantum. Tomorrow the sun will rise on a world where research continues, where understanding deepens, where the quantum foundations of life gradually yield their secrets to patient inquiry. But for now, there is only this moment, this breath, this gentle settling into rest.